صباح الخير للجميع. تحياتي دكتورنا. It's not it's on YouTube. Wait a minute.
صباح الخير.
صباح الخير جميعا.
صباح الخير دكتور إيهاب. صباح الخير
صباح
يا
الخير
أحمد.
دكتورنا، كيف حال دكتور
الله يبارك فيك،
معتصم؟
كيف حالك؟ شو
كيف
اخبارك؟
صحتك؟ الله
زمان ما
يخليك.
شفناك.
الله يحفظك، الله يبارك فيك دكتورنا.
صباح الخير، السلام عليكم.
السلام ورحمة
عليكم
الله وبركاته.
السلام ورحمة
صباح
الله وبركاته.
الخير يا دكتور محمد. صباح الخير.
يعطيكم العافية. دكتور
الله
يعافيك.
معتصم،
يعافيك.
يعطيك العافية، أهلا وسهلا،
الله
شرفنا
يعافيك.
معرفتك.
أهلا يا دكتور محمد.
حبيبي.
دكتور إيهاب، دكتور ناجي، الله يعطيكم العافية جميع
الله
الحضور.
يحفظك.
الله يعافيك.
إيش دكتور إيهاب، نبدأ ولا نستنى كمان 5 دقائق مثلا؟ عشان العدد حتى الآن يعني 15.
We can wait 3 minutes، يعني ما فيش مشكلة.
أوكي.
ماشي.
ما فيش مشكلة.
مش عارف the other participants ما عملوش confirmation كلهم.
Yeah. دكتور
بروفيسور
جوليا.
بروفيسور هوتي
Mm.
from Italy يعني.
Yeah.
هي جوليا ولا جيليا؟
جوليا. As they have said to us.
جوليا. كويس.
جوليا وهم امبارح كانوا موجودين يا دكتور، يعني إن شاء الله اليوم راح يكونوا
إن شاء
موجودين.
الله.
ماشي. أنا يعني بس كنت حابب أتأكد إنها موجودة
Mm.
يعني. في فابريزيو كالديرا، في دكتورة مي،
موجود.
دكتورة نسرين، دكتورة فايزة، أعتقد كلهم يعني، بس اللي من إيطاليا ما عملوش كونفirmation.
Yes.
think we can start, Dr. إيهاب.
اوكي،
نبدأ دكتور إيهاب.
تفضل على الله.
تفضل.
بسم الله الرحمن الرحيم. Good morning everyone. Dear respected participants, colleagues and guests, it's my great pleasure to welcome you to the second day of the third International Conference on Pharmaceutical Sciences. We are pleased to begin our program today with the chemistry session, which includes a number of exciting scientific presentations. Now it's my honor to invite Professor Ihab Al-Masri and Professor Najid Dahoudi to moderate this session. Professor Ihab Al-Masri is the Professor of Medicinal Chemistry at the Faculty of Pharmacy and the Dean of Postgraduate Studies at Al-Azhar University of Gaza. Professor Najid Dahoudi is a Professor of Physics and Material Sciences Engineering at the Faculty of Science, Al-Azhar University of Gaza. So, Professor Ihab and Professor Najid, the floor is yours.
شكرا دكتور محمد. بسم الله الرحمن الرحيم. نبدأ على بركة الله وتوفيقه تنطلق فعاليات الجلسة الأولى من اليوم الثاني للمؤتمر الدولي الثالث في العلوم الصيدلانية، متمنين التوفيق للجميع. Distinguished guests, esteemed colleagues and fellow researchers, good morning and welcome back. Welcome to the first session of our second day, which is focused on pharmaceutical chemistry and drug discovery. My colleague, Professor Najid Dahoudi, and I are honored to moderate this virtual session today. You are welcome to submit your question through the chat box at any time or raise your hand at the end of presentation. It is now my distinct privilege and personal honor to introduce our first keynote speaker, who was also my master and my PhD supervisor, Professor Mu'tasim Taha from the University of Jordan. Professor Taha is a professor of medicinal chemistry and drug design and the head of the drug discovery unit at the University of Jordan. His pioneering research focuses on computational drug design, molecular modeling, and novel polymeric materials for drug delivery. Today, he will present on a highly advanced topic, structure and data-driven kinase inhibitor discovery, integrating pharmacophore modeling, docking, and machine learning for lead identification and optimization. Please join us in welcoming Professor Mu'tasim Taha. Dear Professor Mu'tasim, the virtual floor is yours.
Thank you very much. I'm honored to be with you in this conference. Assalamu alaikum wa rahmatullahi wa barakatuh. Bismillah. My talk today,
Can you share your presentation?
I will
Your
share
screen.
my presentation first. I think, yeah. Can you see it now? No? No.
Yes, yes.
That's okay.
It's okay.
Yeah, it's clear now?
Yeah.
So I'll be talking about the research that we have been conducting over the last maybe around 20 years. It deals with structure and data-driven kinase inhibitor discovery. The research started early, maybe 2005 or 2004, and we have several milestones as we go through until now. It started with simple ligand-based modeling coupled with QSAR and drug repurposing. We started with targets like GSK 3-beta, glycogen synthase kinase 3-beta. And as we go through, we started modifying the technique. For example, in 2010, we worked upon CDK1, and it was also ligand-based modeling. In 2016 to 17, we started working structure-based methods like docking-based comparative intramolecular contact analysis. This is a method that we developed ourselves here. So, and later on, we started combining machine learning methods together with ligand-based and structure-based methods. Like in the 2022, we published work on CLK4 And more recently, we started to carry out something called augmentation, data augmentation, whereby we use docking poses generated by docking to augment data, especially in cases whereby you have limited number of known ligands, like in the case of so-called dark kinome or dark kinases with limited studies. And this is until now. So we recently published work using augmentation and regression machine learning on CDK16, which is also one of the dark kinases. That is kinases that are not very well studied. So it's a long study, a long interval of time, and we published nearly 200 papers in this area in major journals. Do you still hear me or not?
Yes.
Yes.
Okay.
Okay. So the first question, why kinases? Why we are interested in kinases? The drug discovery challenge. Why we need to integrate computational framework, different methods, ligand-based, structure-based, machine learning, pharmacophore modeling, docking, and so on. Why do we need to have that? Why do we need to focus on pharmacophores and pharmacophore modeling in particular? I will be talking also about pharmacophore modeling, whether ligand-based or structure-based. Also, I will be talking about machine learning and data augmentation. I will talk about some examples, recent examples, and eventually, finally, conclusions and future directions. Why kinases? The kinase, human protein kinase, about 518 kinases, which are considered to be numerous, really. The drug discovery market is about 3 billion. So it's an active area of research. There are more than 80 known FDA-approved kinase drugs currently used usually for cancer as well as other inflammatory diseases, autoimmune diseases, and so on. Many of the kinases are linked to cancer. This is one of the kinases here, which is FGFR1. This is involved in cancers, certain types of cancers. As you can see, the kinase can exist in two states. This is the active state, DFGN, where it can receive client proteins here, at this yellow part here. And it can receive ATP here between the hinge region and the glycine-rich region. So it receives ATP from this side and receives client proteins from this side, and phosphorylation happens. Kinases phosphorylate other client proteins to activate them. This is the active state, DFGN, and this is the inactive state, whereby the activation loop is going outside. We call it DFG out. This is the DFG region here, aspartic acid, phenylalanine, and glycine in this region here. So we have DFGN and DFG out. This is the active state, this is the inactive state. Kinases are really flexible proteins, and they, as you can see, they can exist in at least two states, active and inactive. So this actually adds to the complexity of kinases and adds to the challenge of drug discovery for kinases. The kinase region and contains, as I said, many drug molecules. The earliest is the imatinib here. And also we have sunitinib, ibrutinib, and as I said, there are about 80 different FDA-approved kinase drugs currently in the market. So key challenges in kinase drug discovery, we have the active and inactive states, as I said. The ATP binding site is highly conserved. And if you block one kinase, it's really hard not to block other kinases. So selectivity is an issue to select the disease-causing kinase from others. Also, kinase medicinal chemistry involves lots of activity cliffs. Activity cliffs are molecules that are very close in structure, nearly identical, but still they have a great difference in affinity. Activity cliffs can really complicate drug discovery efforts, and they complicate classical QSAR. There is continuous need for novel chemotypes, and there are so-called dark kinases or dark kinome, which are underexplored. Yeah, many of those kinases, for example, out of the 518 kinases, there are about 150 dark kinases. That is kinases that have limited studies or that are explored to a certain limited extent. Here, why do we need to integrate ligand-based, structure-based, and data-driven modeling? Kinase ATP pocket combine conserved hen region and target specific subpockets. And they have conformational states, separate conformational states, and solvent effects. So we have lots of issues regarding the ATP binding pocket, which means that you don't have a single method is sufficient to build active inhibitors. So ligand-based methods miss residue level context. They miss the binding site context. While docking alone can misrank poses. Sometimes docking engines produce poses that are not in the correct rank. Static crystal contacts can be either too restrictive or too promiscuous. From our experience, if you use static crystallographic images to build pharmacophores, they tend to be promiscuous. They tend to capture numerous molecules without selectivity. So therefore, lead discovery required us to combine different methods together, whether they are ligand-based, structure-based, and so on, docking, machine learning, and so on. So the basic method that we use, so the recurring solution is the integration of pharmacophore exploration, and those pharmacophores were either produced by docking or by ligand-based methods with machine learning and experimental feedback. So what we do really is to generate pharmacophore models starting from either binding sites or from ligand-based or structure-based, and then you allow those pharmacophores to compete with each other within machine learning or QSAR settings to find the best model that can explain the observed bioactivity. Okay. So the question now is why we are interested in pharmacophore models? Pharmacophore models represent the basic features that are required for binding. As you can see here, for example, blue spheres represent hydrophobic regions. Green vectored spheres represent the hydrogen bond forming regions. So if you know what are the basic features required for binding and how they are dispersed in space, how they are arranged in space, you can actually you can find molecules that are totally different from the original known inhibitors. For example, in this case, as you can see, those are inhibitors. This is not a kinase paper anyway. This is it's pseudomonal quorum sensing inhibitors. Pseudomonas bacteria tend to combine with each other and form biofilms and collaborate with each other by something called quorum sensing. So these are known quorum sensing inhibitors. If you put them together, align them against each other in a ligand-based sense, you can identify those common features, hydrophobic regions and hydrogen bond-forming regions, and forbidden regions or exclusion spheres, those gray ones here. If you pick this pharmacophore model and you use it as a search query to screen molecules, you will identify molecules that are totally different chemically from the starting molecule. So pharmacophore models have this advantage of allowing you to hop, to scaffold hop, to jump upon scaffolds, to change the basic scaffold of the of your training or your starting compounds. That's why we are interested in pharmacophores mainly. As I said, pharmacophore modeling has two basic strategies, either ligand-based or structure-based. In ligand-based, you generate pharmacophore models from diverse subsets of active compounds. Usually, active compounds are converted into multiple conformers, and then those conformers are from different active compounds are aligned against each other to identify common features. Then you validate pharmacophore models, usually using ROC analysis, which is to test the ability of the pharmacophore model to separate active from inactive decoys, active compounds from inactive decoys. Then the valid pharmacophores are allowed to compete within either genetic function QSAR or genetic function machine learning settings against a large list of training and testing sets. We might augment the training and testing sets. Augmentation in data science means to create data from the to create more data from your limited list, and we do that by creating different conformers from the ligands. So by doing so, you increase the number of training molecules and testing molecules, and by doing so, you can build satisfactory machine learning models. We have many examples on that, as I said, for example, Aurora A kinase, CLK4, CAMK2, and so on. Numerous examples on ligand-based methods. Some examples are recent and some are older than that. Structure-based methods, on the other hand, start either from crystallographic complexes, dot complexes, or MD simulations, molecular dynamic simulation frames. So you start from structures. Even you can start from homology models or AlphaFold models, for example. Models, virtual models of proteins, if you don't have crystallographic images, and so on. So you either generate pharmacophores from receptor-ligand complexes, virtual or crystallographic, or you regenerate contact fingerprints. When the ligand binds within the receptor, it forms like contacts between the ligand and the receptor, and those contacts are like fingerprints, very specific for each ligand and for each conformation of that ligand. We then allow the pharmacophores or the contact fingerprints to compete within genetic function algorithm QSAR or machine learning models. We might augment the training and testing set, as I said, by multiple conformers. We perform shape analysis. This method is used to identify the significance of different features within a machine learning model, different pharmacophores or contacts, something called shape analysis. And we have numerous examples of that. For example, STAT3. STAT3 is not a kinase, but still it's a very interesting target, transcription target. We have also published on HER2 and PI3K-C2 alpha and so on. Structure-based methods. So ligand-based pharmacophore models are generated from ligands. Structure-based from protein ligand complexes, whether crystallographic or virtual or even molecular dynamic simulation frames. And then we allow the resulting pharmacophores are allowed to compete within machine learning settings. This is the basic framework of our work. Here, one example or example workflow, ligand-based pharmacophore modeling and QSAR. As you can see, we start with certain number of ligands. We generate several pharmacophores from the ligands, and then we combine them with physicochemical descriptors and allow them to compete within machine learning settings. Here, the several machine learning models were evaluated. The best were random forests and XGBoost, and these were used to select the best pharmacophores. The best pharmacophore was also validated by ROC, used as virtual screening, and hits were tested experimentally. 3 hits showed low micromolar and submicromolar IC50 values. This is against Aurora A kinase. Here an example on docking-derived pharmacophores, structure-based pharmacophores. As you can see, 6 ligands were docked into the binding pocket of PKN2, and these were used to pick the 360 top docked poses, and these were used to extract pharmacophores. These were allowed to compete within GFA QSAR, and the best one was used to screen the NCI database to find active hits. We could identify 3 low micromolar leads. PKN2 is one of the dark kinomes. It is of limited exploration. As I said, the study involved augmentation, data augmentation. Also in structure-based methods, we were interested in contact-centric modeling, whereby ligands are allowed to fit in the binding pocket, and then we collect the contacts that exist between the binding site and the ligand. Those contacts are then either used as ligand receptor fingerprints or used for something called docking-based comparative intramolecular contact analysis, or DBCCA. So here you dock each ligand into the kinase binding site, numerous poses. Then ligand receptor contact fingerprints are collected. Then you train machine learning model on augmented dataset. The machine learning model picks the most critical contacts between the ligands and the binding site. Those contacts can be used to build pharmacophore models. So simply, you pick those contacts that correlate with activity. Not all contacts between the ligand and the binding site really are significant or are involved in binding. Many of the contacts are useless. So what we are interested in the most significant contacts that can correlate with affinity, with reported affinity. Such contacts can be used to build pharmacophore models. in DBSICA, not only we were interested in the contacts, we are interested in which contacts can be summed together, summation of contacts, because affinity is something that comes from a collection of contacts, from cooperativity of attractive interactions. So we're looking in DBSICA for those contacts that their sums together correlate with affinity. This is the DBSICA, as you can see. Several molecules are allowed to dock into the binding site. We identify the contacts between each of those ligand and binding amino acids, as you can see here. 0 for no contact, 1 for contact. We identify the contact to be of a distance of 2.5 angstroms. And then we sum up, we start using genetic function algorithm to pick up columns and allow them to sum. We sum them up, those contacts that were picked by genetic function algorithm, and we start searching for the summations that correlate with affinity. Such summed contacts can be used to build pharmacophore models, as you can see here. Or we can pick the contacts, as you can see. We allow different ligands to dock. Then we build contact fingerprints, and we move them to machine learning settings. And then we identify those contacts that control affinity through shape analysis. Then we use them to build pharmacophore models. The major machine learning algorithms that we normally use are random forests, extreme gradient boosting, XGBoost, gradient boosted trees, bagging, support vector machines are very successful also. K-nearest neighbors, naive Bayes neural networks as well. We also use genetic function algorithm to pick significant features within any machine learning model. Shapely analysis to identify which features and how they are related to activity. And we go for consensus docking scores, and we validate our models by ROC AUC, by Matthew coefficient, leave one out cross-validation, and Y scrambling. We also became interested in molecular dynamics-based pharmacophores as well. Here, we have two ideas, really, two general methods. We either use molecular dynamic simulation or similar methods like simulated annealing, or we also developed a method called stochastic deformation relaxation, whereby the complex is allowed to, we deform the complex, the ligand receptor complex, and then we allow it to relax back again. We use those perturbation-aware methods to build perturbation-aware pharmacophores that are related to molecular flexibility. As you know, proteins and biological complexes are not static. They keep perturbing, they keep moving around, and they keep changing their conformation. So we use this idea to build pharmacophore models. We have two approaches. As you can see here, these are the perturbing forms of the complex in the middle here. We either use molecular dynamics or simulated annealing, or our method, deformation relaxation approach. And we either monitored which of the contacts that are resilient enough to stay, to keep valid during the simulation, as if you are shaking the complex heavily, and any contact points that remain resilient during the dynamic simulation or during the simulated annealing or deformation relaxation method. Any of those contacts that remain resilient are considered to be valid pharmacophore contacts. Or you go through the trajectory, the molecular dynamics trajectory, and you generate pharmacophores across the trajectory, and you monitor which of those pharmacophores has got the best ability to distinguish actives from inactives. The best pharmacophores are then allowed to compete within machine learning context. This is another way of looking at dynamic-based simulations. So we published also several articles in both methods, which are, we call them perturbation-aware pharmacophores. As I said, في عنا اللي هو the pose augmentation approach, whereby multiple docked poses are used to augment data in case of limited data. Augmentation in machine learning and deep learning science is whenever you create information if you have limited number of information. So if you want to machine learn a system to distinguish, for example, a dog from a wolf, and you have limited number of dogs and wolves, you can take several photos for the dog and several photos for the wolf and use them to train your machine learning model. So this is called augmentation. We used multiple docked poses or multiple conformers as a method for data augmentation, especially since we deal with dark kinases of limited explored structures. بالنسبة للvalidation, regarding validation, we usually validate our models using ROC, receiver operating characteristic curves. These are for the pharmacophore models. These methods distinguish the ability or identify the ability of the pharmacophore in distinguishing actives from inactives. Also, we validate our models by our machine learning models by cross-validation metrics like accuracy, Cohen's kappa, predictive R-square, and so on. The feature selection, we couple it usually with interpretability using shape consistency and applicability domains and so on. Also, we check the novelty of the active hits. We usually check, accept or publish the active hits that represent real scaffold hopping that are outside the known chemical space of the training compounds. For example, these are hits for HER2, and as you can see, they represent significantly different molecules from the rest of the training compounds, as can be illustrated here using principal component analysis. Okay, we have several case studies. I will go through them very quickly. Here, as you can see, this is for TTK, dual specificity kinase. As you can see, the training compounds were different totally from the hits. One of the hits is quite significant. Another example here, CLK4, which is involved in cancer. This is CLK4. Once again, you have different the training compounds. This is one of the training compounds that was used to build the pharmacophores that we found later to be significant. And these are the hits. one of the hits is really potent. It's a nanomolar inhibitor, experimentally, that is. Aurora A kinase is another example. STAT3 is a transcription factor. It's not a kinase, but still very interesting. We implemented docking-based augmented machine learning and molecular dynamics derived pharmacophores, two different approaches and two different articles, really. And so on. Here, state 3, this is the dynamic approach. And quickly, you pick up a complex, you generate molecular dynamic frames. Those frames are then converted into pharmacophores, and the pharmacophores are allowed to compete within machine learning settings against known STAT3 inhibitors.
Yeah, Dr. Mu'tasim, can you finish in 2 minutes, please?
Okay. Okay, I finished actually. This is the HER2 example and so on. Future directions, we are now involved in deep learning, not only shallow machine learning, AI and deep learning, graph neural networks. We are also interested in isoform-aware selectivity design. Kinases can exist in many isoforms as well, so we would like to go for selective isoform inhibitors. And we are also interested in expanding the dark kinome analysis. Eventually, we would like to go into PROTACs, modeling of PROTACs. In conclusion, the integrated framework of combining pharmacophore modeling, flexible docking, and data augmentation can successfully identify novel kinase inhibitory leads. Docking pose augmentation expands training site datasets up to 100-folds, which improves the robustness of the resulting machine learning models and their generalizability. We can use ligand receptor contact fingerprints and shape analysis to bridge machine learning model interpretation and pharmacophore hypotheses. We have many examples. I mentioned 7 cases, 7 target kinases. And it can be used and explored into the unexplored dark kinases as well. Scaffold hopping capability enables circumventing or going around IP restrictions and addressing drug resistance in oncology. Thank you very much.
Thank you, Professor Mu'tasim, for your valuable scientific presentations. Now we have 5 minutes for questions, so please raise hands if you have one. Any question? Dr. Muhammad Taha.
Many thanks, Dr. Mu'tasim, for the interesting presentation. So I am not expert in pharmaceutical chemistry and drug design, but my question that, as you know, that many drugs have off-target effects. So can by this strategy we avoid or minimize these off-target effects?
Yes. Yeah, you can. In fact, if you select a particular ligand and you build your pharmacophores against that ligand, then part of the study is to study, part of the analysis is to also carry out pharmacophore modeling for off targets. And then you pick a ligand that maps, that fits the targeted pharmacophores and avoids off-target pharmacophores. Yes, of course, selectivity is an issue. And normally we test our hits against off-targets as well, or close analogs or paralogs. Any of those kinases, if you pick any kinase, you check it in the UniProt site, you will find a list of paralogs or homologs, other related proteins. and that you can also model and see that your hits will not fit in the other ligands, the other proteins. So of course, selectivity is an issue and it has to be checked. And usually we do that as well, yeah.
And second question, please. So you also mentioned for the validation, it's a computational method. And after many slides, you mentioned the IC50 for some hits. So can you just let me know how you do the experimental validation for your targets?
What we normally perform, we first pick our ligands, acquire them. We get the ligands, either we acquire them by purchasing them or by getting them from the National Cancer Institute as a free gift. You get the molecules and then you screen them at 10 micromolar against your protein.
Okay.
For example, against the HER2 or Aurora A kinase or whatever the target is. The kinase requires the ligand is first placed with the kinase, and then you add the substrates, the ATP, as well as the client protein that it gets phosphorylated. And then you monitor the concentration of ATP or ADP from the reaction. You compare that to uninhibited control, and you identify the inhibition percentage at 10 micromolar of the ligand, of the captured hits. Then you carry out something called dose response. As you can see, maybe you notice that we have some sigmoidal
Yes.
curves.
Yes.
These represent the dose response curves, experimental dose response curves that we use to identify IC50 values. So it involves experimental bioassay details other than the computational part. Yeah, of course.
Thank you, Dr. Mu'tasim.
Thank you.
Okay, Dr. Mu'tasim, I have questions, please. You focused during you talk about the importance of scaffold hopping. Could you clarify why it's important to have scaffold hopping in drug discovery?
Yeah, scaffold hopping is important for several reasons. First, for going around or circumventing or going around the property rights, the patent rights. If a molecule is known to be owned by a certain company, or you would like to have another molecule valid against that particular target, you would like to go outside the patent rights, the space, the molecular space of that particular company. So you carry out scaffold hopping. You jump away from their molecule. Also, scaffold hopping is important for pharmacokinetic purposes. You might have a chemical structure or chemical scaffold of limited oral bioavailability or of certain pharmacokinetic profile, may be eliminated mainly by the liver or by the kidneys, does have limited access across the blood-brain barrier, and so on. And you would like to have a different pharmacokinetic profile, maybe enhanced oral bioavailability or elimination by some other route to treat, for example, an individual with a liver failure. So you need a molecule to be eliminated mainly through the kidneys. Or you treat somebody with a kidney failure, you would like to see a molecule that is eliminated mainly by metabolism. So to go around pharmacokinetic issues, one of the methods is to carry out scaffold hopping as well. So scaffold hopping is a general method to, as I said, overcome patent rights and to sort out pharmacokinetic issues. Sometimes it could be useful for reducing the cost of production. If the original molecule, the first molecule to inhibit a particular target is so expensive to produce, you might go for another molecule that is cheaper to produce and so on. Yeah.
Thank you, Dr. Mu'tasim. And also, okay, we can also may get rid of certain side effects for the previous. Yeah.
Right.
Thank you very much. Thank you very much, Dr. Mu'tasim, for your valuable lecture. Now we can go to the second lecture on our schedule. My colleague, Professor Naji, will introduce the second speaker.
Thank you.
Thank you.
Thank you, Professor Taha, for this invaluable theme for this conference. Good morning, everyone. I would like to welcome all of you in the ICPS 3. We will move to the second talk today with Professor Giulia Huti from the Department of Chemistry, University of Torino, Italy. Her talk will be titled pH-sensitive dextrine-based bioporous polymers for drug delivery, cross-linkage effects on swelling, rheology, mucoadhesion, and drug release. Professor Huti, the microphone is with you. Please, you can start for 10 minutes and we will have 5 minutes discussion. And you can share now your slides. Please, welcome, Professor Giulia.
Good morning, Sabah El here. Sorry for misspelling, but just we have learned some Arabic words.
Yes.
Okay, let me share my screen and okay, can you see my screen now?
No, still not.
Yet. Sorry, I just will.
Yes, now.
Okay. Can you see my screen now?
Yeah, now it's okay.
And
you can start.
Can you please just tell me if I move to the other slides, if it's fine, so you can see now I'm in the second slide, third.
Yeah, it is okay. Perfect.
Okay. Hello, everyone, and thank you very much for the kind introduction. It's a great pleasure to be in such a very interesting and very well-organized event, and especially in kind of particular circumstances. And I'm very happy to speak for my topic that is like more from the synthetic part of from the chemical point of view that leads towards the pharmaceutical applications. And I would like to thanks too much the organizer, the university, for this kind of a great opportunity. As I'm introduced, I'm part of Department of Chemistry, University of Turin, Italy, and I'm going to give some general knowledge regarding to our polymers and which are the properties that can be affected and that can be used towards the various applications. We are a team that usually work on those, and I'm very grateful to all of them. So I would start first to tell you why, or to discuss together why the drug delivery system matters. Because usually the challenge in the modern therapeutics is not only the drug itself, but how to deliver it effectively. The challenges are usually the poor solubility, the low bioavailability, the short duration of action, the high side effects, and many others that can lead to the limitations of the drug delivery. Therefore, our group's aim is to provide more advanced drug delivery systems which can provide more controlled release, improve the bioavailability of the drugs, to minimize the side effects of the drugs, and of course, to lead to the better therapies and better lives. But seeing that the clinical need is to improve the therapeutic efficacy and to enhance the patient compliance, we usually are focused in the biomaterials that are safe, biocompatible, and biodegradable. Which building blocks we usually use? We usually use the starch-driven materials which are sustainable, can be easily functionalized, and are renewable. Specifically, we are focused on the cyclodextrin and dextrins, which are very well known for their inclusion complexes, molecular recognition, and drug encapsulations. But the outcome of this, how we use, is the polymeric nanosponges that we modify the cyclodextrins, which are the cross-linked hydrogel networks that provide a more advanced drug delivery platform. Why we use the biomaterials is because are considered to be designed for interacting safely with the biological systems and providing a more controlled and efficient delivery of therapeutics. And over the conventional well-known materials, they are provided to be more biocompatible. They can be quite easily tunable for adjusting themselves to protect drugs and to provide much easier releases. I'm going to explain in a very summarized way the C3 strategy that we use for our work, the construct, how we do construct the functional nanoporous biomaterials, how the structure property relationships control the performance of our materials, and how those materials can be used further as a future therapies. The main block that we usually are focused is the cyclodextrin, that are oligosaccharide that can host small molecules due to the peculiar structure that has outer hydrophilic surface and the less hydrophilic inner surface. Thanks to this toroidal cone shape, it can encapsulate various molecules as gas molecules, improve their property, and also lead to a more controlled system. But of course, also the cyclodextrins has its own limitations, and we as a team are focused to go to the more like nanoporous sponge system or a cross-link structure by finding the most suitable cross-linking agents and forming a more cross-link structure that it can be ideal for oral, central nervous disorders and other challenging drug delivery applications. Just to give an overview why the cross-link polymer it's used more, it's because all the units compared to the branch and linear polymers, they are cross-linked within each other. Therefore, we have this kind of more stable structure that can host the molecules and then affect also the properties for further usage. This is the mechanism of how the inclusion complex are formed, where the cyclodextrin cavity, those kind of not very well ordered water molecules. Then the moment we put the molecules, it all puts out, expels those molecules water, then to reach a more energetic favored conditions. And therefore, this favorably energetical complex, it's proven to improve a lot of properties of the drugs. The cross-linking as a process itself is the chemical modification of the polymer by using various cross-linker agents. It can be through chemical or physical cross-linking. That chemical cross-linking, it's a covalent bonds forming in the polymer network that in the presence of water, it expands the polymer network. And this swelling, the equilibrium swelling, affects the properties then for the application and also the mechanical stability, two key properties that are very well known for their applications. We usually use various methodologies for quantifying those parameters. And I will explain more how those properties affect the swelling and cross-linking and mechanical properties. Among years, the cyclodextrin-based nanosponges have been progressed in various generations, from the simple cross-linking of cyclodextrin, going to the functionalization, then stimulative-sensitive cyclodextrin polymers, molecular imprinted polymers, also the cyclodextrin nanosponges functionalized with biological ligands. They have been used in various applications, but the main application that is more well-known is as a drug delivery systems. It's a very synthetic approach that we mix the building blocks with various cross-linking agents, depending from the final structure that we want. We also use water as a solvent. We use also the green solvents, such as natural dipautotic solvents. We also develop the solvent-free synthesis until we reach the homogeneous white powder for further usage. So how do the structure property relationship control the performance of those polymers? Some of them are the swelling, cross-linking density, the pore size, and the drug release. Here it's in the investigation when we synthesize the polymer by changing the molar ratio from 1 to 2 to 1 to 9. In this case, we use pyromellitic dianhydride as a cross-linking agent, so it's an ester nanosponge. And as we can see, more we increase the cross-linking agent to the structure, we create also a more cross-link structure, and then we let less space for penetrating the water inside, and therefore also affecting the molecular weight and the cross-linking density. But even though we increase the molar ratio, we still reach a kind of maximum of the cross-linkages that can be used inside the polymer network. Whereas in the case of the rheology, we understand the same principle, but even though it's another technique. So we use the Flory-Renner theory to determine, based on the equilibrium swelling, the cross-linking density, and we use also the rheological measurement based on the viscoelastic properties to determine the cross-linking efficiency. From the viscoelastic behavior, we have proven that our material, in the presence of water, it expands and forms the hydrogel form, and by having the storage modulus higher than the loss modulus, we have a formation of the gel. Otherwise, if the storage modulus would have been lower than the loss modulus, then we would have just a viscous, not an elastic product. In this case, we have played a bit more with the virus pH conditions and virus cross-linker conditions, and also the building blocks such as cyclodextrin and linear dextrins. We have seen that the pH, also the cross-linker, all affect the swelling. Therefore, we can consider our polymers as a pH responsive materials. A very important property for the further usage in the mucoadhesive formulation and the oral delivery applications. In this case, we have determined some mucoadhesive measurements to prove that our polymers are mucoadhesive polymers, and we have seen a very good behavior in the case of the interaction with the mucin in various pHs, specifically in the intestinal and the gastric simulated fluids. When we did the comparison with the chitosan, that is the most well-known commercial mucoadhesive polymer, through the rheological synergism that we use the rheological measurements to determine, we have noticed that our polymers even provided a more stable properties as a mucoadhesive polymers, because in all the pH, they show the positive rheological synergism, providing a very good interaction with the mucin, whereas the chitosan show the in a very low pH positive synergism, because then by deprotonating more, also it loses the interaction or the properties of the with the mucin. To just show what we use very quickly as a drug delivery system, just one example among a lot of others that we usually investigate. This was part of the European project that I was involved, which was the apomorphine as a molecule, which is known to be used for the neurodegenerative disorders, more specifically for Parkinson's disease. And it's known that the apomorphine has a very low oral bioavailability due to the strong first-pass liver metabolism and the very short half-life. And therefore, we loaded in nanosponges various formulations, and beta-cyclodextrin cross-linked with the PMDA show the highest loading capacity and the very good encapsulation efficiency. And also based on the in vitro releases profiles that we performed in the simulated intestinal and gastric fluids, we observed that compared to the free apomorphine, the polymers provide a more controlled release of apomorphine, showing a very good formulation for the further usage as an option for the oral delivery of apomorphine. Also, we did some physicochemical characterization, which I summarize up just two of them, such as zeta potential and particle size, which provide a very good stability of those formulations. And all these, yes, all these, it's an aim to go towards the clinical translation of our polymers. And of course, we need a very interdisciplinary expertise for providing or for proving that our materials can be used as an option towards applications. And seeing all those, we call those nanoporous point systems that they show very good properties and they can be promising candidates for in vivo studies. I would like to acknowledge our department, the Department of Chemistry, University of Turin, Italy, all the research group for the work done, and also my colleague that is from Palestine, Ibrahim Hussein, that he also was the one introducing to university for this, and to all of you for your kind attention and for all these great organization. Shukran.
Thank you, Dr. Houti, for this interesting talk. So we have one or two questions, please. Please raise your hand.
Shall I stop sharing my screen, or?
You can still have it.
Okay,
as you like.
if you want to see something.
Yeah, yeah, there's some. Is there someone have a question? Yeah, Tasneem. Dr. Muhammad Taha.
So I can start, or Tasneem?
Tasneem.
Because you said Tasneem.
Yeah.
Okay, Dr. Muhammad.
Yeah, Muhammad, you can start, Dr. Muhammad.
Thank you, Dr. Julia, for the amazing lecture. Yeah, it's really interesting. So you mentioned in your talk that there is for the release of the drug from the polymer, it's like polymer swelling. So this means that for the mechanism of a drug release mainly depends on the swelling, or maybe for polymer degradation, or maybe diffusion.
Okay. Thank you very much for your interesting question and also for your feedback. In fact, that's is the main point of this topic, is that by having different molar ratio, we have also different swelling. So what we are trying to understand is, because so far we just chose what was the best that provide the better profile. So what we are currently investigating based on the chemical investigation that we have so far, we want to understand that now how the molar ratio is also affecting the drug release, because the lower molar ratio, they are much more swellable, and of course also the biodegradability would be faster. But as far as we have this kind of good profile also for 1-2 molar ratio, let's say when we have less cross-linker, of course we can keep with that one. So yes, it affects a lot all the swelling and all the pH and everything in the final release.
And please, how can you evaluate the kinetic of a drug release?
Okay, as we do so far is that we are doing or with the membrane dialysis by putting our polymer, and then we check for over time, for days, for weeks, and see how it changes and how it's released the drug.
Thank you.
Thank you very much.
Okay, is there a question from Tasneem, or I have mistaken? Yeah, okay, Julia, I have a question. Did the cross-linkage density increase continuously with increasing the BMDA content, or there was a saturation point beyond which additional BMDA did not produce the effective cross-link?
Yes, thank you very much. Yes, I know that I went a bit quick because I just want to give the overview idea, so to let the part for the discussion. Yes, because we tried from the molar ratio 1 to 2 up to 1 to 10, so we increased till 10 moles of the PMDA, and we have noticed that the maximum was to the 6. So after the molar ratio 1/6, we have a saturation of the cross-linkages with the OH groups that are in the cyclodextrin group. So therefore, more we added, it didn't change. So it just kept stable and then also started decreasing. Then how we realize also from the previous question that we want to understand how much worth it is to keep increasing the cross-linking or to just use the lower one, and also it's also more kind of saving the material in this case.
Did you try your non-sponge for other application like pollution adsorption or something like this?
Yes, sure. We have some articles, not me directly, but the polymers that we are using for the drug delivery are very well known also for some environmental application, for water purification. So yes, for sure, it can be also there for gas encapsulation. We have a lot of work carried out for also not only for the neurodegenerative disease that we are just investigating for the apomorphine. We have tried also for the pulmonary delivery, developing also cream formulation for the transdermal delivery. So yeah, we want to understand how our strategy can be used for various applications.
Yeah, last question from Dr. Khalid Al-Masri, please. Last question. Dr. Khalid? Dr. Khalid Al-Masri. Yeah.
Hello, everyone.
Yeah, تفضل. Please.
Thank you for excellent presentation. I have one question. Do you think this polymer can improve drug targeting and reduce side effect comparing with conventional dosage form?
Thank you very much for such interesting question. To be honest, for the drug targeting, we need to work more to the functionalizing it. That also we work also with our colleague that is from Palestine. He was very good focused on that. So yes, we can use for targeting by functionalizing with a depending from the target and how you want to use. And regarding to the side effects, I think this is the aim why we are developing those formulations and for seeing the real improvement and for providing a more like a value or something that like more true, realistic. For now, it's not that I have something to say because the application is done from our collaborators. But as far as we work towards this, I think, yes, it can be reached. Because so far we have seen very good improvement to the drug behavior compared to the conventional formulation. So I think, yes.
Thank you very much.
Thank you very much for all the interesting questions and feedback.
Yeah, okay. Thank you, Dr. Roti, for your great job, great presentation, and we will move to the...
Thank you. Have a nice day and enjoy the conference.
Thank you. We move to Professor Ihab Al-Masri to introduce the third participant for this session.
Thank you. Our next esteemed speaker is Professor Fabrizio Caldera from University of Torino, Italy. His presentation is about novel glutathione responsive cyclodextrin lipoic acid nanosponges for controlled doxorubicin delivery. Please give a warm welcome to Professor Caldera. Professor Caldera, the floor is yours. Let's unmute.
Hello, good morning. Can you hear me?
Yeah.
Okay. Well, I will just try to share my presentation now.
Yeah.
Can you see it?
Yes.
Okay. Can you also see the second slide?
Yes.
Okay, perfect. Okay, so good morning, everyone. First of all, I would like to thank the organizers for giving me the opportunity to contribute to this conference and As my colleague Julia Hoti said before, I would also like to thank Ibrahim Hussein, one of our PhD students, for introducing us to this event. Okay, so today I will present a new glutathione responsive cyclodextrin lipoic acid-based nanosponge for the delivery, for the controlled release of doxorubicin. Okay, as we know, doxorubicin is a very effective anti-cancer drug, but its clinical use is limited by severe side effects, irreversible cardiotoxicity, and like other anti-cancer drugs, it is not tumor selective. So what can we do to improve cancer treatments? We can maybe chemically modify doxorubicin, or we can find other drugs. We can discover other drugs, but this is a very tough task. It takes a lot of time, a lot of money, and chances of success are very, very low. Or another strategy, we could try to find another way to deliver doxorubicin and similar anti-cancer drugs more effectively, reducing as much as possible their side effects. And this is basically the purpose of this project that I'm presenting today. So what did we think? We thought, let's try to exploit some biochemical difference between cancer cells and normal healthy cells to trigger the release of doxorubicin and direct it mainly to cancer cells. So as we know, one of the major differences that we can observe between cancer cells and normal cells is the concentration of glutathione. Glutathione is a reducing agent, is a peptide that is able to break cleavage disulfide bonds by oxidizing itself, according to the reaction that you see here. So, and we know that the concentration of glutathione in cancer cells, especially chemo-resistant cancer cells, is very high. It's 2 to 3 orders of magnitude higher than normal tissues, which means that we could exploit glutathione as an internal endogenous stimulus to trigger the release of a drug. If we load this drug in a carrier that is made whole together by disulfide bonds. So our goal was to synthesize this new carrier, and we decided to basically combine 2 different materials to get this new material, beta-cyclodextrin and lipoic acid. Why beta-cyclodextrin? Because beta-cyclodextrin is an oligosaccharide, it's biodegradable, it has a good biocompatibility, and it's a cyclic amphiphilic molecule. It dissolves easily in aqueous media, and it has an internal cavity that is quite hydrophobic, so it can host lipophilic molecules like many drugs by forming an inclusion complex. The formation of the inclusion complex is a reversible process, so it means that the drug that is encapsulated in cyclodextrin can also be released. So beta-cyclodextrin has very good encapsulation properties, but beta-cyclodextrin and cyclodextrin and most of cyclodextrin polymers in general are not responsive to glutathione. On the other hand, lipoic acid is a biological molecule that contains a disulfide bond, which can break in the presence of glutathione. And moreover, lipoic acid can form polymers upon heating. If we just heat it at 70 degrees, we can obtain linear polymers that are that contain a high number of disulfide bonds between the repeating units. And therefore, certainly, polylipoic acid is sensitive. It responds to redox environment, to the presence of glutathione, but it has very low, almost negligible encapsulation properties. So the idea was to combine together beta-cyclohexene and lipoic acid to have both the outstanding complexation properties of cyclohexene and the glutathione responsiveness of lipoic acid. To prepare this material, we developed this chemical strategy. First, we functionalized beta-cyclohexene with lipoic acid molecules. CHNS analysis confirmed that we achieved a degree of substitution of approximately 6, an average, of course, degree of substitution. Then the second step was just a mixing of the lipoic acid functionalized beta-cyclohexene with an additional amount of lipoic acid. And then just by heating without solvent, so it's a very green step, we managed to polymerize and obtain this kind of three-dimensional chemical structure made of cyclohexene and lipoic acid. Both solubility tests and some chemical characterization that we performed with TGA, FTIR analysis confirmed the successful formation of a cross-linked structure. And usually this kind of cyclohexene-based polymers that are cross-linked and insoluble in all solvents, we call them nanosponges. This is the term that we normally use. Okay, then we also observed that this nanosponge shows a significant swelling capacity. It swells both in water and DMSO. We used the results from swelling tests to apply the Flory-Renner theory and determine the degree of cross-linking. Then we also investigated the ability of this nanosponge to encapsulate organic molecules, and we used phenolphthalein as a probe molecule to see quickly and easily the ability of the nanosponge to absorb. And as you can see here from these pictures, the pink solution of phenolphthalein is really absorbed and becomes colorless. So it means that the nanosponge has encapsulation capacity. And then more importantly, we evaluated the response to glutathione. We did it using a nanoparticle tracking analysis. And by monitoring the behavior of the nanosponge, we observed that in the presence of glutathione, we have, both particle size and particle number decrease. Whereas nothing happens when the nanosponge is stirred in just water or physiological solution. So it clearly indicates that glutathione can break the disulfide bonds of the nanosponge and consequently trigger the erosion of the nanosponge structure. So after confirming the properties of the nanosponge, we loaded the nanosponge with doxorubicin. We obtained quite a satisfactory loading capacity, and then we performed release studies under both physiological and reducing conditions, and also at two different pH values, 7.4 and 5.5. As you can see here from these release profiles, we observed the highest release under reducing conditions and also at slightly acidic pH, which is a good thing because it mimics better the intracellular
Dr. Fabrizio,
pH.
you have 2 minutes. Can finish, please.
Yeah. It mimics better the intracellular environment of cancer cells. So we confirmed that our nanosponge can release much faster with a faster kinetic doxorubicin in the presence of glutathione and at acidic pH. So we propose a release mechanism that is basically due to two different contributions acting at the same time. The diffusion of the drug through the polymer network and the erosion of the nanosponge structure. So in conclusion, we have successfully synthesized a new carrier combining together the properties of beta-cyclodextrin and the glutathione responsiveness of lipoic acid. In future, we will, of course, try to optimize the polymer formulations and move to the evaluation of the biological activity of this nanosponge, both in vitro and in vivo studies. Thank you very much for your attention.
Thank you, Professor Fabrizio, for an excellent and valuable presentation. The floor is open for a quick 2-3 minutes question. Please raise your hand. Is there any question? Dr. Muhammad Taha, please.
Yes, thank you, Dr. Fabrizio, for the nice talk. So just I have like a naive question. Because you mentioned that when there's increase in the pH, I think there is the increase of the release of the drug.
Yes.
So do you think there is a difference between the pH value inside the tumor cell compared to the normal cell? So in this case, that if there is no difference, this means that the drug release also can affect the normal cells.
Yeah, actually, we observed a higher release at lower pH values, around 5.5. Yeah, we saw from the literature that some kinds of tumor cells, usually there's a slightly acidic pH. So we believe that also this kind of pH responsiveness could be helpful. The pH responsiveness, I didn't tell it clearly, derives from the structure of this nanosponge because it contains some carboxyl groups, COH groups, so they can deprotonate and change depending on the pH of the surrounding environment.
Thank you.
Thank you.
Other questions? No questions, so we can transfer to our next speaker. Thank you, Professor Fabrizio. Very nice. Thank you.
Okay, so we move to the next talk with Professor Maya Ramadan from the Faculty of Pharmacy at the Lazar University. Her talk will be about the analysis of synthetic colorant in pediatric OTCs and exposure risks to children. So, Professor May, you have 10 minutes, and we will have 5 minutes for a question for you, and you can share your slides now.
Good morning, everybody. I want to share now my... I have a little technical problem. Do you see my presentation now?
Yes.
And I can move the slides? also?
You can move the slides.
Okay, thank you.
You can
I will make one.
to show. Yes, it's okay now. You can start.
Okay, thank you. Good morning, everybody. I am glad to be here today, and I would like to express my sincere gratitude for the Faculty of Pharmacy for organizing this conference and for giving me the opportunity to share my work with you. I am Dr. May Ramadan from Al-Azhar University, Gaza. My participation today is entitled Synthetic Colorants in Pediatric OTCs and Exposure Risk to Children. Let us go through a brief introduction about synthetic colorants. Synthetic colorants as excipient designed to enhance visual appeal of pharmaceutical products. Synthetic colorants are favored over natural in industry due to superior chemical stability, high dyeing efficacy, and cost-effective issues. It has potential side effects like hypersensitivity, genotoxicity, and behavioral exacerbations in children with ADHD. Accordingly, regulatory authorities have established strict acceptable daily intake ADI thresholds. In this table here, it summarizes the 8 synthetic colorants involved in the study, and some parameters like E number, LOC-P, pKa values, and the acceptable daily intake ADI values. The aim of the study was to develop a robust solid phase extraction reverse phase HPLC method for a quantification of colorants simultaneously in pediatric OTCs within the Gaza Strip, and then evaluate potential daily intake risk posed to children. Now we will go through the methodology. We have 26 OTCs in this study, which were of different dosage forms: syrup and suspension, chewable tablet, multivitamin gummies. Each dosage form presents a distinct chemical matrix containing active ingredient and a series of excipients. Sample pre-treatment include dilution of viscous syrup and suspension, a grinding and addition of EDTA for chewable tablet to mask the aluminum from colorant slake, and dissolution using heat for gummies to destroy the gelling agent. For extraction, the pH was adjusted to 7. Samples were put in ultrasonic bath at 50 degrees centigrade with frequent vortexing. After centrifugation, the supernatant was transferred for the next step, which is solid phase extraction. For the solid phase extraction, we use a ChromaBond HRXAW cartridge, which is a mixed mode weak anion exchange cartridge. This stationary phase preserves neutral and acidic elements, weak acids of pKa less than 5, and strong acid with pKa less than 1 during loading phase from aqueous matrices. For washing, 2% formic acid in methanol was effective to eliminate the weak acids from the cartridge matrix. The elution followed by the basic 5% ammonia in methanol solution. We will go to the chromatographic analysis. The optimal chromatographic conditions were achieved when the stationary phase was Schimback VPODS column. The mobile phase was a combination of 80 millimolar ammonia acetate of pH 7 and methanol acetonitrile mixture at a ratio of 70:30. The elution setting were gradient elution at a flow rate of 1 ml per minute at room temperature. For detection, the diode array detector was used using different wavelengths, as you see, 420 nanometer, 480 nanometer, 510 nanometer, and 620 nanometers. And the result, as you see here, Here we show the HPLC chromatograms for these 8 colorants and the elution at the chromatography was achieved within 18 minutes. The HPLC chromatogram recorded at selected wavelengths demonstrated effective separation of target colorants. The method is highly selective with no interfering peaks observed at the retention times of the analytes. The next step was the validation of the developed method. The method was validated for synthetic colorants. The calibration curves showed excellent linearity. The correlation coefficient R² was circa 0.999, while recovery values ranged from 72% to 117%, indicating efficient extraction. Regarding the precision data, the method was validated for intra and interday precision, were confirmed by relative standard deviation, the RSD. Values are recorded here, which were below 2%, indicating excellent precision. After that, the developed method was applied for the OTCs. And here we show you the syrup and suspension. We have 20 samples. The 20 pediatric OTC syrups and suspension were analyzed using the validated method. Allura red AC was the predominant colorant. The products contained either individual or multiple colorants. And the figure here presents the highest concentration expressed in milligram per kilogram of each detected colorant through the 20 OTCs examined in this study. We have next the 6 OTCs products, 4 chewable tablets and 2 multivitamin gummies were analyzed. Again, Allura red AC was the predominant colorant. Chewable tablets contained synthetic colorant aluminum lakes. This figure presents the highest recorded concentration of synthetic colorants expressed in milligram per kilogram in the tested samples. And now we go to the main objective of this study, is the application. We then estimated the daily intake of colorants by a child 4 to 6 years old, weighing 18 kilograms, receiving the manufacturer's maximum recommended dose. 2 products contributed substantially to the acceptable daily intake, the ADI, and ibuprofen suspension provided 45% of the ADI for Sunset Yellow, while acetaminophen red syrup provided 35% of the ADI for Carmozine. These findings indicated that medications can be a significant source of synthetic colorant exposure in children. Thank you for your attention, and I welcome any question or comment.
Thank you, Dr. May, for this interesting talk. We have 2 minutes for a question from anyone. Nahid? Yeah, I see Nahid raising.
Yeah.
Yeah.
I have any questions if no one wants to ask Dr. Naji.
Hanadi raised her hand. Hanadi? I will give you Dr. Ehab.
Okay.
Okay, Dr. Ehab.
Thank you, Dr. May, for your nice presentation and good analytical work. If I understand you in the last slide,
Yes.
You told us that one of the colorant was at the maximum limit.
No. It wasn't at the maximum limit. We have the concentrations calculated in the dosage forms, all dosage forms. for the colorants which are present. Then there is another calculation. We will go to the maximum recommended dose. How much can a child take daily? 40 ml from from a syrup or suspension as a maximum dose per day. And when you have this, for example, 40 ml as a maximum dose, then I will calculate how many colorants will be ingested through these 40 ml. And then we have values for the acceptable daily intake. How much is acceptable to be taken daily for body weight? And I know for 18 kilograms a child, what is the maximum acceptable dose daily? And I will calculate how much a child weighing 18 kilograms will ingest through the maximum dose if he administered, for example, 40 ml from suspension of ibuprofen suspension or the acetaminophen syrup. These I calculated for all dosage forms, but these two were significant. We speak about 45% of the ADI for sunset yellow, for example. And we know children have many sources of exposure to colorants. They take a lot of foodstuffs, colored foodstuffs, ice cream, sweets, jelly, beverages, and so on. So they are exposed to many sources. And here we look for the possibility of medication to be a source which can be considered risk. Yes.
Thank you for your clarification. Thank you.
Thank you for
we can take the next question. Okay, so there is no question. So thank you very much, Dr. Mai, for your participation, and we'll move to the next talk.
Thank you
for
having me.
Yeah, thank for you. Dr. Rehab.
Our next speaker is Dr. Nesrine Speta from Mario Negri Institute, Italy. Her presentation is about SAR modeling of bioactivity of chemicals towards protein associated with molecular initiating events of organ-specific toxicities. Please give a warm welcome to Dr. Nesrine. Dr. Nesrine, the floor is yours.
Hello, everyone. Can you see my screen?
Yes.
Okay. First of all, I would like to thank you for having me, and thank you, Dr. Ihab, for presenting me as a fellow researcher from Mario Negri. I would also like to be presented as an alumni from the Faculty of Pharmacy in Gaza. You were one of my lecturers, actually.
Welcome.
Thank you. So as you mentioned, my presentation is going to be about SAR modeling of bioactivity of chemicals towards proteins associated with molecular initiating events of organ-specific toxicities. I know it's so chunky, but let's break it down. So the aim of my work is to build and validate SAR models for the prediction of chemicals' ability to trigger molecular initiating events causing adverse outcomes. So in my work, I am actually integrating two concepts. The first concept is the adverse outcome pathway. And the second concept is the computational part, which is the SAR modeling. In this work, we have investigated 5 adverse outcomes, and we've modeled 29 proteins associated with the adverse outcomes using SARPy software, and we evaluated the performance of the models using external test sets. A brief biological background. We have investigated steatosis and cholestasis in relation to the liver. Steatosis is known for the accumulation of fat in liver cells. We modeled several proteins, among which are AHR and LXR. Cholestasis, which is a condition characterized by impaired bile flow. We modeled BSEP, MRP2 and 4. In kidneys, we investigated the tubular necrosis, which is the damage to kidney tubules. We modeled COX-1 and OT1. In developing brain, we modeled 2 adverse outcomes. The first one is cognitive functional defects, which is an impairment in the cognitive function, like memory and attention. We modeled several proteins, among which the thyroid hormone receptors, alpha and beta, transthyretin, etc. We also modeled proteins related to the neural tube closure defects, which are defects related to the brain and spinal cord in the developing area or stage. We modeled proteins, among which cytochrome P26, histone deacetylase, and others. The first concept, which is the adverse outcome pathway, and as you can see, it's like a roadmap. It starts with the binding of chemicals to the first column, as you can see here, which represents the molecules. So we have a chemical molecular interaction that would elicit a series of events, which is called key events, that will lead to the upcoming or upstream adverse outcome. Here, for example, we investigated the steatosis adverse outcome pathway network, and it says simply that if a molecule binds to PPAR gamma, for example, it will increase fatty acid synthesis, leading to intracellular lipid accumulation, leading to steatosis and fatty liver. We then integrated this to the computational part, which is the modeling part. We built SAR models. SAR models, it relates structural part, we will call it structural alert, to a biological or toxic property under investigation. So it's computational. So what is going to be the input to the software? It's going to be the SMILES. What is the SMILES? SMILES is a representation of a molecule that a software can understand. I can't just give the software the benzene ring as it is. I need to modify it to be understandable by the software. So we can see that the benzene ring is actually represented as a series of symbols. And the SMILES represents a full molecule, while we have another representation, which is the SMARTS. SMARTS is a SMARTS arbitrary target specification. It's a language also, but it specifies the substructure. So a part of the structural of the structure, which is in this case the structural alert. In case we have now the materials and methods. The first step was the collection of biological data and chemical data. Biological data were collected from the published AOPs, adverse outcome pathways, and the chemical data were collected from Kimball 35 database. Then the data collected were used to create the data set. The data sets created needs to be curated, and the step of curation in the case of computational modeling is very important, because there is a proverb, a very famous one, which says garbage in, garbage out. So if you haven't done the curation step very well, you will have a low quality data, then a low quality model. So it's a very important step. And then the splitting of the data. So we will have 100% of the data split into 2 subsets. The first one is the training set, which is used by the software to be trained. And then we have an external test set, which was not used in the training of the model, in order to use it for the evaluation. So the data set will look like the table on the left. It will be consisted of SMILES and the category, as it's a classification model. So the model will classify a compound, whether it's active or inactive. So we have the first table. We will use this table as an input for the modeling software, which is in my case SARPy2. And then SARPy2 will create a rule set, which is the model. A rule set will be column of smarts representation. Here it's written smiles, but it's not, it's smarts representation, and the target and the TP, FP, PPV. Well, here, what does it say? It says that, okay, if you see a molecule that contains this specific smarts, sorry for that, this specific smarts, you will need to flag it as active. And trust me, because this molecule predicted 256 true positives and only 1 false positive with a PPV of 1, which is a high result. Then the evaluation step, which is the usage of the 20% subset as an evaluation set in order to get the performance of the model with data that were not introduced in the training process. So the results, we had a criteria of balanced accuracy, which is the average between specificity and sensitivity, above or equal to 70%, and coverage above or equal 70% on test set as an acceptable performance for the models. So we modeled 29 proteins. 22 out of these 29 proteins performed very well, achieved the criteria. For example, we have one of the several successful models here. For example, the thyroid hormone receptor beta and alpha, the PPAR alpha, COX-1, FGFR2, and glucocorticoids, all complying with the criteria. Why did the other 7 data sets not achieve well performance? Well, we found 2 factors really affecting this, which are data set size and balance. So data set size, we realized that if the data set of the training was less than 100 molecules, then the performance of the model would be very low. It would be bad. So for example, we had the LXR, the SMO, and FGF data sets. They were very small, and therefore the performance was very bad. We can see it in the graph here. There is another concept, which is the balance. The balance is, ideally, I would like my data set to have 1-to-1 ratio between active and inactive, but it's ideally, it can't be achieved in all the cases. So if the data set is highly unbalanced, the performance will actually collapse. There is a statistics, it's called meta-correlation coefficient, which is sensitive for the balance. It will show the more, let's say, it will be sensitive for the imbalance, as we can see in the graph in the bottom. So future need will be the availability of bigger data sets, and not just bigger, well-balanced data set. I did also a functional group enrichment analysis. So I checked the presence of some functional groups in both classes, active and inactive, because it's not logic to just check the presence of a functional group in the active, because there is the possibility of the presence of this molecule also in the inactive rule set. And in that way, it will be not significant to say that, oh yeah, we have aromatic alcohols in the active, so aromatic alcohol will be toxic always. No, this is not the case. That's why I did the functional group enrichment analysis. For example, we found that neural tube closure defects, the aromatic amines and aliphatic amines were highly enriched in the active structure alerts, and this is actually align with the natural ligands, for example, neurotransmitters. Let's say that these functional groups actually form hydrogen bond and hydrophobic interactions with target binding points, and I did some literature review to confirm these findings. Okay, in conclusion, we have created 22 well-performing models out of 29 models. These models were part of the OnTox project, which is a project containing a multi-lab and multi-institutional parties. The models are integrated in a software called Vera to support new approach methodology frameworks. The models also will be used for preliminary testing to prioritize which molecules should go further in testing, whether it's to go in vitro or in vivo assays in toxicology and drug discovery. It's also what is interesting about this work is that it combines the AOP, which gives a interpretability to the predictions. So the models will have a regulatory and scientific acceptance, which is normally not very present in the case of in silico models, as not all of them would provide explainability. Why did you predict this molecule as active or inactive? But this work will say that, okay, it's active because it will detect the binding with this specific protein that will elicit the following key events that leads to the upper or upstream adverse outcome. And this is my work now. Thank you so much. If you have any question, please go ahead.
Thank you very much, Dr. Speta, for sharing your valuable insights in this research. I think that your research is under the toxicological molecular modeling title, yeah?
Yes, you're right.
Okay, we can accept a few questions because we are out of time. Is there any questions? Ahmed Al-Baba? No. We have no questions till now. Dr. Muhammad Taha, and then I can ask Dr. Speta some questions. Dr. Muhammad, please.
Thank you, Dr. Nasreen, for the interesting topic. So I tried to follow up your presentation because I am not expert in pharmaceutical chemistry.
Very specialized.
Yeah. So just my question that what is the biggest challenge or challenges in to transfer what you obtained to be used as a drug in the clinical application?
Okay, to be used as a drug. Well, my models is actually used to predict the toxicological effects of the drugs in the early stage testing. So it's actually now used in a tiered approach. So in the drug discovery, or even when we are introducing a new chemical to the market, whether it's herbicide, cosmetic, etc., it needs to go through a tiered approach of testing. So the models I created along with my team, of course, are used as the first line of testing. So what would happen is they would have a chemical. This chemical will need testing. The first testing will be in silico testing. In silico testing is the use of computational methods to make predictions. Okay, so the first one will be this chemical X is toxic. Then I will actually have this alert next to it. Should I go farther with this molecule or not? Why is it toxic? I will need to make other predictions, and then according to the let's say criteria that is set by the guidelines for the testing, they would specify whether it actually moves to next steps or not. I hope this answered the question.
Okay, Dr. Nasreen.
Thank you,
know, Dr. Muhammad, that the drug discovery process is very take long time and need a lot of money. So the failure is devastating in this situation for companies. So they are doing a lot of preliminary in silico modeling to ensure that the success rate is high and the failure is low. One of these toxicological, pharmacokinetic parameters, bioavailability, all its tests are carried early in the drug discovery process, so that cannot reach the clinical part and then fail. This is will cost the companies a lot of money. This is a very important work. Dr. Speta, you said in your lecture that the parameter PA above 70% is acceptable. Yeah?
Yes.
Okay, 70% on what basis we selected this 70%? Based on the literature?
It was in the literature, and it was also agreed upon the multi-layer approach for the modeling results. Because my work was part of several strategies for the testing. So we had the SAR area, and we have also QSAR, and we have docking. So it was agreed upon the team that we need models that achieve above 70% for the SAR, and others achieve above certain other criteria.
Okay. Obtaining for some targets, there is no success in obtaining a model because you told that it's a highly unbalanced data set. I want to ask, what is the accepted criteria when we can talk that there is accepted balance? A ratio between active and inactive, 30 to 70, 40 to 60?
I would say it depends on the data set itself, because it's not just the balance. It's I realized that the size of the data set has a heavier effect on the performance than the balance. But ideally 1:1, not achievable, but let's say 1:2. So it's actually, it depends on the performance of the model.
Yeah.
Exactly, it's a multifactorial. It's the training set size. It's the chemical space. Because while I was modeling, for example, I realized that I have a model that performed 99% and a coverage of 100%. And I'm like, okay, this is really great. I'm happy, but why? Because it's so perfect. I realized that the chemicals in the data set were contained only two distinct pharmacophore or toxicophore in my case.
Yes.
And this model, yes, theoretically it says that it's performing well, but it can only detect the chemicals that has this specific pharmacophore. So yeah, it's a multifactorial thing.
Thank you, Dr. Nasreen, for this valuable information. Now we will move to our next lecturer. Our last guest speaker in this session is Dr. Faiza Diaba from University of Barcelona, Spain. The presentation is about sustainable photo-induced synthesis of enantial pure beta-lactams with anti-cancer activity. Please give a warm welcome to Dr. Faiza. Dr. Diaba, the floor is yours.
Thank you very much for your presentation. Do you hear me?
Yeah.
Perfect. So now I'm going to
share. Okay.
Assalamu alaikum and good morning. Today I will be presenting some of our last results related to sustainable photo-induced synthesis of non-pure gamma-lactams with anti-cancer activity. N-alkyl tricloacetamides have been widely used in our research group for the synthesis of lactams under radical conditions. In the presence of radical initiators such as IDN or metal complexes, we can generate easily the dichloromethyl carbamide radical, which has got 2 chloride atoms and the carbonate group, and which make it a highly electrophilic radical. But the interesting thing about this radical is that it reacts with all kinds of alkenes, electron-rich, electron-poor, and inactivated alkenes. After addition of the radical to the double bond, a new radical is generated, and this radical then abstracts hydrogen atom under reductive condition in the presence of 3-butylene hydride or 3-methyl-silicylane to generate the corresponding lactam. It can also abstract the chlorine atom under atom transfer radical cyclization in the presence of a metal catalyst to provide the corresponding lactam where all the chloroatoms have been maintained. The preparation of these substrate and alkene tricloacetamide is very easy and is very affordable. There are two ways to prepare these substrates: by acylation of the corresponding secondary amines in the presence of tricloacetyl chloride and triethylamine in dichloromethyl as a solvent, which we use in our research group, and also by using Overman rearrangement of the corresponding tricloacetamidate prepared easily from the corresponding allylic alcohols and the tricloacetonitrile in the presence of a strong base. So this methodology was successfully used in our research group for the synthesis of different kinds or advanced intermediates of natural compound who have been able to prepare the tetracyclic structure of ABCD ring of monoamines D, E, and F. Also from tricloacetamides 2 and 3, we have been able to prepare indole intermediate that were transformed to the corresponding ACDEF pentacyclic core of the phenylene and the ABC ring of hemalensine. More recently, we have applied this methodology also to the synthesis of a tricyclic structure of the 2-deoxy-macropodimine A. All these reactions are under reductive condition, as you see in the presence of tributylene hydride and diene as the radical initiator, but we have been also to use the atom transfer radical condition in the presence of groups catalysts or copper chloride to prepare also the corresponding morphine, as you can see, which were useful intermediates to access the ABC fragment of calcicylene A and the triclic structure of potent immunosuppressant FR901483. More recently, we have been able to, for the first time, from these tricloacetamide, we have been able to carry out the reaction under photoredox condition in the presence of iridium catalyst and under blue lead irradiation. And later, 2 years later, we have performed a more green sustainable methodology for the synthesis of lactams from these substrate in the presence only of a solvent, tetrahydrofuran or 2-methyl-tetrahydrofuran, under violet LEDs. We have demonstrated in this work that the solvent acts as a radical initiator, and not only as a solvent, but also as a radical initiator. With some of these molecules synthesized prepared in our research group, we also evaluate their cytotoxic activity against cancer cell lines, and we have demonstrated, or we have found that beta-lactam 1 was the most reactive against cancer cell line A431. And also we have found that lactam 2 has promising selectivity for the same kind of cancer cell lines in comparison with non-tumoral cell lines 3T3. All these tests were carried out with racemic mixture, but at this moment we decided to prepare enantiomer lactams in order to test their cytotoxic activity and their safety, because as you know, chirality plays a crucial role in biology because biomolecules can distinguish between enantiomers of the same compound. As a result, enantiomer offer diversity in their biological activity, toxicity, and therapeutic effects. And we have an example of the historical example of talidomide, where the R enantiomer has a sedative activity, whereas the S enantiomer has a teratogenic effect. We have another example of natural compound, neplanosine A, which minus enantiomer exhibited significant higher toxicity than the plus enantiomer. So for the preparation of this enantiomer, we have followed the methodology developed in our research group by allylation of enantiomer amine as you can see in this scheme, allylation, tricloacetylation, and then with the triclosamide was submitted to violet LEDs into tumetic DHF alone without nothing, with no additive added, and we were able to prepare the we obtained the two epimers with an acceptable yield and nanomolar ratio. We decided also at this moment to prepare also the enantiomer of these two molecules from the corresponding R enantiomer, which were obtained also in an acceptable yield and in a 1:1 ratio. All these compounds were easily, the two epimers in every reaction were separated easily using column chromatography. Having in hand these four stereoisomer, we then, and after ensuring ourselves that they have no non-hemolytic activity, we started our cell viability assays using two non-tumoral immortalized cell lines, 3T3 and HACAT, and three cancer cell lines, as you can see, A431, HeLa, and MCF7. We used the two endpoints, MTT and NERO, to evaluate the cytotoxic activity. And as you can see, the compound 3 and its enantiomer 4 showed the higher cytotoxicity and say higher activity. With the NERO assays, the compound 3 was more active against the cancer cell lines and tolerable to non-tumoral cell lines. As you can see, also compound 3 stood up as the most cytotoxic, since half maximal inhibitory concentration is lower, especially with the A431 cancer cell lines. The comparing selectivity and disease calculated as the ratio of IC50 of non-tumoral cell lines to the IC50 of tumoral cell lines. Most compounds show low selectivity when compared with the non-cancer cell line 3T3, but compared to HACAT cell lines, the selectivity indexes were generally higher, and compound 3 emerged as the most promising candidate. We have also performed the in vivo assay on zebrafish embryos, and compound 4 showed the greater toxicity than compound 3 in zebrafish embryos, consistent with its higher cytotoxicity in HACAT cells. And both compounds, unfortunately, induced developmental abnormality and were classified as potential teratogens. As a conclusion, among the lactams evaluated, compound 3 emerged as the most promising candidate, demonstrating potent and selective cytotoxicity towards cancer cell lines while sparing non-tumor control, namely HACAT. Future research should further explore structural modification to enhance efficacy and specificity while minimizing adverse effects. This work has been achieved in the Faculty of Pharmacy of the University of Barcelona. I would like to thank my collaborator, Dr. Maria del Carmen Moran and Dr. Elizabeth Teixeira, who participated in this work, with the financial support of Fundación PES Igimpera, and what I would like to thank you for your kind attention.
Thank you, Dr. Faiza, for your excellent and valuable presentations. Now we can have 2 minutes for questions. If there is any question, please raise your hand. I can ask question, Dr. Faiza.
Yes, of course.
What about the toxicity of your allylamine starting material?
What do you mean by allylamine? The starting material.
Yeah.
That we use to prepare.
I think
for example, A and B.
Yeah.
No, we haven't tested them.
Yeah.
We have other works, it's not, this is, you know, since I have got 10 minutes only, I have tried to summarize all things in a short time. But we have done other works. We have done other works where we have compared a lot of molecules, and seems to be that the lactam, the gamma lactams, beta and gamma lactams are more reactive. But it's an idea. Why not? We can test them.
That's good. Dr. Muhammad Taha.
Thanks, Dr. Faiza. Just before my question, can you please go for your last slide?
Last slide.
Yes.
The conclusion, no?
No, last one.
Ah, okay.
The last one.
The last one, yes.
Yeah, this one. Okay, thank you so much. Just I have to take. Okay, thank you. So my question, because in your title is sustainable method. So why it's sustainable method?
Because, you know, it has been since COVID, you know, I am a synthetic chemist, but I work also I also test my, I used to do synthesis, you know. We are working in the laboratory, we do synthesis of natural compound and bioactive molecule. So now, since about 5 years ago, I started my own project with sustainable and green chemistry, because I think, you know, we can do a lot of things. We have to think not only about preparing compound using what is available in our laboratory, but we have also to think about doing this, you know, sustainably. Here, for example, when the reaction that we performed, the last one, where we performed in only the solvent, we don't put anything. We don't put other things. We don't put the photocatalyst, which is, you know, very expensive and not available for everybody. We can prepare it in the laboratory, but we use we don't use diisopropylethylamine. We don't use other solvents that are toxic. And 2-methyl THF is recognized as one of the most green solvents, since it can be generated biogenerated, you know, from transformation of plants, you know. And also we use here we don't use heating. That means there's no energy involved. We use only irradiation with violet LEDs, and the starting material are maybe the starting material, they have chloroatoms, but most of them are maintained in the last molecule, which make it, you know, how do we say that? The material, we don't lose material during the synthesis. You understand? I think we maintain all the principle of green chemistry in this reaction.
Thank you, Doctora. That's our last question. Doctora May Ramadan. Doctora May.
Yes.
تفضل دكتورة مي.
Thank you, Dr. Ihab. This delay for the unmute process. Thank you, Dr. Faiza, for this great talk. This is a target synthesis to produce molecules. An issue of enantiomer synthesis is a hard issue. I have a question about how have you tested the enantiopurity, and you see and you show us in the slides the enantiomeric excess ratio, how much it was, how it was tested, because I am interested in analytical issues.
Yeah. No, because we started with an enantiopure compound. Here we started with a amine, which is enantiopure. The starting material is like chiral pool, you know what we call chiral pool. That means we started with an enantiopure molecule. So after we maintain this stereogenic center is maintained during the synthesis. But after cyclization, we can have the one is stereoisome. This we can have R or S. It depends on the. You understand me? So in the end of the synthesis here, we have two epimers. I am going to show only one. We have two epimers that are considered diastereomer. We can separate easily by chromatography. You understand me?
Okay.
Dr. May, you understand me?
Dr. May, unmute. She is muted. Dr. May. It's muted.
Okay. Thank you. I understand that. You have.
And after that, when we prepare the other one, you can see that this one is minus 102, and this one is mass 105. So they are enantiomer between each other. And here also minus 150, and this one is more 150. I mean here, it's not an enantioselective synthesis. We start from compound, which have no chirality. Here we started with an enantiomer molecule. So in the end, we are sure about the purity of our compounds.
Okay, thank you.
Thank you, Dr. Faiza, very much for your.
I would like to finish with thanking all the organizing committee for accepting my participation and to this Congress, and I wish the best for the University of Gaza, Al-Azhar of Gaza. Really, I wish the best for you. Thank you.
Okay, thank you, Dr. Faiza. Now this is the end of our session. Our deep thanks for all speakers and participants. Dr. Muhammad Taha, now. We can
thanks,
now.
Thanks, Professor Ihab and Professor Naji. We are going now for 10 minutes break, and after that we can join the second session. Thank you.
Thank you all.
علم وضياء يا وطني الأكرم نون قاف فاء كي تبقى الأعظم نور علم وضياء يا وطني الأكرم نون قاف فاء الأعظم راية العلم تسود ينحني لها الوجود ليس للعقل حدود راية العلم تسود أنت الأزهر شامخ علم شمس نور صوت الحق الصارخ نور يعلو نور.
أنت الأزهر شامخ علم شمس نور. صوت الحق الصارخ نور يعلو نور. إننا للعلم قمنا
نبتغي ذات المراد،
إننا بالعلم جئنا
نعتني كل الصعاب.
إننا للعلم قمنا نبتغي ذاك المراد. إننا بالعلم جئنا نعتني كل الصعاب. جيل نصر نحن هيا علمنا أصل الهوية. 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 ها هنا لب القضية، ها هنا لب القضية. نحن جيل التضحيات، نحن جيل التضحيات. ها هنا لب القضية، ها هنا لب القضية. نحن جيل التضحيات، نحن جيل التضحيات. أنت الأزهر شامخ علم شمس نور. صوت الحق الصارخ نور يعلو نور. أنت الأزهر شامخ علم شمس نور. صوت الحق الصارخ نور يعلو نور.
ضيعونا، دائما يركبونا وكسروا خطر لا يكسر خطر. شعره كهري ولا تختفى بإيديا يا معلم.
Good morning, ladies and gentlemen, and welcome to our second session in the second day of the conference, the Pharmaceutics and Biotechnology session. I'm pleased to welcome and introduce our esteemed moderators for this session. We have Dr. Muhammad Talib. He is our colleague and the head of the Pharmaceutics Department at the Faculty of Pharmacy at Al-Azhar University of Gaza. And dear Dr. Rana Abu Taima, she is Assistant Professor of Pharmacokinetics from Al-Zarqa University in Jordan. The stage is yours, professors. تفضلوا. دكتور محمد.
السلام عليكم.
وعليكم السلام. We have Dr. Rana Abu Taima with us.
Yes. Thank you for the introduction and giving me the opportunity to join you for the conference today as a moderator. So I'll introduce our first keynote speaker for the session, Professor Muhammad Galzar Ahmed. He is the principal and dean of the Unopia Pharmacy College and Research Center in India. Professor Muhammad is a distinguished academic researcher and administrator with over 26 years of experience in pharmaceutical education and research. His expertise spans pharmaceutics, novel drug delivery systems, and pharmaceutical research, with extensive contributions in teaching and academic leadership. His presentation today is titled Nanotheranostics in Cancer, Current Trends and Future Perspectives. So please join me to welcome Professor Muhammad. The floor is yours.
Assalamu alaikum. Am I audible? Assalamu alaikum. Am I audible?
You can start, Professor Muhammad. Hello?
Yes, I'm presenting.
Yes.
My slide is visible?
Yes.
My video. No, the camera is there. Once we put on the camera, also you can do. Can I start with the permission?
Yes, please.
Hello. Yeah. Assalamu alaikum. Good morning, all the panel members. First of all, I express my sincere thanks for the organizers for giving me an opportunity to be a keynote speaker on this Congress and conference. So let me start. My today's topic is the nanotheranostics in cancer, the current trends and future perspectives. So I'll be go through my presentation on these following headings. So first, the concept of nanotheranostics, the types of nanomaterials used as nanotheranostics, molecular imaging probes, application in cancer therapy and diagnosis, clinical development, minimum criteria for translation into clinical settings, and the future implications. Cancer continues to be one of the most significant global health burdens, and despite decades of progress, we still largely treat diagnosis and therapy as two separate journeys for the patient, often with a costly delay between them. Theranostics, the term itself, was first introduced back in the year 2002 by John Fraunhofer, and it was represent a major paradigm shift in how we approach patient care. Humanity is suffering from the cancer, which has become the root cause of untimely death of individuals around the globe in recent past. Nanotherapy integrates therapeutics and diagnostics to monitor the treatment response to the enhanced drug efficacy and safety. Ultimately, the ideal theranostic system for cancer operates like a smart autonomous medical guide. It could first accurately diagnose the specific class of cancer, actively image the true homogeneity of the tumor in real time, and apply a tailored treatment directly based on those diagnostic insights, and finally monitor the exact efficacy of the treatment as it happens. By combining molecular imaging with molecular therapy, it could be applied in many aspects of personalized treatment, such as early detection of disease, disease staging, therapy selection, treatment planning. An ideal theranostic system for cancer could first diagnose the type of cancer class, image the heterogeneity of the tumor, and apply a tailored treatment based on diagnostic and imaging results, and finally the monitor treatment efficacy. In this session, I'll walk you through where the field currently stands, the major classes of nanotheranostics driving this process, and the translational challenges we still face, and where I believe this field is headed over the next decade. So if theranostics is the conceptual goal, how do we actually execute it in the clinical settings? And the driving force, the key player is theranostics is actually nanotechnology. Utilizing the particles at the nanoscale level provides the numerous advantages in both diagnostics and the treatments. Nanosystem can measure a large variety of biomarkers in a small sample volume, and same nanosystem can deliver drugs to the tumor site via active ligand or passive targeting. So by engineering our platforms down to the nanoscale level, we gain a unique set of advantages because we are finally opening and operating the same dimensional scale as body's own biological machinery. This allows a single unified nanosystems to perform two very different, highly complex tasks simultaneously. First, let's look at the diagnostic advantage. These nanosystems are incredibly efficient. They give us capability to detect the measure a vast array of biomarkers, and they can do this using only remarkably small sample volume. But the true breakthrough happens system used for detection vehicle. We achieve this tumor-specific delivery through two primary mechanisms. The first one is the passive targeting, and next one is the active targeting. In the passive targeting, we exploit the tumor's own flawed biology, specifically the leaky blood vessels and poor lymphatic drainage surrounding it. This known as the EPR effect, or the enhanced permeability and retention, allowing our nanoparticles to naturally accumulate right at the tumor site. And in the case of active targeting, for even greater precision, we use conjugated surfaces. We effectively engineer the nanoparticle with a chemical key that actively seeks out binds to the specific overexpressed receptors on the cancer cell surface. So we can say that nanotechnology can transform a simple drug into a smart dual action system that finds the tumor reports its status and delivers exactly where the needed most. These nanotheranostics mainly made up of the 4 broad components, that is targeting moiety for specific cellular binding, therapeutic agents, that is for the treatment, then diagnostic agents for non-invasive imaging, then polymer coating or matrix that imparts colloidal stability and provides functional groups for the bioconjugation. Let's look under the hood. What exactly is a theranostic nanomaterial made of? To make this dual action system to work, we essentially have to build a highly customized microscopic delivery vehicle. Generally, this requires 4 critical constructs. So things of it like engineering a smart spacecraft where the stealth shield, that's polymer coating matrix, before we add to the cargo, we need a stable chassis. We use a polymer coating like polyethylene glycol, which acts as a stealth sheet. It imparts colloidal stability, meaning it stops the particles from dumping together to the bloodstream and hides the particle from body's immune system so it isn't cleared too early. Crucially, this matrix also provides the chemical hooks or functional groups we need to attach our other components. In GPS system that is targeting moiety, next we need navigation. We attach the targeting moieties to the surface for highly specific cellular binding. For instance, we might use specific peptide or antibody that acts like a key circulating the body until it finds its exact matching lock, such as an overexpressed receptor or the squamous cell carcinoma tumor. The third one, the diagnostic agent as well as the treatment, the therapeutic agent. Once the vehicle arrives, it needs to do the job. We load it with a therapeutic agent. This could be the powerful chemotherapeutic like 5-fluorouracil safely encapsulated inside so that it doesn't cause severe toxicity and to healthy tissues along with the journey. And the final, the diagnostic agent, finally, we need a way to happen from outside. We include diagnostic agents such as iron oxide nanoparticles for MRI and for a fluorescent dye. This allows for non-invasive real-time imaging to confirm the payload has reached target. Now you might ask, how do we fit all these onto something so incredibly small? This is the true mechanism and the nanoscale, because the nanoparticles have extraordinary high surface area to volume ratio. It provides the countless docking stations to absorb, the dissolve, or covalently attach all these targeting, imaging, and therapeutic agents to the stable multifunctional complex. To understand clearly, let's have a schematic diagram that shows a nanoparticle, the nanotheranostics combination of the diagnostic agents, the therapeutic agents, as well as the nanosystem. You can see in the right side where the therapeutic agents, which can be used as the photosensitizer molecules or chemotherapeutic agents like doxorubicin, paclitaxel, methotrexate, or in the radiotherapy, we can use any of the radio imaging agents. Now coming to the diagnosis, we have the various diagnostic agents, also the scanning methods. It may be the MRI, magnetic resonance imaging, or it may be positron emission tomography, or it may be fluorescence, or it may be photoacoustic imaging. All can be used for the treatment as well as the diagnostic purpose with a nano formulations. The drug delivery system, maybe the different kind of nano formulations can be used. Let us take a single nanoparticle or the nanotheranostics, which combination of all the therapeutic agent, the diagnostic agent, the coating polymer, and it encapsulates the nanoparticle. If we deconstruct a nanotheranostics system, it's as shown in the 2021 review by Sifaka and colleagues. We can see that essentially a highly engineered modular platform. It's a base. We have core nanomaterial itself, which might be gold nano stars, carbon nanotubes, or iron oxide nanoparticles. To make this core safe for the human body and extend its circulation time, it is encapsulated in a biocompatible coating materials like polymers or liposomes. From there, we add the navigation system. We decorate the surface with targeting moieties using biological or chemical agents, ensuring the package specifically binds to the tumor environment rather than healthy tissues. Finally, we load the system to our active agents. We can incorporate radionuclides like copper, gallium, sensitivity for PET imaging, or embedded optical contrast like ICG for near-infrared fluorescence, allowing for precise real-time visualization. Let's have a quick look the types of nanomaterials used as nanotheranostics agents. So take example like the carbon nanotubes, then the polymeric nanoparticles, the dendrimers, the ethosomes, the solid lipid nanoparticles, the quantum dots, and all these can be used as the materials for the nanotheranostics agents. At the top, we have inorganic and carbon. So totally we have categorized two. We have the inorganic carbon-based materials such as gold nanoparticles and quantum dots, and supermagnetic iron oxides, which offer unique optical and magnetic properties ideal for high contrast imaging and physical treatments like photothermal therapy. Conversely, at the bottom, we see the organic lipid-based biological carriers, including liposomes, polymeric dendrimers, and viral vectors, which are highly biocompatible and excel in securely encapsulating complex drug payloads to transport them across the cell mechanisms. Let's have the small understanding of the molecular imaging probes. So basically, the imaging probes are categorized into three. So one is the optical imaging, that is indocyanine green, rhodamine G, quantum dots. Then we have magnetic resonance imaging, that is MRI. So where we are going to use the gadolinium, iron oxide, and cobalt. And we have the nuclear imaging, where we are going to use 64 copper, then gallium, fluorescence, as well as iodine 123. Once we have chosen our nanomaterial carrier, we need to equip with these right diagnostic tools to visualize the tumor, nothing but the molecular imaging probes. As mentioned on this slide, so for real-time high-resolution visualization, often used for surgical guidance, we use optical imaging probes like fluorescent iodocyanine as well as green and highly stable quantum dots. Second, if you need precise anatomical details of deep soft tissues, we really rely on magnetic resonance imaging, loading our nanocarriers with a strong contrast agent such as gadolinium or iron oxide. And last, finally, for whole body tracking with ultimate sensitivity, we utilize nuclear imaging by incorporating radioisotopes like copper 64, fluorine 18, and we can perform the PET or SPECT. So these are the various nanomaterials used in nanotheranostics. Have a quick look with the A, B, C, D I have marked. Where the first one is the polymer drug conjugate, polymeric nanoparticle, and C is a solid lipid nanoparticle, D dendrimers, E liposomes, mycelium, and gold nanoparticles and carbon nanotubes. You can see in the right side, it is marked as the red one is actually the therapeutic agent, whereas this is actually a targeting agent, and the green one is actually the diagnostic agents. Let's quick look for one with examples. The first one is the polymer drug conjugates, where the metal can be used as HPMA. Then therapeutic agent is 64 copper as well as diagnostic 64 copper, and where the targeting agent is arginine, glycine, and targeting agent, and advancement is cancer imaging radiochemistry. The second one is actually the polymeric nanoparticles. It is also having all these moieties. It can be example for the PLA TPGS material can be used. Docetaxel is a well-known therapeutic agent, and the diagnosis is the quantum dots, and the size should be in the 250 nanometer. Folic acid is the targeting agent. We can use the co-delivery. So similarly, one more example is the solid lipid nanoparticles, where we can use the low density lipoprotein and the quantum dots as the diagnostic agent, paclitaxel as the drug, and 130 nanometers the size, and we can use again for the multiple modality. Like that, we can use almost all these nano particles. Combined with that, we can use with the scheme for the treatment for the nanotheranostics. These nanotheranostics are going to work on several principles and basic mechanisms. So these mechanisms are nothing but the pH responsive mechanism, redox reaction responsive, hypoxia responsive, enzyme responsive, and nucleic acid responsive, and small biomolecule responsive. As we know, during the cancer, so a lot of the pathological changes as well as the physicochemicals are going to change, and naturally there will be a greater change in the pH, greater change in the oxygen condition. Based upon these, we can encash and we can make it. So with our nanoscale vehicles, the schematic shows that we can use this platform. Now with these different mechanisms, thus we can design based upon the requirement of particular treatment. We can target by using either pH responsive or hypoxic responsive. Let us take an example of the various applications of this nanotheranostics in different cancers, like we have breast cancer, the lung cancer, colorectal cancer, skin cancer, as well as the prostate cancer. And for all these treatment, we have to decide what kind of nanotheranostics, what should be the drug moiety, and what should be the targeting agent, as well as what kind of the polymer and which one is a suitable nanoparticle. Based upon that, definitely we can target to all kind of cancers by using the nanotheranostics agent. So one example I have taken to explain very clearly how these applications of the cancer in a real time with the help of this one. So here the fluorescence based whole body imaging is widely used in this evaluation. Nanoparticles in small animals often combined with a quantitative analysis of indicating their spatio-temporal distribution following systemic of administration. Now have a quick look of this diagram. So where animals are receiving the different dose of a drug and different percentage of the diagnostic agents, and where we can see in a one group are receiving 0.4 milligram of the agent as well as this the 0.4 milligram of the drug showed weak fluorescence signal in the liver and spleen and no signal in the tumor at all, likely due to the insufficient amount of the dyes in the system. With the next group, where we have taken the 4 milligram with 0.5%, so bright signals were observed in the liver, spleen, tumor, and adipose fat pad in the buttock, irrespective of the surface layer. And in the last group, it's very clearly shown where the 4 milligram of the drug is received with 0.5% showed a fluorescence signal in the liver much brighter than that in the tumor. In contrast to the other group, showed an ever increasing fluorescence signal in the tumor over 72 hours. And for exceeding that, the intensity in the liver. So this shows at the 72 hours, the clear difference between the dose of the drug as well as the specific concentration of the diagnostic agents. The next application where the using positron emission tomography PET imaging to monitor the quantitatively analyze the delivery of localization of nanoparticles, a widely used photothermal agents, is essential to optimize the therapeutic agents. Similarly, the last example of the applications in the cancer, so where to demonstrate the feasibility of the RGD 64 copper with the AU, we are used in in vivo PET guided photothermal therapy. We injected a solution with in a solution where we are going to show the intravenously into the xenograft model in the whole body PET imaging of the mice, liver, spleen, and tumor were all clearly visualized. The photothermal treatment was carried out in 24 hours post-injection when the tumor uptake reached the maximum as indicated with PG. So these are the various cancer applications. You can see the examples in the column wise where the first one is the lipid-based system drug imaging agent, and the detection method is MRI, and a tumor is the soft tissues. Like that, like we have the various treatments for the tumors like melanoma, then TNB is the triple negative breast cancer, and for all these the different liposomes can be used, and the detection methods, so all can be used for the cancer applications. Now several under the clinical trials. So these are the some of the products already started developing and gone for the clinical trials. The one of the example here, the docetaxel, the drug is taken by the company and a clinical phase 1, and the therapeutic modality is actually docetaxel, and diagnostic modality they have made as a PET with a zirconium, and the proposed indication is solid tumors and a CT identifier. Similarly, the other 4 or 5 companies, they started with developing under clinical trials, and once the results are out, then definitely it may be helpful for the next level of the treatment for the actual patients. So similar, some more example for under clinical trials, you can see here the PEG liposomes, where the drug is actually doxorubicin, and detection method is SPECT or the CT, and the cancer, this one is ovary, and clinical phase is early study, and we need to be start. And similarly, the HER2 target PEG liposomes, the PET advanced breast cancer phase 1, and this also undergoing the treatment. Like that, many clinical trials are under trial, and once it's going to be complete all the phases, and definitely it's a boom for the both the diagnosis as well as the treatment. Now, criteria for the translation into clinical settings. So successful application depends on the choosing the most suitable imaging and contrast modalities for the right clinical conditions. Regulatory agencies should update the guidelines for nanotherapeutic platforms in order to facilitate the assessment of efficacy and safety, and ultimately their introduction into the clinical use. Those who developed these nano tools, it might be challenging to simultaneously optimize dose levels and administration frequencies using a single delivery platform. In addition, when designing these formulations, cautions need to be taken so that neither of the components are prematurely released for the delivery system. Drug encapsulation optimization, ligand conjugation efficiency, and high reproducibility with low-cost biomaterials are essential issues for clinical applications of nanotheranostics. For most nanotheranostics developed so far, safety in humans has not yet been completely studied. Nanoparticles should be structurally well-defined, reproducibly synthesizable, and derived primarily from biodegradable and or biocompatible materials with a deep understanding of the mechanisms by which nanotheranostics are cleared from the body and interact with the immune system. Future implications: cancer therapy using nanotheranostics, that is metal-based nanosystems, is at present stepping towards treatment without using traditional drugs. Currently, the use of liquid metal nanoparticles, the nanoparticles and quantum dots for theranostics applications is being widely investigated, but safety is the biggest challenge. The size, shape, surface functionality, and stability of nanoparticles can also lead to cause toxicity on biological systems. Therefore, the proper formulation and manipulation can advance nanotheranostics towards the next generation medicines. So we are stepping towards an exiting paradigm shift from in oncology. We are moving towards the future where advanced metal-based nanosystems might allow us to treat cancer entirely without relying on the traditional highly toxic chemotherapeutic drugs. The exploration of these novel platforms, such as liquid metal nanoparticles and quantum dots, are the best proven maybe for the treatment. However, this immense potential comes with one primary hurdle: the clinical safety, because the nanoparticle's physical properties, its size, shape, surface chemistry can inherently trigger biological toxicity. The path forward not just about the discovering new materials, but about the mastering about them. The ultimate success of nanotheranostics hinges entirely on rigorous, precise pharmaceutical formulations. By mastering the design, the manipulation of these nanosystems, we can overcome these safety challenges, toxicity, and successfully use in the next generation personalized cancer medicines. So with this, I conclude. This is from the references, and thank you for the opportunity.
Thank you, Professor Muhammad. Any questions? يا دكتور محمد طالب. I think your mic is muted, so please unmute.
Yes. Good morning for all, and fully thankful for Dr. Muhammad, Prof. Muhammad. It's nice lecture. And I have a question that I'm seeing in your lecture that some cancers is specialized for these techniques in your why?
Yeah, means now at present actually we have started to develop these nanotheranostics, and initially as a cancer targeted treatment we have started. So therefore, as a development now, some of the cancers they are started as a targeting, and once the advancement completes, maybe in future we can again the studies may going to continue for the targeting of still many more different types of cancer.
Yes, and I'm think that's focusing in HER2 targeted breast cancer. That's
because around 5 to 6 different principles are there. Whatever the cancer is going to happen, based upon it may pH responsive or hypoxic condition or redox reactions. Based upon that, we need to finalize first that cancer is going to happen on what basics reasons, and based upon that, I think definitely we can decide the type of drug moiety as well as the targeting agents. Definitely we can do. That's what my assumption.
Yes, thanks, Doctor, and we appreciate your sharing for this lecture.
Thank you.
Yes.
If no more questions, I thank the organizers for giving me an opportunity to be a keynote speaker on this auspicious session of your ICPS Congress. I wish all the best for your Congress and also for your country. Thank you.
Thanks, Prof. Yes. And now we transfer to the second lecture with one of Dr. Ibrahim Hussein, with one of our students have knowing from many years, and inshallah you are will introduce your lectures as you have from many years. You are very clever and very innovative students. I remember you.
Thank you, Doctor.
Yes, Dr. Ibrahim.
Okay, thank you, Dr. Muhammad, for your introductions. First, Bismillah ar-Rahman ar-Rahim. I'm really happy to present today in front of you and in front of my teachers during my bachelor degree. So today, actually, I'm a postdoc now. I just started a new positions at University of Milan, La Statale. So today I'm going to share my story in design and evaluation of cationic beta-cyclodextrin nanosponges for targeted SERNA delivery in colorectal cancer cells. So first we will start for, just wait to remove this because I cannot see. Yeah, okay. So first I will do a short introduction about the gene delivery. So the gene therapy is a technique for correcting defective genes responsible for disease development, and we can improve our gene therapy by gene augmentations by adding a functional gene and correcting the functional proteins, or we can inhibit the defective gene that's responsible for diseases by using inhibitory sequences. or we can do gene editing by cutting the defective gene and adding the corrected gene, and we can do like gene editing by using CRISPR-Cas9. So we have a lot of RNA-based therapy in our project. We use CERNA. CERNA is a small interfering RNA, non-coding, double-strand. It is short. It's targeted to mRNA and degraded and doing regulated for gene expressions. Thanks to Ferran Milo that explored, and they won a Nobel Prize in 1998 after they noticed that the small double-strand RNA that effective to inhibit the muscles protein in worms. So actually, how does work CERNA? In our normal cells, we produced CERNA by using double-strand RNA and by cutting them by diacer enzymes. And we activate the CERNA by using RNA inhibiting silencing complex and bind complementary with the mRNA and degraded and regulate our gene expression and cell growth. This is in normal cells. And also in terms of cancer cells, we don't have regulations, we have over expressions. So we have to have it externally to deliver it for targeted some genes to inhibit the cell growth. As I said, for the clinical applications, we use it for inhibit the cancer cells and cell growth. And also we use it for gene disorder for inhibit the defective gene. And also we use it for neurodegenerative diseases by inhibit or by stop productions of denaturated protein that affected our nerve cells, as well as we use it for inflammatory and some infectious diseases. So to have a good gene therapy, we need a good gene delivery. How we can do a gene delivery? We have two main methods for gene delivery. We have a viral method and non-viral methods. The viral methods like adenovirus, retrovirus, and lentivirus, and non-viral like biological, organic, and inorganic nanoparticles. In terms of the viral vectors, we remove the viral gene and we insert our targeted gene to deliver it. But there is some limitations with the viral vectors, like it has immunogenicity, and also plus it's high cost and it has limitation for productions. And also the main challenge is not easy modifications to overcome these limitations. A lot of studies the past few years that they were going for and still going for the gene delivery and gene therapy. And as I mentioned, for the limitation, it was a death of 18-year-old boy in 1999 after receiving the adenovirus vectors during clinical studies. But here I have to mention also we have limitations in polymeric nanoparticles, but we can over these limitations by choosing biocompatible polymer or by modified the polymer for targeting and other purpose. So we used nanosponges based on cyclodextrin for a gene delivery, a cationic nanosponges. It's based on beta cyclodextrin. The cyclodextrin, as my colleague before explained, is oligosaccharide, like consists of glucose unit bind with the glucosidal bond. We have alpha, beta, gamma, depends of the number of the glucose unit, 6, 7, and 8, and with a different size of cavity. Cyclodextrin has a hydrophobic cavity that's able to encapsulate the hydrophobic drug and hydrophilic surface. And what is the nanosponges? Nanosponges is a cross-linked nano materials we preformed by preform a cross-linking between the polymers, especially the beta cyclodextrin, and it's preformed for us a 3D structures, nice structures with porous to improve our sustained release, and it's also biocompatible and safe. I have to mention this is important because a lot of studies for the lipid nanoparticles for the gene delivery, we have some limitations with the lipid nanoparticles, like the thermal instability, and it has a prolonged of effect and actions. That mean it has a chance to attack by immune cells, and also it does do the hypersensitivity reactions, as well as it has a high cost. produced. So we perform, as I said, cationic pegylated nano sponges by using chemical reactions. First, we functionalize the beta-cyclodextrin with the cationic molecules, quaternary amine, glycidyl trimethylamino chloride. And then we activate the polyethylene glycol by using carbon diimidazole, and we add it to the cationic beta cyclodextrin. So we have the pegylated cationic beta cyclodextrin that we preform the cross-linked and nanosponge structures by adding 1-to-8 cross-linked carbon diimidazole. So we preform at the end 3 nano sponges, 1 cationic and 2 pegylated, one with polyethylene glycol 2000 and one with TBGS. It's actually polyethylene glycol binding with the tocopherol, the vitamin E, to prove our internalizations. And we encapsulate our cationic nanosponges with a gene by preforming electrostatic interactions. We used IBCAM Cerna. IBCAM protein is overexpressed on the cancer cells. And we used 2 different mass ratios, 1-10 and 1-20. So we made physiochemical characterizations, a lot of physiochemical characterization to prove our synthesis, like FTIR to prove the cross-linked peaks in wavelengths 1720, and we perform a TGA to see our pegylated chain binding by 2-step degradations. And also that's important to characterize and to see the particle size by dynamic light scattering. We noticed the particle size were reduced after we add the pegylated chain. That's mean we reduce the aggregations. And also we have with the narrow bullet dispersed index, and we have a cationic zeta potential from 24 to 28 for all our formulations. So we perform, to prove our encapsulation to the gene, we perform gel electrophoresis for binding and release at 2 mass ratios, 1-10 and 1-20, and all our formulations were able to encapsulate the Cerna gene and release it by using heparin 1000 international unit per ml. And we also prepared a stability test, and our formulations were stable in the BBS at pH 7.2 at physiological pH of the body. And also here we performed another analysis, physiochemical analysis for CHNS elemental analysis to be sure our quaternary amine was binding, and also to be sure about the purifications that we have a 0 sulfur %. That's mentioned for the DMSO. That's mean our formulations were purified well. So we made the biological evaluation for our formulations. We incubate our formulations in non-differentiated CACO2 cells. It's colorectal cell lines, cancer cell lines. And we analyze it by fax machine, by flow cytometry. We noticed here the second pegylated formulation with the TBGS has the highest uptake up to 6 hours, and a good knockdown after 24 hours, up to 50% for the IBCAM protein expressions. And also we performed the cytotoxicity test for all our formulations. We made a cell titer test, and all our formulations were safe up to 24 hours. So as a conclusion, we have a good formulations, pegylated cationic nanosponge that improved our uptake and the knockdown in CACO2 cells. This result, it was a promising results to expand our project and to go further for different studies and in vivo studies. And the last take-home message, and this is important for the stability of the nano particles. Usually when we perform nanoparticles in a small particle size, we have a higher energy in the surface, and it's moving in the random moving. We name it Brownian motions, and to get stuck with each other and perform aggregations, and that's what we don't want. So the electrostatic repulsion and the repulsion force, it's helped to avoid these aggregations, but sometimes the repulsion force is not strong enough to avoid this aggregation. So we should have also steric repulsions by adding a stabilizing polymer chain to our nanoparticles to make a space between the particles to improve the stability in the colloidal system. So finally, I would like to thanks all of you and thanks my colleagues that they were represented today, Julia and Fabrizio, and I'm happy to receive your questions.
Yes, Dr. Ibrahim, thank you for your interested lectures and any questions. And apologize for Dr. Ibrahim, that's Dr. Ibrahim from Turin University of Italy. Any questions, colleague? Okay, Dr. Rana, for the...
I have a question, Dr. Muhammad.
Yes, Dr. Muhammad Taha.
Thank you, Dr. Ibrahim. It's a very interesting talk. So you mentioned for the uptake mechanism.
Yes.
Or the uptake profile. So my question: how did you check the uptake for the drug or for the combination?
Yeah, it's a really nice question. Actually, we used a fluorescence gene, CERNA, and we encapsulated with our nano system, and we apply it to the CACO2 cells. And after that, after incubated for 6 hours and fixed our cells, we tested in the flow cytometry machines that show us how many fluorescence intensity inside the cells. So in this case, we measured the uptake.
Yeah. And the second question, you validated your model by checking the protein, the expression of a protein level.
Yes.
So what about the gene level, the messenger RNA? Did you check that?
What do you mean for the no, because like the gene, it's express the proteins. If we notice the decrease or inhibit for the proteins, that's mean we decrease the gene also as well.
Yes, but in this kind of studies, usually there is expression for both, for the gene expression and for the protein, because maybe the effect in the decreasing in the protein expression maybe is related for the vector, maybe is related for the virus. Yeah.
Yeah, but in our gene, the IBCAM CERNA was targeted to the IBCAM genes, the mRNA, to degrade it. But we need further studies for doing like Western blot to see the quantifications of the proteins. Didn't perform yet, but we need the further studies for it for quantify the proteins.
Thank you, Dr. Ibrahim.
Thank you.
Thank you, Dr. Muhammad Taha and Dr. Ibrahim. Dr. Rana, we can transfer to the third speaker.
Okay, so our next speaker is Dr. Muhsina Padvekar from Lokman College of Pharmacy in India. Her presentation is titled Functionalized Silver Nanoparticles with Vancomycin, a Novel Strategy to Improve Antibacterial Activity. Dr. Muhsina, the floor is yours.
Assalamu alaikum. Hello, everyone. Good afternoon.
Good afternoon.
Okay, my slides visible, ma'am?
Yes.
Yes.
Yeah, okay. So good afternoon, everyone. Thank you, first of all, to Al-Azhar University for providing me an opportunity to speak something on my topic. So let me begin with my today's topic. That's nothing but functionalized silver nanoparticle with vancomycin, like a novel strategy to improve antibacterial activity. So the background and rationale, what I'm going to say is the antibiotics resistant, it has become a global challenge in the entire world. Initially, we were striving to get rid of the organisms. And then once we found out good antibiotics, then we started to develop a resistance onto these antibiotics. So now the antibiotics have become a major problem where we are becoming so resistant to these antibiotics that we have to look on to different options available. So Staphylococcus aureus, especially the MRSA, that is a methicillin resistant Staphylococcus aureus, shows the resistance to several categories of antibiotics. Vancomycin, which basically still remains an important treatment for the gram-positive infection, but resistance is greatly increasing increasing in the entire world. So we can have different categories of infections related to the hospitals, related to the clinics, related to different categories of infections, where we are trying to treat these infections with vancomycin, and the treatment options are, available for a limited period of time, because the person or the patient usually develops the resistance to these antibiotics. So we have been trying to explore different options where we are trying to convert it into few of the products like silver nanoparticles, which are actually having a very broad spectrum antibiotic activity. So what we have done is we have combined the vancomycin and the silver nanoparticles together, so that we can produce an enhanced antibacterial efficacy with a very good synergistic effect. So we are just, if you look at the structure, this is the surface silver antibodies of silver nanoparticles. This is partially oxidized, and if it is being oxidized, it reveals the core of the silver nanoparticles. So what is the aim of the study? We are basically primarily aiming to enhance the antibacterial potential of the vancomycin by coating it onto the silver nanoparticles. So we have targeted two different types of organisms. One is Staphylococcus aureus, that's a gram-positive organism, and E. coli, that's nothing but a gram-negative organism. So basically, we had a hypothesis of vancomycin capped silver nanoparticles. That is, when silver nanoparticles would exhibit the stronger antibiotic activity than the single alone if we are using the vancomycins, or separately if we are using only the silver nanoparticles. So we just have made the precursor solution in which the silver nitrate, then it was reduced with the help of the trisodium citrate, and then we have capped the silver capped the silver nanoparticles with the vancomycin, and we then got the stabilized product after incubation. We got a very good product, that's an capped product of silver nanoparticles of vancomycin. So what is the mechanism of action? The vancomycin binds basically to the citrate capped silver nanoparticles through the hydrogen bonding, which is merely, you can say, a very strong bond compared to that of weak van der Waals forces. So silver nanoparticles releases the silver ions, generating the reactive oxygen species and disrupting the bacterial cell wall. So the major aim is nothing but destruction of the bacterial cell wall, so that our vancomycin can enter inside and they can destroy the bacteria. The conjugate may enhance the cellular penetration of these silver capped nanoparticles and interaction with the bacterial cell membrane. So we basically have the synergistic effect here. That's the combination produces a greater antibiotic activity than the individual component. Like individually, if we try to put the vancomycin into the bacteria, they enter, but a very few concentration of vancomycin were entering inside, or they were easily developing the resistance to that particular antibiotic. So what we try to do is we have capped the drug with the silver particles, and these silver particles are helpful in penetrating into the cell and disruption of the bacterial cell membrane, as well as they are inhibiting the cell wall synthesis. They are helpful in inhibition of the replication of the genetic material. They are helpful in degrading or down regulation of the protein synthesis, which indirectly is helpful in synergistic effect of our antibiotic activity. So synthesis of the vancomycin silver nanoparticles is nothing but we have taken the precursor in the form of silver nitrate. We have taken a reducing or capping agent in the form of trisodium citrate, and it was employed, as I said, as a reducing as well as a capping agent. And we basically used a chemical reduction form to cap the silver nitrate nanoparticle. Functionalization, we have taken the vancomycin into different concentrations like 0.05, 1, and 0.3 millimolar, and the medication is utilized for storing on the surface area for the formulation of the silver nanoparticles. So we have characterized these nanoparticles with the help of the UV visible spectroscopy, confirming the nanoparticle formation as well as the drug conjugation. So we have used the double desalination water for each of the solution, which was prepared using the water that has been twice deionized. So experimental methodology, we have, as already discussed, we have taken two organisms like Staphylococcus aureus as well as E. coli. We have made an Acar well diffusion method. Sample tested, we have tested simple alone vancomycin. We have tested a citrate capped silver nanoparticle alone, and we have taken a combination of vancomycin nanoparticle, and we have used saline as a control group. So outcome measures, we have checked by a zone of emission into the millimeters, and for the control silver nanoparticles without the drug and saline was use. So you can see here onto the E. coli, that's a gram-negative organism. The silver nanoparticles, they did not show any effect. While the combination of the, you can say the nanoparticle as well as the vancomycin, they are showing a pretty much good inhibition. While in case of silver nanoparticles, you do have the entire, like silver nanoparticles alone, even also if you use, you do have the activity of inhibition of the bacteria, but the combination effect was far more greater than that compared to the vancomycin alone. So antibacterial activity results same, like vancomycin alone was active against Staphylococcus aureus, but as discussed already, it's ineffective against the E. coli. But the vancomycin silver capped showed the enhanced activity against both the organisms, that's Staphylococcus aureus as well as the E. coli. So the highest activity was observed at 0.3 mm, which was around 27.7 into the gram-positive Staphylococcus aureus, as well as into the gram-negative E. coli, it was showing around 7.8 mm zone of inhibition. So if you look here, these are the vancomycin silver-coated nanoparticle. This is only silver-coated nanoparticle. This is vancomycin. And here you can see this is control. This is the combination of the vancomycin and silver-coated nanoparticle. This is into number one, this is into the Staphylococcus, and this is under the E. coli organisms. Into E. coli, you don't find any category of zone of inhibition, while along with the combination, you do find a little bit of zone of inhibition when you are trying to use it along with the vancomycin and the silver-coated nanoparticles. So comparative effect of the vancomycin silver-coated nanoparticle into the treatment, the silver, if we are trying to only go with silver nanoparticles, it is not showing any zone of inhibition, while if we try to go with vancomycin alone, it showed the effect into the Staphylococcus aureus, but there were no effect onto E. coli. That's a gram-negative organisms. While coming on to the combination, where we have taken the vancomycin cap silver nanoparticle, where the zone of inhibition of Staphylococcus aureus was around 26 to 27 in an average, while E. coli, it was ranging from 7.5 to 7.8. So vancomycin cap silver nanoparticles produced the largest zone of inhibition against both the bacterial stains, and the conjugate converted the vancomycin from the inactive against E. coli to the measurable antibacterial activity. So compared with the vancomycin alone, the conjugate basically increased the activities of Staphylococcus aureus by approximately 45%, which was a huge gain. So advantages of these nanodrug conjugates, rather than using the single drug, we tried to make it into a combination format, and few of the advantages what we found is like it had an enhanced antibacterial potential due to the combined action of the vancomycin with the silver nanoparticle. They did had a broad spectrum antibiotic activity as well as antibacterial activity, extended its effectiveness from both the gram-positive as well as the gram-negative bacteria. So targeted delivery potential against the silver nanoparticles can be transport the vancomycin closer to the bacterial cell wall. As we have already seen into the mechanism, it can disrupt the bacterial cell wall, it can disrupt even the DNA, it can even denature the proteins, and that is helpful in killing of the bacteria. So reduced resistance pressure by attacking the bacteria through the multiple mechanisms, and we do have different options available for trial and error with these, and because of which these combinations, they do have a reduced resistance compared to that of single antibiotics if we are trying to use it. So stability improvement, obviously, when you are trying to cap it with silver nitrate, so obviously it is having a very good stability ratio, which can be stable for a longer duration of time, and potential for the controlled release also. We do have like at what time we have to release the organisms, like timely manner, maybe in case of personalized medication also we can utilize these things, improving the therapeutic efficacy of these medications. So translational relevance as well as the future application. So vancomycin silver nanoparticles may offer a compromising strategy for treating the multi-drug resistant bacterial infection. It has enhanced antimicrobial activity. It improves the outcome against the gram-positive pathogens. Potential applications, it's basically been used into number of the wound dressings, catheter coatings, implant coating, topical formulations, targeted antibacterial therapies. So future investigation, in vivo efficacy study, toxicity profiling, pharmacokinetics, long-term stability, as well as dose optimization. So research impact demonstrated that the potential of the nanotechnology has revitalized the existing antibiotics which we are using, and these antibiotics which are trying to prove lot many resistance into different disorders. So treatment options have lowered down because of the resistance, but we do have an option where we can try to combine it with the nanoparticles, and we do have lot many options of stability improvisation, therapeutic improvisation, and better availability of these medications. So coming on to the conclusion, the vancomycin coated silver nanoparticles were successfully synthesized using the citrate cap silver nanoparticle. Vancomycin capped silver nanoparticle demonstrated significant improved antibacterial activity against both the S. aureus and E. coli. It has enhanced synergistic rather than additive, and vancomycin may serve as an effective drug delivery and antibacterial system. So here I conclude my part. I'll be glad to answer the questions.
Thank you for the interesting presentation. I think we have two questions, one from Professor Ihab Al-Masri and from Dr. Muhammad Abu Talib. So, Professor Ihab.
Thank you, Doctor, for your nice talk and very useful information about nanoparticles or conjugate of silver and vancomycin as antibacterial agent. You said in your last slide that recommended further toxicity studies and pharmacokinetic studies for this conjugate. I wonder, do you expect that there is a potential toxicity for such conjugate and difficulties in their delivery to inside the cells, or difficulty in their bioavailability?
Yes, sir, that's an important question. Our study actually primarily focused on evaluating the in vitro antibacterial efficacy of vancomycin coated particle. But there can be, toxicity study depending upon different factors, like you can consider particle size, the concentration, maybe what surface coating you are using, what are the route of administration, and maybe at the higher concentrations also, it may induce a high amount of oxidative stress. Maybe it can lead to inflammation also, DNA damage. So we do have to go with a comprehensive toxicity studies, which would be basically focusing on cytotoxicity study, maybe onto a genetotoxicity study also. We have to look onto it, sir.
Thank you, Doctor. Thank you.
Yes, Doctor. Thankful for your interested lectures, Dr. Mohsina. And
Thank you very much, sir.
Yeah, this is affordable work because I'm doing that before. But I have a question that why use vancomycin silver nitrate for S. aureus and Escherichia coli, which is usual. I think that's treated by vancomycin. Yani why cannot use in your in vitro that's Pseudomonas aeruginosa, Klebsiella, because this is the challenges for these techniques. S. aureus and yeah.
Yes, sir. Can you just please repeat, because it's little bit not audible?
Yes, I ask about why use and your in your experiments and research that Staphylococcus aureus and Escherichia coli, which
Yes, sir.
is usually treated by vancomycin. We not
Correct, sir.
yeah. Yes. We're not obligatory to use silver nitrate in many events. So why not use Pseudomonas and Klebsiella for these resistant bacteria, and it's considered as challenges?
Sir, actually, we selected vancomycin. We included both, selection of vancomycin was basically done because it was an important glycopeptide antibiotics. And it was, the last line treatment for gram-positive organisms, what you can say. And coming on to your second part, that Staphylococcus aureus, you can say like silver-coated conjugate could exhibit activity on both the gram-positive and gram-negative bacteria. But if you go only on vancomycin, that was having an effect on only gram-positive organism. While coming on to Pseudomonas aeruginosa, it's a clinically important, multi-drug resistant gram-negative pathogen, So as the vancomycin, it's active onto gram-positive. So we have focused basically onto gram-positive only, but it had found a great effect onto gram-negative as well. That was on E. coli.
Yes, and in the silver nitrate dose, we know that it has a toxicity and has electrical, it's considered in your research for silver nitrate citrate cap.
No, sir, it was just an in vitro activity. So for the toxicity study, as I said we still have to proceed with the toxicity study for the same.
Yes. Thanks, Dr. Mohsina, and we are appreciated for your sharing us in this conference.
Thank you very much, sir.
Yes, inshallah, in other conference, most welcome. We will
Thank you very much.
Yes, thanks. We will now transfer to the fourth participants, Mais Saeed. Mais Saeed is graduated from University of Petra, Jordan, and the title is The Impact of Hydroxyapatites Nanoparticles on the Cellular Processes of Stem Cells Derived from Dental Tissue Sources. Most welcome, Mais, and introduce microphone.
Thank you, Dr. Muhammad, for the introduction. Thank you for accepting my study in this conference. And I'll start by defining my title. The title of my study is The Impact of Hydroxyapatite Nanoparticles on the Cellular Processes of Stem Cells Derived from Dental Tissue Sources. This is the outline of my presentation. And I'll start by talking about the significance of my work. My work talks about the synergistic effect of hydroxyapatite nanoparticles and stem cells yields a highly effective therapeutic strategy by augmenting dental pulp stem cells differentiation. This approach holds great promise for the field of regenerative medicine. Our project aimed to prepare the hydroxyapatite nanoparticles. Then we aimed to characterize these hydroxyapatite nanoparticles by measuring their size and shape. Later, we evaluated the biocompatibility of these HANPs or hydroxyapatite nanoparticles on the biological processes of DPSCs, dental pulp stem cells. We evaluated their proliferation, stemness, and toxicity. And finally, we aim to explore the impact of these HNAPs on the osteogenic differentiation of DPSCs. The methodology that was applied or employed in this study was, first of all, the preparation of hydroxyapatite nanoparticles, then, of course, the characterization. Later, we started by cell culture maintenance and expansion, the MTT assay, colony forming unit, cell cycle assay, cytotoxicity assay, migration assay, and finally, the osteogenic differentiation assay. In our results, of course, the first result was characterization of hydroxyapatite nanoparticles. We used freeze drying to characterize the concentration of hydroxyapatite nanoparticles, and the known concentrations were ranging from 1.633 milligram per ml and going down until 0.102 milligram per ml. Later, we imaged those nanoparticles. As we know, the hydroxyapatite nanoparticles are spherical shaped nanoparticles. So we use transmission electron microscopy to determine that they are spherically shaped. And also, it was seen that their average size was 42.21 nanometer. We confirmed their size by doing dynamic light scattering, and the average size was seen to be 60 nanomolar, which is also acceptable in characterizing hydroxyapatite nanoparticles, and the average PDI or polydispersity index was 0.611. We started by evaluating the effect of hydroxyapatite nanoparticles on the morphological appearance of the dental pulp stem cells. And our results have demonstrated that HANP's concentrations of less than or equal to 0.81 milligram per ml maintained the same morphological appearance of DPSCs compared to the untreated control. And this was interesting for the beginning of the experiments or the study. Later on, we did the, of course, and most importantly, before proceeding, we did the MTT assay. And the results showed that DPSCs did not show any changes in cell viability when treated with HNAPs at less than or equal to 0.81 milligram per ml in comparison to the control group. Actually indicating that lower concentrations showed no effect on viability. Then another important assay for assessing the stemness of stem cells, we wanted to make sure that the stemness or the important properties of stem cells were not affected. So we used the colony forming unit. Stem cells usually are known for their colony forming. So in this assay, we found that DPSCs with less than or equal to 0.2 milligram per ml HNAPs showed no significant change in CFU in comparison to the control untreated groups. Also, we assessed the cell cycle of DPSCs after being treated with different concentrations of HNAPs, and the results have shown that DPSCs treated with 0.81 milligram per ml HNAPs showed a significant change in the number of cells in all phases, G0, G1, G2M, and the S phase. Also, DPSCs treated with 0.2 milligram per ml and 0.1 milligram per ml concentrations of HNAPs showed a cell cycle arrest in the G0, G1 and a significant decrease in the number of cells in the S phase compared to the control untreated cells. Also, and one of the most important assays was the apoptosis necrosis assay, cytotoxicity assay. We used flow cytometry to assess this, the apoptosis, and that HNAPs did not shift the DPSCs to the apoptotic or to the apoptosis. And the results showed that DPSCs treated with 0.1 milligram per ml HNAPs exhibited a slightly significant change in the number of healthy cells compared to the control untreated cells. So it was a very slight change in the apoptosis of these cells. Also, one of the most important stem cell properties is their ability to migrate, as it's important for healing or tissue repair. So to assess the migratory effect of the DPSCs after being treated with HNAPs, we performed a transwell migration assay, and our results showed that HNAPs less than or equal to 0.4 milligram per ml showed no significant change in the number of migratory DPSCs compared to the untreated controlled groups. Finally, and most interestingly, HNAPs were assessed with the DPSCs on their effect of osteogenic differentiation. And very interestingly, DPSCs cultured with growth media supplemented with HNAPs less than or equal to 0.4 milligram per ml showed the same differentiation pattern as well as the cells treated with osteogenic media supplemented with HNAPs less than or equal to 0.4 milligram per ml, which means it induced osteogenic differentiation, not only showed no effect. And finally, in conclusion, the conclusion of my study, we concluded that HNAPs did not disrupt the normal biological functions of DPSCs, as HNAPs were biocompatible with the DPSCs. Also, HNAPs exhibited no changes in the main characteristics of stem cells or their morphological appearance and stemness and self-renewal potential of the treated cells. HNAPs induce the osteogenic induction of DPSCs without the addition of osteogenic supplements. The combination of HNAPs and DPSCs can be considered a very promising approach that can be clinically applied in tissue engineering and regenerative medicine applications of heart tissue such as bone and teeth. I would like to acknowledge those who worked on this research. And those are some of our pictures in the laboratories and our research team. And thank you, everyone. And I'm ready for any questions.
Fully thankful, Mace.
Welcome.
Yes, yes. Nice lectures. And if we have any question, Dr. Muhammad. Dr. Muhammad Taha.
Dr. Ihab, yeah, Dr. Muhammad, Dr. Ihab.
Dr. Ihab al-Masri.
Thank you, Dr. for your nice talk. I would suggest future work for you for such a good approach to improve the osteogenic behavior of for dental care. If you can add more one than additive in your nanoparticles, such as vitamin D or hyaluronic acid, they have strong osteogenic activator. Do you think that will be a nice work for extending your research?
Thank you for the suggestion, and of course it would be taken into consideration. Of course, nanoparticles are good delivery systems, and such a promising nanoparticle should be developed for future work. So thank you very much for your suggestion.
Thank you very much.
Mace, you are a dentist or pharmacist?
No, I'm a pharmacist.
Yes.
So I'm mixing pharmacy and biomedical sciences. So that's close to pharmaceutical biotechnology.
Yes, I know that. Yeah. Okay, thankful, Mace. And we have a question from Dr. Khalid Masri. Dr. Khalid? Dr. Khalid?
Hello, everyone.
Yes.
Thank you, Dr. Muhammad Talib. Thanks for Dr. Rana, Dr. Muhammad Taha, Dr. Ihab. Thanks for all. Thank you for your excellent presentation. My question is about you think hydroxyapatite nanoparticles could have similar effect on other types of stem cell, such as mesenchymal stem cell from bone marrow or adipose tissue?
Yeah, that's a great question. Yeah, of course. Actually, stem cells are very similar in markers, especially mesenchymal. When you talk about mesenchymal stem cells, most types of dental stem cells are mesenchymal. So it might be reliable, and it might give us a scope that next research would be successful. Of course, I can't give 100% because I didn't try, But this could be also one of the excellent future works to try other types of stem cells, especially if they are mesenchymal, even if they are not dental source. So yeah, I think it would be promising if they were synergistic with other types of mesenchymal stem cells from other sources, such as bone marrow, as you mentioned previously.
Yes, Dr. Khaled, I know that I want to mention to Mais that Dr. Khaled is specialized in stem cells and has more project. Yes, Dr. Khaled? No more question for our guest, Dr. Khaled. Yes?
No, it's okay.
Yes,
It's okay.
yes.
No, feel free to do so. Of course.
Fully thankful. We have no more time. Thankful, Mais.
Thank you very
Jordanian, yes, for all Jordanian participants, most
Thank
welcome.
you.
Yes,
Thank
yes.
Thank you. It was a pleasure joining a big university in Gaza. يعني انتوا فخرنا.
Yes. We appreciate it by you. Okay. We now transfer to Ahmed Asiri and Dr. Rana.
Let's welcome our next speaker, Mr. Ahmed Asiri from University of Exeter in the UK. His presentation is titled Impact of GLP-1 receptor agonists on the pharmacokinetics and safety of combination oral contraceptives in women in reproductive age, a systematic review and meta-analysis. So welcome, Mr. Ahmed.
Hello, good afternoon.
Good afternoon.
Can you hear me?
Yes.
Sorry, I couldn't open the camera because my PC hasn't have camera, so
that's fine. Okay. So could you please share the slides with us?
Sure. Can you see my slides?
Yes.
So good afternoon, everyone. Today I will present my systematic review and meta-analysis impact of GLP-1 agonist on the pharmacokinetics and safety of combination oral contraceptive in women of reproductive age. As you know, GLP-1 are widely used for treatment type 2 diabetes and obesity. Because these medications can slow gastric emptying, there is concern that may affect the absorption of oral contraceptive. Okay, in this presentation, I will explain the background of study, the method, and the main finding, and the clinical implications. So, combination oral contraceptives are one of the most common used methods of contraception worldwide. They contain both estrogen and progesterone, which work together to prevent pregnancy. Recently, the use of GLP-1 agonists has increased rapidly because there are effects in both. Can you hear me? Hello?
Yes, but if you could raise your voice, please. Yes.
Okay. Recently, the use of GLP-1 agonists has increased rapidly because they are effective for both diabetes and weight management. However, this medication delayed gastric emptying, which may affect the absorption of oral medication, include oral contraceptive. The aim of study was to evaluate the effect of GLP-1 agonist on the pharmacokinetics and safety of combination oral contraceptive. The study also aimed to determine whether the study indicate the interaction could have improved the clinical implications. The method. A systematic review and meta-analysis was conducted follow the PRISMA guideline. A comprehensive search was performed in 4 electronic databases: PubMed, Scopus, Web of Science, and Coherent Library. randomized control trial, and cohort study were included if they evaluated the GLP-1 agonist together with oral contraceptive. The main pharmacokinetic outcome include area under the curve, maximum concentration, half-life, clearance, volume of distribution. Also, safety outcome. Also, the inclusion and exclusion criteria study will include if the woman using a combination of oral contraceptive and evaluated the effect of GLP-1 result. The literature's research identified 132 records. After removing duplicate and screening titles and abstract, 35 articles were assessed for eligibility. 6 studies met inclusion criteria and were included in the meta-analysis. This study included a total of 589 participants. Characteristics of included studies. The 6 studies were conducted in Denmark, United States, and the United Kingdom. The several GLP agonists were investigated, including semaglutide, liraglutide, dulaglutide, and exenatide. This is the Prisma flowchart. Quality assessment. The randomized control trial show a low risk of bias using the ROP2 assessment tools, and the cohort study were assessed by using the Newcastle-Ottawa scale, were related a moderate to high quality. Overall, the quality of this study was considered good. So the analysis. This slide presents the result of area under the curve, and analysis show a statistically significant increase in the area under the curve following treatment GLP-1 agonist. Maximum concentration. The difference wasn't statistically significant. So they suggest that GLP-1 agonists do not statistically affect of peak. Time of concentrations. This finding is expected because these medicines slow gastric emptying. As a result, oral contraceptive require more time to reach their maximum concentration. Half-life, no statistically significant difference was observed between the drugs. This indicates that GLP-1 agonists affect absorption rather than eliminations. Clearance, no statistically significant was found. Also, this suggests that GLP-1 agonists do not affect the body ability of eliminate. Volume of distribution, no significant difference was observed. This indicates that the absorption of contraceptive hormone with the body remain unchanged. Total adverse effect. The analysis showed that adverse effects were common among participants received. Most adverse effects were mild to moderate in severity. Nausea. Nausea, one of the most common reported adverse effects. Approximately 1 out of every 6 participants experience nausea. Gastrointestinal disorder. also frequently reported included symptoms like diarrhea, constipation, and abdominal discomfort. Publication bias. The publication bias was assessed using funnel plots. Strength of studies. This study follow the PRISMA guideline and include comprehensive literature search, multiple pharmacokinetics and outcome were evaluated. The limitation the only one or two limitations, only 6 studies were available for inclusion, so we need additional high quality studies in future. Conclusion. In conclusion, the systematic review and meta-analysis show that GLP-1 agonists significantly increase the area under the curve and delay the maximum of concentrations. No significant change in half-life, clearance, and volume of distribution. So we need large clinical study in the future to determine the pharmacokinetics and also to determine the interactions between GLP-1 agonist and combination oral contraceptive. Thank you very much for your attention. Any question?
Thank you, Dr. Ahmed, for the interesting presentation. Any questions? Okay, I have a question. And basically, it's all about pharmacokinetics. So I just need to ask about the hypothesis when you built it that there might be an interaction between the contraceptives and the GLP-1 agonists.
Yes,
but
yes.
The GLP-1 agonists, they are biologics, and for the contraceptives, they are small molecules. So if we are looking at the ADME process, they are completely different. So the incidence or the potential for a pharmacokinetic interaction is quite rare. And looking at the delivery system or the route of administration, we can tell that it's the pharmacokinetic interaction is unexpected. So my question is, how did you formulate the hypothesis for this research? On what basis, I mean?
I think because nowadays they increase use the GLP-1 and most of pregnant women use contraceptives. So I want to check if there is any interactions between these groups.
Yes. So apologies, but I'm just asking here as a reviewer because this is the first thing that you will be asked about. So it's good that now you need to defend it when you submit it, but it's an interesting research, knowing that there might be a certain kind of interaction. It might not be a pharmacokinetic one, but it could be on the pharmacodynamic level, whether on the contraceptives themselves or the GLP-1 agonists. So I think we have another question from Dr. Muhammad Taha.
Yes, many thanks, Dr. Ahmed, for the nice talk. So my question, because did any of these studies reported, for example, for some cases of contraceptive failure or unintended pregnancy associated with the using of GLP-1 receptor agonists?
Sorry, I didn't catch your question.
So because you evaluated the effect of the GLP-1 receptor agonist on the pharmacokinetics of the contraceptive, yes?
Yes.
So did any of these studies that you analyzed reported some cases of contraceptive failure? I mean that if there is unintended pregnancy occur or associated with the using of GLP-1 receptor antagonist?
No. There's no any.
Yeah. So, okay. And the second question, because as you know that I'm speaking as a pharmacist, so there are different drug related to GLP-1 receptor agonists. So, and I think that these studies just evaluate different type of these drugs. So all of the GLP-1 receptor agonists have the same effect, or there are differences?
No, it's totally different. No.
So they mentioned which.
You mean in interactions?
Yes.
Yeah, it's totally different. Some of the GLP-1, for example, has side effect like bleeding, and another has, for example, more in pancreatitis, and another more in retinopathy. So it is totally different in adverse effect. It's not the same.
Okay. Many thanks, Dr. Ahmed.
Thank you for your questions.
Take a talk here. He talked about the biological effect, non-biological effect of this GLP-1 agonist, which is delayed gastric emptying. Delayed gastric emptying causes the nausea, a major side effect of these compounds, and also can delay the absorption or delay the effect of certain drugs. This is the hypothesis he built on his research. The pharmacological effect on delaying gastric emptying of GLP-1. So he expected that drugs could be affected by delaying their absorption or bioavailability.
Yes, thank you, Dr. Professor Ihab, for explaining this. It makes sense. So, any other questions?
Thank you, Dr. Ihab, for your clarification.
You are welcome.
Okay, so Dr. Muhammad Talib, now you can introduce our next speaker, please.
Yes, thanks, Dr. Rana. Now with the next speaker, Nusayba Ali, Applied Sciences Private University, Jordan, as the titles of research, development, and in vitro evaluations of mucoadhesive buccal films loaded with sumatriptan nanoparticles for enhanced mucosal retention. Most welcome, Nusayba.
يعطيكم العافية. السلام عليكم ورحمة الله وبركاته. Good afternoon, everyone. My name is Nusayba Ali, and today I am pleased to present my research entitled Development and In Vitro Evaluation of Mucoadhesive Buccal Film Loaded with Sumatriptan Nanoparticles for Enhanced Mucosal Retention. Okay. In this study, we focus on sumatriptan, one of the most commonly prescribed medication for treatment of migraine. Sumatriptan is classified as selective serotonin receptor agonist. Okay, now before discussing our formulation, it's important to understand the physical chemical properties of sumatriptan, as this properties strongly influence its behavior and therapeutic performance. As shown in the chemical structure, sumatriptan has molecular weight of approximately 295 grams per mole, which is considered relatively low and variable from drug absorption and formulation. In addition,
We have tertiary amine group, which become positive charge under physiological condition. That's mean inosomethyltryptan is classified as basic drug, although somethyltryptan is high water soluble, low lipophilicity due to poor membrane permeability and high ionization. Consequent that drug absorption is limited. They highlight the need for novel drug delivery system to improve bioavailability. Okay, despite its clinical effectiveness of treatment migraine attacks, somatostatin has several pharmacokinetic limitations. As we know, absorption, distribution, metabolism, elimination. First, absorption. Somatostatin has a low oral bioavailability, approximately 14 to 15%, maintaining what? That maintain only small fraction of the administered dose reach the system circulation. This poor bioavailability maintain that attributed low its limited membrane permeability and extends the first pass metabolism. Second, distribution. Because somatostatin is relatively hydrophilic, its distribution into tissue is limited. That means that constant with the low lipophilicity and high degree of ionization. Another limitation, metabolism. The metabolism somatostatin undergoes rapid hepatic metabolism by monoamine oxide, making inactive metabolism. Due to its rapid metabolism, elimination half-life have short elimination half-life, approximately 2 hours. Overall, this pharmacokinetic limitation emphasized need to advance drug delivery system that improve drug absorption, bioavailability, and therapeutic effect. To overcome this challenge, several alternative drug delivery approaches have been investigated. The first, we have the buccal drug delivery, where the drug administered through the buccal mucosa inside cheek, allowing direct absorption into system circulation, will bypass the first pass metabolism. Then the second, mucoadhesive delivery system, which adherent that mucosal surface prolong the formulation residence time and enhance drug absorption. Another promising strategy is develop a polymer past sustained release, which maintain the therapeutic drug concentration for extended period and prolong drug action. Okay, advantage of this alternative to bypass first pass metabolism, rapid absorption, and good bioavailability will provide a prolonged therapeutic effect. Okay, consider this advantage. My study designed with the following objectives: to evaluate the linearity and accuracy of somatostatin UV analysis method, to determine the maximum complexation capacity of somatostatin acacia, to obtain the optimized formulation with enhanced mucoadhesive and sustained drug release. Okay, to begin with, I will start by discussing my method used in my study. The first step was the UV spectrophotometric method for the analysis of somatostatin. Somatostatin was dissolved either distant water or phosphate buffer, and the maximum absorption wavelength lambda max was determined to 282 nanometers. Then the stock solution with concentration 1 milligram per milliliter was prepared, following by standard dilution ring as shown in this table. Okay. Each experimental was prepared triplicate to evaluate the method linearity. Finally, we have a calibration curve where then considered to determine the relationship between concentration and absorbance. Okay, so results were the calibration curve obtained in both distant water and phosphate buffer demonstrate excellent linearity. In both calibration curve were excellent as coefficient of determination R square higher than 0.99, and both were considered acceptable. Okay, now next we investigated the absorption behavior of somatostatin onto acacia. Somatostatin complex was prepared using a dialysis bag method Combining with the shaker system, okay, the formulation containing 1 milligram per milliliter somatriptan and 2% acacia with phosphate buffer used as the solution media as a solvent. Then we have 2 milliliter of the formulation were placed inside the dialysis bag, while the external media combining that 50 milliliter of phosphate buffer. The system was maintaining at temperature 37 degrees, shaken at 50 RPM for 24 hours. All experimental were contained interplicate. Following that experiment, we have both the total amount of somatriptan and adsorbed onto acacia were determined. Okay, the results show. Okay, now the results show as the total somatriptan will the concentration increase. The amount of adsorbed onto acacia also increased. The result, together with obtaining curve and regression equation, confirmed that absorption following is zero order model, while R square is the value of 0.99. The slope of the regression line 49.42 milliliter per gram represent absorption or bending capacity of acacia, while the intercept 15.089 milligram per gram reflect that absorption capacity. Overall, this finding confirm the acacia show high absorption capacity for somatriptan and exhibit strong linearship between somatriptan concentration and the amount was absorbed. Okay, now we have another method used in this study was evaluate of mucoadhesive properties of the formulation, a strong adhesion maintaining prolonged containing with the oral mucosa. We use the texture analyzer. Use texture analyzer to first to measurement strength of adhesion between dry film which was prepared and mucosal tissue. Second, to evaluate how long the dry film remaining attached to the mucosa. Okay, then the slide show the device. This is a texture analyzer, as I said, to use to measure the mucoadhesive strength of the film. The film was attached here in a probe. The film was attached while the mucosal tissue was placed on the stage at temperature 37 degrees, stimulated oral cavity. The probe touch the tissue, then move up. The load cell measurement that force need to detach the film. The highest force recording is mucoadhesive strength. In my study, the optimized formulation show the values of 33.8 Newton, indicate that good adhesion to the mucosal tissue. After measurement, the mucoadhesive strength, the result will include in Box-Penken design. This figure is a Box-Penken design, represent that surface methodological that evaluate the effect of formulation variable and to obtain the formulation. Now we have a 3D response plot. This demonstrated that the film forming polymer concentration and the plasticizer concentration on mucoadhesive strength. The formulation was successfully optimized in this point. The high mucoadhesive strength of 33.8 Newton was attached, induct strong adhesion, and suggest prolonged retention of the buccal film on the mucosal surface. Okay, in addition to the method, I have already discussed several other methods were also occur on my study. However, due to limitation presentation time, I will only maintaining them briefly without going into further details. This include somatriptan acacia nanofilm preparation and in vitro drug release and stability study using a dialysis bag method in phosphate buffer. والphysiological and morphological characterization do الDSC, الX-ray diffraction, FTIR, scan electron microscopy, transmission electron microscopy, and the zeta size, following by pharmacokinetic and absorption enhancement rationale in rats. Okay, thank you for your attention. I would be happy to answer any question. Please feel free to contact us email, and please scan the QR code to access our research group profile and publish article. Thank you all.
Thanks, Nusaiba. We are very proud by Jordanian professors and researchers. This is really Jordanian Day, Dr. Mu'tasim, Prof, and Prof. Dr. Rana, and Nusaiba, and Mais. I have one question, if anyone, yeah. Omar Ali. Yes. Yes, Omar. Yes,
Unlock,
Omar.
unmute.
Yes, Omar.
Unmute, Omar. Yeah.
If we can
Yes, I hear you.
Yes,
I
yes.
hear you. Are you listening to me? Okay. Thank you very much. I appreciate your effort and the clear explanation. I ask you, why use it absorption isotherm? Are you hearing me?
Yes, okay,
Yes,
Sam,
yes,
thank you.
yes.
Was prepared that absorption isotherm to determine the maximum bending of capacity of acacia. It's optimize drug to the polymer ratio before film preparation.
Yes. Marwa.
Okay, thank you.
Okay. Marwa.
Okay,
Yes.
can you hear me?
Yes.
Yes.
Thank you, doctor, and thank you, Nusaiba, for this amazing presentation and work. I just have one question. In your experiment, why did you use the 282 nanometer wavelength, and also why the prolonged resistant time was important in your procedure?
Okay, thank you. Good question. Let me go back to this point. We use the 282 nanometers lambda max UV method because it's maximum absorption of somatryptan. That's mean high sensitively and accurate. About question number 2 is please repeat. بس إيش كان ثاني سؤال؟
Yes. Again, Marwa. You can repeat your question, your second question, Marwa.
Please repeat your second question.
Marwa, I think it's not on.
About
yeah.
the time, it's resistant time. Why we have a long time? Okay, this we will have longer resistant time by to use the sustained release, drug release, and increase the operate of the drug absorption through the buccal mucosa. We need for a long time.
Oh, yes. Thank you. Yeah. I was just asking why the residence time was important in your
I hope that
experiment.
answered your question.
Yeah, you did. Thank you.
Okay, thanks, Marwa. Nida Hussein.
Hi, everybody.
Yes.
I questions about the buccal is as how use this determine the ممكن أحكي عربي لأني مش كتير أنا
Yes, no problem. Yes.
هلا في
We
معظم
have
المستشفيات
English
عندنا
translation.
بنابلس في مراكز في نابلس بيستعملوا زي سرنج أو بوتكس عشان يقللوا كمية المايجرين اللي في الدماغ، فهمت كيف؟ فهلا هاي المادة بتكون فيها سومتريبتان بوكال يعني عن طريق الخد ولا كيف؟ شو طريقتها عندك معرفة؟
نعم، هي عبارة عن طريقة بكل ميكوزا، يعني عبارة عن فيلم حيكون بالبكل باللثة من داخل حيتثبت، وحناخذ شو اسمه يعني حيعطي الفعالية، هو كنوبل دوج فورم.
Yes, Nida. Okay. Nusaiba, I want to ask you one question. Yes, hear me?
Yes, yes, doctor.
Yes. About the theory. That's يعني I can say that how did the incorporation of nanoparticles influence the balance between mucoadhesion, drug release, and the mucosal permeation? Yeah, you know, يعني was there
No,
any was there any trade-off between prolonged retention, because you are the titles, it's retention in vocal, and rapid therapeutic onset? Was noted that rapid therapeutic onset when you are prolonged retention of vocal roots.
yes, it does. It's to prolong the time.
Yes. يعني
Sustained release.
you know that migraine from clinical. We have no time, so, because this session is more extended, Dr. Ihab. We are sorry, but I think that's it. Migraine clinically, it's not treated. All we know that. And the goal of a treatment is that the widening the gap between more than attack, widening the gap between attack. If this technique, yes.
اوكي
Yes.
تمام. على حسب فهمي السؤال، ان احنا اللي احنا عندنا مشاكل بالدوج فورم اللي موجوده حاليا، فاحنا حنحسن الدوج فورم الموجودين، ولكن احنا المايجرين مش حيتعالج، ولكن هنعمل له زي وقايه.
اوكي، هل عمل برولونكشن البوكر ريتنشن؟ هل عمل onset action is more rapid from your experiment؟
يس، بس احنا لحد الان التجارب ما خلصناهم، لانه في عندنا تجارب فارماكاينتيك، فعلى حسب نتائج الفارماكاينتيك حيبين اكثر. because this is
Okay,
my
thanks.
thesis in master student.
Thanks, Nusaiba, and we are grateful and thankful for Dr. Samar Thiab, supervisor, because it was moderator.
يعطيك
Yes.
العافية دكتور. يعطيك
Yes.
العافية. I'm part of her supervision, Dr. Sama Abdullah, so I need to answer you about your question about the rationale of the mucoadhesive nanogel. It's nice to meet you and nice to have you there. وطبعا كل الديليجيشن والريكوجنيشن شكرا للدكتورة سمر شجعتنا نشارك. بس the main idea here is to first of all to form the somatryptan nanoparticle because of the polymeric system shear strength. Afterward, we have encapsulated the somatryptan inside a film, a gelling film. This gelling film will work as a controlled release influencer for the nanoparticle. As much as the size was lowered, as we can achieve the controlled release we have achieved it up to 6 hours, the controlled release behavior. In order to see if we can have an extended effect, as well as we have confirmed that on the in vivo absorption study. We would like to hear your insight actually on this study, but we have a disclosure actually we have submitted. We can't speak more about these data.
Inshallah, Dr. Sama, and congratulations for this research.
Thank you.
And we can, thanks for your Nusaiba and your co-supervisor and co-supervisor. Now, fully thankful. And now we will transfer to Dr. Rana to introduce the last researcher, and I think it's your student. Yeah, Dr. Rana?
Yes. So now moving to our last speaker, but not least.
You hear me?
Yes. Do you hear me?
Dr.
Hello?
Rana?
Yes. Do you hear me?
Yes, we can.
Okay.
Yes.
So now moving to our last speaker, but not least, Dania Muhammad from Azarqa University in Jordan. Her research is titled Investigation of Herb Drug Interactions between Rosmarinus officinalis L extract and digoxin in animals. So welcome to this session, Dania, and the floor is yours. You can make a start. Dania? Do you hear us?
أنا muted.
Dania? Please raise your voice. I think the mic is we can't hear you well.
There is a problem in your mic, Danny.
Before I begin, Dr. Rana?
We cannot hear you very well, Dania.
Fix your mic.
presentation من اللابتوب صح؟
Yes.
اوكي اذا بتروحي على الساوند سيتنجز بليز.
مساء الخير. دكتور دانيا ممكن تعطيني الماوس بالله اتحكم بعمل لك الميكروفون.
Hello.
لسه سيء الصوت.
الصوت مش واضح دانيا. بس إذا بتعطي permission على عفوا بس الاسم.
باشمهندس أحمد.
باشمهندس أحمد البروفايل الأزهر يونيفرسيتي.
صحيح.
إذا بتعطيه permission عشان يعدل لك الساوند سيتنجز.
في إمكانية تنزل المحاضرة اللي تحت؟ معلق عندي كمان.
دكتورة رنا؟
الصوت لسه مش واضح. يعني صوت بعيد.
تنزل
المحاضرة. أنا خليها توقف الشير لو سمحتي، خليها توقف الشير عشان الباشمهندس أحمد.
لا خليه بلا دكتور الشير لأنه حتفصلني، بس تنزل المحاضرة تحت مش معلق الجهاز عندها.
شو رايك يا دكتورة رنا؟ دكتورة رنا شو رايك تبدي انت لحد ما تعمل ريستارت للجهاز بما انك السوبرفايزر حضرتك.
اوكي.
بعد اذن الدكتور ايهاب يعني والدكتور محمد.
دكتورة رنا؟
دانيا أنا سامعتك بس الصوت جدا بعيد، فهلا مع البرزنتيشن مش رح يكون واضح.
دكتورة رنا خليها تعمل ريستارت لجهازها.
اوكي.
بإذنك.
بليز دانيا بس اعملي ريستارت. البرزنتيشن to be honest هو مش عندي هلا، ف if you have it please انه and share it here we can make a start.
I will check دكتور.
Yes please.
بس احنا دكتورة رنا فعلا اليوم جوردانيان داي يعني الحمد لله.
بالضبط، الحمد لله، ومواضيع متنوعة.
ما شاء الله.
الحمد لله.
يعني سبورت في كل شيء. الله يعطيكم العافية.
الله يعافيك دكتور، ما عليكم زود، يعني إن شاء الله إنه الأمور ترجع أحسن من الأول يا رب.
يا رب يا رب إن شاء الله.
دانيا جربي الآن. دانيا.
أنا لقيت المحاضرة عندي، في إمكانية أنا أعمل شير وحضرتك
في عندي مقترح تاني، ما بعرف إذا بيزبط إنه أرن على دانيا وتحكي من السبيكر، إذا بيزبط.
إذا بيزبط
ممكن
يس، جرب.
ممكن تعملي جون من الموبايل على على الزوم، بكون الصوت واضح، وممكن نخلي الكمبيوتر كمان يعرض يعني
طيب أنا دانيا بليز، هلا عاملين شير للبرزنتيشن من عند دكتور محمد، فبليز جوين اس على الميتنج هذا بس من التليفون، فبتقدري تعملي البرزنتيشن من التليفون.
دكتورة رنا واضحة السلايد لو سمحتي؟
يس.
تمام، لما تكون جاهزة بس تبلغني.
يس. طيب هلا to save time I can make a start, then Dania can join us when she fixes her microphone. So this study was the master's project for Dania Muhammad. She has master's in pharmaceutical sciences and she just defended her thesis last January. Yes. Dania.
أهلا. Welcome دكتور دنا.
في صدى صوت.
تمام.
بتاع اللابتوب سكر المايك.
Welcome دكتور جناب. Welcome دكتور محمد طالب. Before I begin, I would like to honor the remarkable people of Gaza, a community whose resilience and spirit continue to inspire the world. It is a privilege to present at this conference, held under the theme Pharmacy from Rebel Toward Recovery, a theme that captures not only the mission of our field, but the spirit of Gaza itself. I'm honored to present my research today as a small contribution to this spirit of recovery. Good afternoon, everyone. My name is Dania Muhammad, and today I will present my research, investigation of herb-drug interaction between Rosmarinus officinalis and digoxin in animal model, conducted under the supervision of Dr. Rana Abu Taimer, Faculty of Graduate Study, Zerka University in Jordan. Prescription drug use extremely common worldwide, and so is the use of medicinal herbs, often together and often without patient realizing. This can cause herb-drug interaction. These interactions are frequently underestimated, yet they can significantly affect a drug's safety and effectiveness. Such interaction can alter drug absorption, distribution, metabolism, and excretion. It's pharmacokinetic, leading to therapeutic failure or toxicity. This is especially critical for narrow-therapeutic index drugs, where the gap between an effective and toxic dose is very small. Digoxin, a cardiac glycoside used for heart failure and atrial fibrillation, is a classic example. Its therapeutic window is only half to 2 nanogram per milliliter. It has oral bioavailability of 60 to 80%, peaks in 2 to 3 hours, and importantly, is a substrate of PGP. PGP, a transporter that pump drugs back out of cells in the intestine, liver, and kidney. Anything that inhibits PGP can increase the digoxin absorption and reduce its clearance, rising blood concentration. Rosmarinus officinalis is widely used medicinal herb in Jordan and Middle East, valued for cardioprotective and antihypertensive effects, containing bioactive compounds like carnosic acid, carnosol, and rosmarinic acid. In vitro studies suggest rosemary may inhibit PGP, but in vivo evidence is very limited. This gap motivated our research. Such digoxin is PGP substrate, and rosemary may modulate PGP activity. Co-administration could theoretically risk digoxin toxicity, yet no in vivo study has tested this. Our hypothesis, rosemary extract may inhibit PGP and increase digoxin exposure. We used verapamil and established PGP inhibitor as our positive control. Our objectives were to evaluate rosemary effect on digoxin pharmacokinetic, assess the role of PGP modulation, and provide clinical recommendation. This study received ethical approval from Zerka University Research Ethics Committee. We used an experimental in vivo design with male Western rats, randomized into 3 groups of 7. Group A, vehicle only, as inactive groups. Group B, verapamil, 25 mg per kilogram, as a positive group. And Group C, rosemary extract, 40 mg per kilogram, our group of interest. Each group was pre-treated accordingly, and 30 minutes later received a single oral dose of digoxin. Blood samples were collected at 0 to 24 hours, and digoxin concentration were measured using electrochemiluminescence immunoassay. We used non-compartmental analysis to calculate Cmax, AUC, clearance, and volume of distribution. The result, the concentration time profile showed Group B verapamil with the highest concentration throughout, especially early on. Group C rosemary tended only slightly above the vehicle group. Verapamil produced a statistical significant increase in both Cmax and AUC, confirming our model correctly detected a true PGP-mediated interaction. Rosemary extract, however, produced only a slight increase in digoxin exposure, a 1.12-fold rise in Cmax and 1.22-fold occurred around 8 hours in AUC, and neither reached statistical significance. The maximum fold change, about 1-fold, occurred around 8 hours during the elimination phase, rather than absorption, suggesting any rosemary effect related more to reduce clearance and enhance absorption. A limitation and implication, our study used a single dose, so it doesn't capture effect of chronic rosemary use. And animal model differ physiologically from humans. We also didn't analyze transporter expression at the molecular level. Overall, rosemary appear unlikely to cause major digoxin pharmacokinetic change and the dose tested. But given digoxin's narrow therapeutic index, caution, patient education, and the clinical monitoring remain important. In conclusion, rosemary co-administration produced a slight non-significant increase in digoxin serum concentration. Confirmation through a controlled clinical trial is still needed. We recommended 3 next steps. a multiple dose study to assess cumulative effect, a molecular investigation into rosemary impact on digoxin transporter, and the clinical observation of patients taken both together. Thank you all for your time and attention. Standing here today presenting this work in Gaza is not something I take lightly. This conference, Rising from Rebel Toward Recovery, is a living testament to this community strength and determination to keep building, learning, and healing. I'm deeply grateful to Al-Azhar University in Gaza for hosting this conference, to my supervisor, Dr. Rana Abu Taime, for her invaluable guidance, and all for your attention. I would now be happy to answer any question. Any question?
Thank you, Dania. Any questions for Dania? Okay, apologies for the inconvenience that occurred at the start of the presentation, but just to recap on this, our research group is interested in the drug interactions in general, and one of them is the herb-drug interactions, specifically that in the Middle East. We use these medicinal herbs in combination with drugs for chronic diseases, so it's somehow necessary to study these drug interactions at the molecular level. So the in vivo one is just a start to check if there is an interactions that is worth further investigation or not. Yep. So if there are no questions, then we finish our session now, and the next session, which is the poster session, will start at 2 p.m., which is almost 20 minutes from now. So we are on a break, and please, you can come back at 2 p.m. Thank you all for joining us in this session.
Thank you for all.
كي يزول الألم سوف نحيا هنا سوف يحلو النغم موطني 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 ذا الإباء موطني 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 يا أنا رغم كيد العدا رغم كل النقم سوف نسعى إلى أن تعم النعم سوف نرنو إلى رفع كل الهمم بالمسير للعلا ومناجاة القمم فلنقم كلنا بالدواء والقلم كلنا عفو على من يصارع السقم فلنواصل المسير نحو غاية أهم ونكون حقا خير أمة بين الأمم سوف نبقى هنا كي يزول الألم سوف نحيا هنا سوف يحلو النغم كم سهرنا من ليالي للصباح لا ننم كم عراقيل كسرنا كم حفظنا من رزم كم جسور قد عبرنا كم ذرفنا من حمم نبتغي صيد المعالي نبتغي رأس الهرم نقضي ساعات طوال نستقي علم العجم نستهين كل غالي كي نحقق الحلم إنسى إن لا نبالي فلنسير للأمل إن قمة الجبال تستحق لا جرم سوف نبقى هنا كي يزول الألم. سوف نحيا هنا، سوف يحلو النغم. فضلكم يا والدي عمني حتى اللجم، كل هم قد أصبنا زادكم بالطبع هم. إن كل ما جنينا من جهودكم نجم. والدي يا خير عون كان لي عند المحن، أنت يا من تملكين جنة تحت القدم. كل ألفاظ لساني كل شكر قد رهن، اجمعوا كل المعاني من عرب أو عجم. لا توافي شكرهن، لا تجاوز العدم. سوف نبقى هنا كي يزول الألم. سوف نحيا هنا، سوف يحلو النغم. هذه فرحة الأهالي لا يساويها رقم، حين يشهدون حالي بالسرور يبتسم. فرحتي وصرختي تكاد تسمع الأصم. يا نجوم السماء يا عبائق النسم، يا سحائب الرجا يا طيور الحرم. يا رعود الشتاء يا جميع الأنم، اشهدوا هذا المساء أنني قلت القسم. سوف نبقى هنا.
سعيد جدا اليوم تواجدي في جامعة الأزهر، جامعة الحبيبة، وبالأخص كلية الصيدلة، حيث كانت الخطوة الأولى والمرحلة الأولى في بداية مشواري الأكاديمي. من هنا بدأت ومن هنا انطلقت كلية الصيدلة، حيث كنت يوم من الأيام طالبا في هذه الكلية، وتخرجت من جامعة الأزهر كلية الصيدلة في عام 2011، ومن ثم حصلت على منحة الماجستير من مؤسسة الدولي الألمانية في دراسة درجة الماجستير في الصيدلة السريرية من الجامعة الأردنية، ومن ثم حصلت على منحة أخرى من مؤسسة الدولي لدراسة درجة الدكتوراه في ألمانيا في علوم الأحياء الجزيئية وطرائق اكتشاف الأدوية. أنا أعمل حاليا في مؤسسة أو شركة أكسم الألمانية التي تعمل في مجال التكنولوجيا الحيوية وطرائق اكتشاف الأدوية، حيث نعمل مع كبريات شركات الأدوية عالميا مثل شركة روش، بورينجر، شركة باير. وتوجد هناك معاملات بيننا وبين هذه الشركات لاكتشاف أدوية مثل أدوية السرطان وأدوية كورونا كما نعلم، الجائحة التي أثرت على مستوى العالم. فيوجد لدينا بعض من هذه المشاريع في ذلك الاتجاه. لقد شاركت في العديد من المؤتمرات العالمية والأبحاث العلمية المحكمة، وكانت هذه الأبحاث في مجال دراسة تأثير الطفرات الجينية على الإصابة بأمراض السرطانات، وتأثير هذه الطفرات الجينية على استجابة المرضى للعلاج الكيميائي. وأيضا درسنا تأثير الطفرات الجينية على الاستجابة في مرضى زراعة الكلى. ومؤخرا كنت أحد المشاركين في أبحاث علمية أسفرت عن اكتشاف مجموعة من المركبات الكيميائية التي لها طبيعة تخصصية في علاج أمراض السرطان. وطبعا هذه الفئة من الأدوية ذات أو المركبات الأولية لمشاريع أدوية. طبعا هذه النوعية من المركبات الكيميائية لها تأثير قوي على الهدف ولها أقل ما يمكن من الأعراض الجانبية، بحيث تكون ذات فاعلية وأقل سمية على المرضى. ومن هذا المقام أوجه رسالة شكر إلى أساتذتي ومعلميني في كلية الصيدلة على ما قدموه لنا خلال فترة الدراسة من تدريس وإرشاد حتى وصلنا إلى ما وصلنا به الآن. وأيضا أحث الدارسين والطلاب في كلية الصيدلة بأن يستكملوا دراساتهم العلمية حتى يتسنى لهم تطوير هذه الكلية ويشاركوا في تطوير هذا الوطن الحبيب المعطاء. فخور جدا كوني موجود اليوم في حرم جامعة الأزهر حيث بدأت، وأتمنى لهذه الجامعة العريقة مزيدا من التقدم والرقي والنجاح، وأن تكون وتستمر بكونها شعلة للأجيال القادمة حتى تنشر العلم والوعي بين أبناء هذا الشعب الحبيب المعطاء.
نعيم وضياء يا وطني الأكرم، نون قاف فاء كي تبقى الأعظم. نور نعيم وضياء يا وطني الأكرم، نون قاف فاء الأعظم. راية العلم تسور ينحني لها الوجود، ليس للعقل حدود. راية العلم تسور. أنت الأزهر شامخ، علم شمس نور، صوت الحق الصارخ، نور يعلو نور. أنت الأزهر شامخ، علم شمس نور، صوت الحق الصارخ، نور يعلو نور. إننا للعلم قمنا نبتغي ذاك المراد، إننا بالعلم جئنا نعتني كل الصعاب. إننا للعلم قمنا نبتغي ذاك المراد، إننا بالعلم جئنا نعتني كل الصعاب. جيل نصر نحن هيا، علمنا أصل الهوية. 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 ها هنا لب القضية، ها هنا لب القضية. نحن جيل التضحيات، نحن جيل التضحيات. ها هنا لب القضية، ها هنا لب القضية. نحن جيل التضحيات، نحن جيل التضحيات. أنت الأزهر شامخ، علم شمس نور، صوت الحق الصارخ، نور شمس النور صوت الحق الصارخ نور يعلو نور راية العلم تسور ينحني لها الوجود ليس للعقل حدود راية العلم تسور.
السلام عليكم.
عليكم السلام دكتور أحمد.
أيوة.
دكتور هاني.
هادي. هلا دكتور هاني.
كيف حالك يا دكتور أحمد؟
الله عليك.
أهلا وسهلا. دكتورة لورين أهلا وسهلا.
Shall we start?
Yeah, we can start.
Good morning, good afternoon.
Good afternoon, everybody. Good afternoon, Dr. Ahmed. So it's my honor to invite Dr. Ahmed Najm. Dr. Ahmed is assistant professor in public health at Al-Azhar University of Gaza, and also Dr. Hani Dakka, assistant professor in microbiology. So the virtual The virtual stage is yours in order to moderate the poster session.
Yeah. Good afternoon, everybody, distinguished guests, respected colleagues, researchers, presenters, ladies and gentlemen. On behalf of the organizing committee, it is my great pleasure to welcome you to the poster sessions of the third International Conference on Pharmaceutical Science. We are delighted to have you with us today, and we sincerely appreciate your participation and interest in advancing pharmaceutical science and healthcare research. My name is Dr. Ahmed Najm, and I have privileges of serving as one of the moderators of these sessions. It is also my pleasure to co-moderate these sessions alongside my esteemed colleague, Dr. Hani, and I honored to facilitate today's presentations and discussion. Before we begin, I would like to emphasize that posters presentations are an essential component of every scientific conference. While oral presentations provide an opportunity to present research in greater details, poster sessions as well offer a unique platform for focused scientific exchange, interactive discussions, networking, and constructive feedback. Many innovative ideas, future collaborations, and research partnerships begin during poster sessions. Each session today presents months and often years of dedicated work by researchers who have contributed valuable knowledge to pharmaceutical science and healthcare. I encourage everyone attending to actively engage with our presenters, ask questions, exchange ideas, and provide constructive comments. These interactions enrich both the presenters and the audience and contribute to the conference success. Today, we have 6 excellent poster presentations covering a wide range of important topics, including traditional herbal medicine, cancer biology, medication safety, pharmaceutical service quality, molecular oncology, child nutrition, and conflict-affected setting. This diversity reflects the multidisciplinary nature of the pharmaceutical science and demonstrates as well the commitment of the researchers from Palestine and several international institutions as well to providing health and outcome through high-quality research. To all our presenters, thank you for sharing your work with us. We appreciate your efforts and look forward to learning from your research. To our audience, thank you for your presence and participation. Your questions and comments, discussions are an important part of this session. I kindly ask each presenter to adhere to the allocated presenting time. We will provide a gentle reminder when your time is nearly complete, followed by a brief opportunity for questions and discussions. Without further delay, let us begin our poster sessions. Our first presenter is Mr. Armin Hamidat, who will present her work entitled Knowledge, Attitude, and Practice regarding traditional herbal medicine used among patients with metabolic syndrome in Hebron district, a cross-sectional study. Ms. Amin, the floor is yours.
Okay, Assalamu alaikum.
Alaikum salam. Welcome.
Thank you. Good afternoon. Okay, but first I will share you the posts.
Yes, you can do.
Okay, thank you. Okay.
Yes, it's okay.
Okay. Good afternoon.
Good afternoon.
Honorable panel members, esteemed researchers, and valued attendees. My name is Arin Hamidat. I am a master's student in medicinal plants and herbal products at Hebron University, Palestine. In addition, I work as a teaching assistant at Hebron University and as a community pharmacist. I am honored to share with you the results of my research entitled Knowledge, Attitude, and Practice regarding traditional herbal medicine using among patients with metabolic syndrome and Hebron district. A cross-sectional study. Metabolic syndrome is one of the most important public health challenges worldwide. It is characterized by a cluster of metabolic abnormalities that substantially increase the risk of cardiovascular disease, type 2 diabetes, and premature mortality. In Palestine, traditional herbal medicine remains an integral part of healthcare practices and is commonly used as alongside conventional medications. However, despite its widespread use, evidence regarding patients' knowledge, attitudes, and practice, particularly among individuals with metabolic syndrome, remains limited. Recognizing this important gap in this literature, the present study aimed to assess the knowledge, attitudes, and practices regarding traditional herbal medicine among patients with metabolic syndrome in the Hebron district and to identify the factor associated with its use. I would now like to briefly outline the methodology used in this study. This was a quantitative analytical cross-sectional study conducted in the Hebron district, Palestine, between February and May 2026. A total of 412 adults diagnosed with metabolic syndrome were recruited from primary healthcare centers, endocrinology, outpatients, clinics, and community pharmacies. Data were collected using a structured questionnaire, and statistical analysis was performed using SPSS version 29, including descriptive statistics and binary logistic regression. Let's now turn to the key results obtained from the study. Nearly around 46.8% of participants were current users of traditional herbal medicine, demonstrating that herbal medicine represents a common self-care practice among patients with metabolic syndrome. Overall, participants showed a positive attitude toward herbal medicine with a mean attitude score of 3.6 out of 5, reflecting a high level of cultural acceptance and trust. A particularly noteworthy finding was the presence of a significant knowledge safety gap. Although many participants recognized the commonly used medicinal herbs, their scientific knowledge was limited. A considerable proportion were unaware of potential side effects, herb-drug interactions, and the active constituents of medicinal plants. This finding is particularly important because the majority of participants were receiving multiple prescribed medications. Regarding herbal practices, cinnamon was commonly used for blood glucose control, green tea for weight loss, garlic for cholesterol reductions, and hibiscus for blood sugar management. Most herbs were obtained from traditional herbal shops, while family members represented the primary source of recommendations. In addition to these observations, the regression analysis highlighted several important determinations of herbal medicine use. Binary logistics regression demonstrated that positive attitude was the strongest independent predictor of herbal medicine use, increasing the likelihood of use by almost threefold. In addition, greater disease severity significantly increased the probability of herbal medicine use. Interestingly, knowledge level and educational level were not significant predictors, suggesting that patients' decisions are driven primarily by cultural beliefs and disease burden rather than scientific knowledge. These findings provide a valuable insight into the current pattern of herbal medicine use among this patient population. Traditional herbal medicine is widely used among Palestinian patients with metabolic syndrome. While patients generally trust herbal medicine, insufficient awareness of herb-drug interactions and safety issues may expose them to preventable adverse outcomes, particularly in the context of polypharmacy. Based on the study finding, several practical recommendations can be considered. Traditional herbal medicine should be integrated into routine primary healthcare. Healthcare professionals should actively counsel patients regarding herb-drug interactions. Herbalists should receiving appropriate regulation and safety training. and further clinical research is needed to strengthen the evidence on the safety and effectiveness of Palestinian medicine. To summarize, integrating evidence-based traditional herbal medicine into chronic disease management has the potential to improve patient safety while respecting the cultural practices of the Palestinian community. Thank you for your time and interest in this work. I really appreciate your attention and welcome any questions and comments.
Thank you very much, Ms. Irene, for that interesting presentations and for sharing your valuable research with us. Any questions for Ms. Irene?
Yes. Dr. Ahmed?
Yeah.
Thank you very much for this attractive presentation. I have just one question for Doctor, that what is your imagination for the future of herbal medicine in Palestine? That we have a negative attitude about this. Most of people believe on drugs only. What is the solution for that, please?
Ms. Irene?
Dr. Irene, you hear me? Dr. Irene, are you still?
Ms. Irene?
Ms. Irene, you hear me? I think there is a problem, Dr. Ahmed.
Maybe internet failure. Yeah, let us go to the second presenter if there is no question. At this point, I would like to hand over the moderation to my colleague, Dr. Hani, who will introduce the second presenter. Handy, Dr. Hani, the floor is yours. Yeah.
Thank you very much, my brother, my colleague, Dr. Ahmed. I welcome you and Dr. Loreen also. I'm very happy and proud to talk with you from my displaced tent in Deir Balah. And this is the will of the Palestinian that can never be damaged. The second presentation session is for Anis Fatima from Ethiopia University in India, and the presentation is entitled about the cancer microbiome axis, unraveling dysbiosis for targeted interventions. I hope that the presenter is available, and welcome. Anis Fatima from India. Do you hear me? Dr. Anis, are you available? I think she's or he's available. Hello, Dr. Anis. Yes, now. Welcome, Dr. Anis. We are ready to hear for you. Please, could you make a share for your presentation? Make share for the screen. Do you hear me? I think we have a problem, a technical problem. We didn't hear you, Dr. Anis. We didn't hear you well. Please, if you have any technical problem.
Dr. Hani, can you go forward for the next, please?
Okay, I think Dr. Ahmed will go over there. Session 3. Dr. Ahmed, we didn't hear you.
دكتور أحمد المايك لو سمحت، وعلى أساس إنه ننتقل للمتحدث التالي لو سمحت يا دكتور أحمد.
نعم نعم.
Sorry, Assalamu alaikum. Sorry for that inconvenience.
عليكم السلام. It's okay now. Please, Dr. Anis, can you make share for the screen? Hello, hello. We hear you.
Now you can able to hear me. Some problem in my system. That's why I can't able to speak
Okay,
right
now it is okay. We hear you. Just you can make a share for screen, please. To yes, now it's okay. Dr. Anis, please make a share for the screen to see your presentation.
Sorry, some problem in my system. I can't able to share. I will come back within 5 minutes. Sorry for the inconvenience. I really sorry for that.
Okay. Dr. Ahmed, you can present the third poster session for Muhammad Dayan from India. Dr. Ahmed, you hear me? No. Dr. Muhammad
Sorry for the inconvenience. I can't able to share my screen in this device. So give 5 minutes. I will come back with you.
Okay, thank you very much for you. So the third poster session is for Dr. Muhammad Dayan from Mysore, India. And the presentation of this poster is entitled about the development and administration of a steroid alert card, a color-coded safety tool to enhance safety and awareness. Dr. Muhammad Dayan from Mysore, India, do you hear us? Are you with us?
Sir. Assalamu alaikum. I'm here.
Alaikum assalam wa rahmatullahi wa barakatuh. We hear
Thank
you.
you. Thank you for the opportunity.
Thank you very much for you. Yes. You can share the screen, please, Dr. Muhammad.
Yes,
Dr.
sir. I'm doing
Muhammad,
that
thank
right
you
now.
for your joining us. The floor is yours now.
Sure, sir. Can you see my screen, sir?
Yeah, about, yes.
Now I hope you can see my screen.
Yeah, we are about to see your presentation, inshallah. Try again, Dr. Muhammad, if you can try again.
Yeah. It's okay now. Yeah, it's okay.
Yeah.
Yeah, now, it's okay.
Yes,
you can
sir.
talk. Dr. Muhammad, you can talk.
Okay, so I am Dr. Mohammad Dayan Khan. I'm a PhD scholar from the Department of Pharmacy Practice, JSS College of Pharmacy, Mysuru, India. I am currently working on my PhD in the Department of Pharmacy Practice, where I am working in the development and implementation of a steroid stewardship program for the Department of Rheumatology. So we all know what steroids are. They are drugs that mimic the hormone cortisol, which is necessary to treat autoimmune diseases. This drug is widely used in departments such as your pulmonology, rheumatology, or dermatology. So here the concept is to use these drugs in a proper way. As we all know what steroids, what stewardship is. So we are bringing that same concept into these steroids, as these drugs were mainly used for a very long period of time in these respective departments. We try to maybe bring down the patients who are using these drugs, or at least to minimize the adverse effects related to this. And when it comes to steroids, we cannot stop the steroids like, what do you say, completely in one go. So you have to bring down the dose, which is called tapering. So you need to taper the same thing. So for the same reason, what we came up with to implement the steroid stewardship program, where we took up the formation of the alert card that was a part of the whole stewardship program, including the guidelines, the formation of the committee, and also this card was made. So coming to the methodology, there was a committee that was multidisciplinary and was formed under the name Rheumatology Corticosteroid Stewardship Committee. We came up with an idea of the alert card and the guidelines. We were kept in the committee, and they were approved by the committee. Later, the hospital, our hospital, that is the JSS hospital, accepted all this, and they helped us in the implementation of the card and use of the same in the Department of Rheumatology. So coming to the results, what we obtained, here we can see the formation of the committee. This was the steroid stewardship committee. Here the committee was mainly multidisciplinary, where we had accommodated various experts from the various departments, such as the pharmacy practice, the rheumatology, the nursing staff, the hospital administration, and also we had external advisors who were experts in rheumatology, and as well as a community representative. After the formation of the same, we came up with the idea of the development of an alert card. So this is basically not digitalized card. Why? In this era of digitalization, we would think, why not a digital card? So the patients, whoever comes to our hospital, is mainly from the rural side. So most of them have no smartphones or whatever to carry with them along. So they have basic phones, and it was very difficult to teach them all the digital ways. So we came up with the form of this alert card, where this is, you can see the first photo, that is the front part of the card, where we have divided the patient into 3 categories: the red, yellow, and green. This was basically taken from the signal kind of, we know the traffic signals. So here the red color represents the patient who is on long term. So basically we consider a patient on long term when he is taking steroids for more than a period of 3 months. So when a person is taking steroids from around 3 months to up to 1.5 years or 2 years, we consider him as in the yellow category. And above whatever that comes about 2 years, we take him to the red category. And the patients who have stopped taking steroids will go within past 12 months go into the third category, that is the green category. As if the patient has already stopped the use of steroids, yet he has chances of developing some adverse effects. So this is the reason why we have kept the green category. That was the front part. The back part is we have all the necessary instructions for the patient or the caregiver or the doctor with the emergency number, and as well as a QR code. As this card is very compact, we're not able to put in all the information in the compact card. So basically, to overcome that, we came up with a QR code, which when scanned leads to a document, that is a safety guide document, that explains in detail the 3 colors, that is the red, yellow, and green, as well as the sick day rules. Sick day rules is whenever a person goes to any dentist or visits a doctor for any illness, or he is going for some minute surgery, operation, or any procedure, is called a sick day. So there they have to follow rules. As they are not able to stop the steroids immediately, they need to take or follow the permission from the doctor or taper it off properly. So this is what the QR code guides the patient. not only the patient, the caregiver and the doctor. So basically forming these kind of initiatives, that is the guideline or the committee or the alert cards in terms of the steroids, similar to antibiotics, can help because these patients are using them for a very long time. And coming to the applicability of this card, we have already distributed or given more than 200 cards, and the people are understanding the effects caused by the steroids, or it may be what do you say, the harmfulness, or how to prevent those effects of the steroids with the help of the card, and the patients are carrying these cards with them. Basically this is being implemented, and all the guidelines and also are being implemented now. So the conclusion is making these efforts towards the steroids can help improve the patient's outcome and as well as the knowledge of the patient regarding the same. And finally, I would like to thank the organizers for giving me this opportunity, and also I would like to thank my supervisors and the hospital who is helping me with carrying out all my PhD work in their hospital. So thank you everyone. Thank you.
Thank you very much.
Thank you, Dr. Muhammad.
Thank you, Dr. for joining us.
If there is any question.
There is no. Dr. Hani, next one.
Okay, thank you.
Yeah, you're the next.
Now we will transfer to the fourth poster session here from Dr. Arwa Tabash from Gaza, Palestine, about evaluation of patient satisfaction with the quality of pharmaceutical services provided in governmental primary health care centers in Gaza Strip. For known, Dr. Arwa has a Bachelor of Pharmacy from Al-Azhar University, also has a master's degree in health management from Islamic University. She's working as a nutrition officer in Save Children International. Welcome, Dr. Arwa, we hear you. Dr. Arwa. Dr. Arwa, are you available?
I think so. I think she's available. Dr. Arwa?
Arwa? We are waiting.
Yes. Yeah.
We hear you, Dr. Arwa. Go ahead.
I want to share my poster screen.
Please make a share for screen.
Yeah, okay.
Okay, go ahead, Dr. Arwa. We see your presentation.
Okay. Good afternoon, everyone. My name is Arwa Tabash. I would like to begin my extending my warm greeting to you all with special appreciation to my respected teachers and colleagues in my college. This poster represents my master research and focused on an important managerial aspect of pharmaceutical services based on understanding the management and pharmacy are two sides of the same coin, when the ultimate goal is improving the quality of healthcare delivery. My poster, my research, patient satisfaction with the quality of pharmaceutical services in governmental primary healthcare center in Gaza Strip. Pharmaceutical services are a core component of primary healthcare as strongly influence the quality of care and patient outcomes. Patient satisfaction is the key indicator of the service's quality and reflect pharmacist performance. In the Gaza Strip, in the Gaza Fuse studies have assessed pharmaceutical service quality and patient satisfaction in aim to evaluate patient satisfaction with the quality of pharmaceutical services in governmental primary healthcare center in the Gaza Strip and examine the relationship between service quality and patient satisfaction. The design of the study, descriptive analytical cross-sectional study. The study conducted in period between April to August 2023 in governmental primary healthcare center. The study include 385 patients, provisional stratified sampling, the tools structure close-ended questionnaire, cervical, this cervical scale measuring the service quality. Analysis SPSS version 26. I use cervical dimensions as 5 cervical dimensions: tangibility, reliability, responsiveness, assurance, and empathy. The finding showed that 8.06% of participants rated the quality of pharmaceutical services as high, while 78.8% reported high satisfaction level. The results also demonstrate a statistically significant positive relationship, confirming that improving service quality directed contribute to enhance the patient experience and increase satisfaction. Based on this finding, the study recommended strengthening the healthcare staff communication skill, reducing waiting time, improving pharmacy workflow, adopting digital solution in pharmaceutical services, and establishing continuous system for monitoring patient satisfaction and the quality indicator. I like to highlight to important point. This study just days before the outbreak of the war. These findings reflect the healthcare situation at this time. However, the current situation unfortunately changed due to the war, including damage the healthcare infrastructure, shortage of medicine and medical supply. So conducted updated studies to assess pharmaceutical services quality under the current situation, focusing on medication availability, accessibility of service, waiting time, continuity of care, and satisfaction of both patients and healthcare provider. Furthermore, future research should explore strategies that enhance healthcare system resilience and support the improvement of pharmaceutical services during emergency and crisis, providing evidence to guide decision maker in rebuilding and developing healthcare services in the future. Thank you for kind attention.
Next, Dr. Hani, next. دوست الفيش
دكتور أحمد، الدكتور هاني واضح إنه ممكن يكون مش موجود، في إمكانية تقدم أنت المتحدث التالي لو سمحت؟
سامعك دكتور، سامعك، سمعتني؟
تفضل دكتور هاني المتحدث.
The fifth presenter, yeah.
نعم, the fifth poster session from Subhajit Ghosh from University in India about integrative transcriptomimetic and proteomimetic profiling reveals CDK1 and CCP1 as a prognostic biomarkers and therapeutic targets in triple-negative cancer patients, TNBS. Dr. Subhajit, do you hear me? Are you available? Hello?
Yes, yes, you hear me. I hear you. Yeah, yes.
Yes. Okay. The last. Do you hear me, Sajjata? Do you hear me?
Yes, sir. Can you hear me?
Okay, please, we hear you, but please make share for the screen. Okay.
I hope you can see my screen, sir.
Okay, now go ahead. Go ahead, Dr. Sajjata.
Good afternoon. Myself, Sujit Ghosh. I'm a postdoctoral researcher at Yenipoya University, India. I'm presenting our work on integrative transcriptomic and proteomic profiling that reveals CDK1 and CCNB1 as prognostic biomarkers and therapeutic targets in triple-negative breast cancer. So, triple-negative breast cancer is a heterogeneous type of breast cancer. It accounts for almost 20% of all breast cancers, and it has the poorest prognosis among all breast cancer subtypes. The global projected dataset shows that it can reach up to 3.2 million per year by 2050, and it is a burden to all healthcare professionals. And the molecular heterogeneity actually complicates the clinical management as well as our therapeutic approaches. So our objective was to identify the prognostic genes in TNBC via integrating multiple omics technologies, including transcriptomics as well as proteomics technologies. We also wanted to validate the biomarkers across cross-omics platforms and development of prognostic risk models as well as identify some therapeutic candidates for TNBC. Our methodology included differential gene expression analysis using RNA-seq datasets, followed by peptide network analysis to identify the core hub proteins, then functional lineage analysis. After that, multi-omic validation using external sets of transcriptomics, proteomics datasets, followed by prognostic modeling using server analysis, regression modeling, and we also tried to identify the core genomic alterations behind this disease, and identification of few drug candidates which can be utilized against this disease. So we have identified a total 500 differentially expressed genes, 250 upregulated and 250 downregulated, and we have identified 10 core hub proteins among these differentially expressed genes using multiple kind of PPI network approaches. We have also listed these 10 hub genes like CDC-A8, CCNB1, or TOP2A, and used different approaches. For example, gene-gene interaction networks, as well as their expression in tumor metastatic as normal conditions. So you can see we have found that these genes are highly dysregulated in tumor conditions at transcriptomic level as well as at proteomic level. This is the slide where we found that these genes also show different in their expression patterns in different cancer stages as well as different metastatic stages. And through our proteomics approach, we found that these genes also dysregulated in proteomic level, and through the tissue expression analysis, we can see these genes are highly expressed in cancer tissues as well. Followed by this, we have used server analysis using TCGA datasets and found that all these 10 genes are highly important in terms of patient survival. They show a high mortality rate in terms of their expression. And through our prognostic nomogram, we found that several genes, including like TOP2A, CDK1, or CCNB1, were highly important for overall patient survival. These genes, among these genes, genes like EURAK or TOP21 showed highest percentage of alteration, genomic alteration. There are different kind of genetic alterations found, like missense mutation as well as PLAS mutation, etc. And through our literature review and drug repurposing, we identified a drug called AG024322 that is a kinase inhibitor that is highly effective against these two target hub proteins like CDK1 and CCNB1. So in conclusion, our integrative multiomics analysis identified dysregulated mitotic regulatory network that is also a hallmark of TNBC, that is well-established hallmark for breast cancer as well. That and we identified 10 hub genes that can be regulating all these processes. All these genes showed consistent upregulation in tumor and metastatic tissues, both at transcriptomic as well as in proteomic levels. Among these 10 genes, CDK1 and CCNB1 found to be the strongest prognostic biomarkers. Their high expression correlates with overall poor survival. Our multivariate nomogram analysis also confirmed the prognostic collective prognostic utility of all these 10 hub genes as a panel. And CDK1 and CCNB1 found to be promising therapeutic targets, and AG24322 found as a key drug that can effectively interact with these proteins and that can help as a therapeutic in this TNBC. Thank you very much.
Thank you, Dr. Subhajit Ghosh from Minnelovia University for this attractive poster sessions. Actually, the breast cancer is a vital issue and very critical issue. So many research should be implemented about this. If anybody has a question before transforming to the last poster session. Anybody raise hand or have any reaction or comment about this poster? Okay, thank you very much, Dr. Subhajit Ghosh from India. Thank you for your sharing.
Thank you, sir.
Okay. Dr. Ahmed Nijim.
Dr. Hani, our final presenter in this poster session is Christina Murtaja.
Okay, now.
From Palestine, she will present her study entitled Malnutrition and Anemia Among Children, aged 6 to 24 months in conflict-affected areas, Gaza. Ms. Lina, thank you for being with us today. The floor is yours, Lina.
Okay. Ms. Lina, you hear me? Lina Murtaja, you hear me? You have shared the screen, but we didn't hear your voice. Ms. Lina, you hear me?
First. Yeah. Ms. Lina?
I think she has a technical problem, Dr. Ahmed. Ms. Nina, you hear me? We have a message, Dr. Ahmed. We have a message from Dr. Anis Fatima. She is ready now from Unobia University. Dr. Anis Fatima from Unobia University. Are you ready?
Yeah, yes, sir. I'm ready. Assalamu alaikum.
Alaikum
Yeah.
salam. We hear you. Go ahead, Dr. Anisa. Go ahead. We hear you.
Yes, sir. My screen is visible, sir.
Go ahead, Dr. Anisa Fatima from Minob University in India. We hear you well. You can start.
Okay. Yeah, myself is Anis Fatima. I am working under Namul Hassan Mahadar at Enapoya University. Right now, I am presenting my metastatic pathway linking dysbiosis to tumorigenesis and immune modulation is my review article. So I am like to present to this conference. My aim of the study to provide a comprehensive overview and of recent advances in host microbiome interaction, how gut microbiota composition drives oncogenesis, influence treatment response, and inform the emergence microbiota targeted therapies. Let's I start the introduction part of my presentation. Cancer remains a major global crisis. It's causing 10 million deaths annually. It is a traditional derivation to genetic defects and environmental factor, but recent studies are showing that microbiome actively drives cancer risk, progression, and therapeutic outcome. That is a homeostasis is maintaining gut barrier interacting digestion and promotes anti-inflammatory product. Dysbiosis trigger a chronic inflammation and comparison epithelial barrier and generate oncogenic toxic metabolites. This state is linked directly to inflammatory bowel disease and metabolic syndrome, diabetes and cancer pathogenesis, and etc. The microbiome is critically reshape host response to chemotherapy, radiotherapy, immunotherapies, immune checkpoints, altering the both efficiency and toxicity. So, the gut microbiome dysbiosis and role in tumorigenesis when the gram-negative microbes are enter into our guts, it leaking that making a leaking a guts barrier, and it's inflammate is causing a pro-inflammatory cytokines that causing chronic inflammation and that that help to tumorigenic pathway. Then it's also alkylating DNA alkylation and DNA alects to DNA damage. The two all interact into a tumorigenic pathway, then it causing a tumor cells. So and the dysbiosis of gut microbiome also help to causing a cancer disease. The role of gut microbiome in immune modulation and cancer regulation. What are the gram-positive bacteria in our gut, like intestinal microbiome, like lactobacillus and bifidobacterium? All those things are have the short chain fatty acids that help to getting a immunotrophils and B cells in our body. Then the macrophages and TNF alpha, those things are helping to anti-inflammatory and fight against the inflammations. Then it's help to uh, then it's help to apoptosis pathways. Then it's reduce your tumor cells. So emerging microbiota on target therapies or first one, fecal microbiota transplanted. That means we have to take the donor samples and progressive via colonoscopy that restoring a diversity. Probiotics and prebiotics things, specific bacteria strains and dietary fibers that help to modulate a community composition. Then synthetic communities and engineered bacteria to modified microbes to deliver drugs that targeted drug delivery. Personalized nutrition and metabolic therapies plus short chain fatty acid that interact into a metabolic pathways that improve a patient outcome and cancer treatment stratification. The conclusion of my presentation, the gut microbiome represents both potential biomarker and therapeutic target in cancer. Integrative mechanistic understanding the clinical and computational approach will be essential for realizing precision oncology statistics and harness the microbiome to improve prevention, treatment, and long patient outcomes. Thank you.
Thank you, Dr. Anis Fatima from Unobia University in India. If anybody has a question. Thank you very much. The last poster from Lina Murtaja. Lina is ready. Dr. Ahmed, you can. Dr. Ahmed, we didn't hear you.
Our final presenter of this photo session is Ms. Lina Murtaja Styles.
The last poster from Lina Murtaja.
I'm here, you.
Yeah. The floor is yours, Mr. Lina.
الصوت واضح كده؟
Yeah. It's okay. Go ahead.
You are welcome. Good afternoon, distinguished colleagues, researchers, and guests. My name is Lina Fadil Murtaja. I hold a master's degree in maternal and child health nursing. I'm a registered nurse with the Palestinian Ministry of Health, and I'm currently a lecturer at the University College of Applied Sciences. It's my pleasure to be here today to present my research entitled Malnutrition and Anemia Among Children aged 6 to 24 months in conflict-affected Gaza. The period between 6 to 24 months of age represents a critical window for child growth and development. When children don't receive adequate different nutrients, they become vulnerable to different forms of malnutrition, including stunting, wasting, underweight, and anemia. During this period, children depend on appropriate complementary feeding alongside breastfeeding. In Gaza, the ongoing conflict has significantly worse living conditions, poverty, food insecurity, limited health care services, and repeated displacement have greatly increased the risk of malnutrition among young children. Therefore, our study aimed to assess the nutritional status of children aged 6 to 24 months attending primary healthcare centers in the Gaza Strip and examine the factors associated with malnutrition. This study used a cross-sectional design. The study included 250 children aged between 6 and 24 months together with their mothers. The research was conducted at Shuhada Daraj Primary Healthcare Center in the Gaza Strip. Data were collected during 2025 using a valid and reliable structured interview questionnaire. The questionnaire gathered information about socio-demographic characteristics, infant feeding practices, maternal nutritional knowledge and practices, and children anthropometric measurements and nutritional status. As illustrated in the conceptual framework, child nutritional status relation to several interconnected dimensions, measured through indicators, stunting, wasting, underweight, and anemia, was examined relation to several factors, women knowledge and practice of breastfeeding and complementary feeding, socioeconomic factors of the family, demographic characteristic, child demographic factors, and role of primary healthcare center nurses in maintaining child health. Now moving to the main findings of this study. The bar chart showing prevalence of malnutrition among 250 children included in this study. 12.5% were stunted, 5% were wasted, 21% were underweight, and 60.9% were suffering from anemia, making anemia the most common nutritional problem identified in this population. These findings demonstrated that malnutrition remains highly prevalent among young children in Gaza, especially anemia. First table showing significant factors associated with malnutrition. Were found statistically significant associations between child age and both stunting and wasting. Underweight was significantly associated with food shortage and previous treatment of malnutrition, while anemia was significantly associated with the father's employment status. All these associations were statistically significant with p-value below 0.05. Second table showing maternal knowledge and practice scores. The mean knowledge score is Y 9.5 out of 11, and the practice score averaged 51.7 out of 60, indicating a generally good level of awareness and engagement among participants. Third table showing the role of primary healthcare centers nurses in nutritional education. Notably, 70% of mothers reported receiving nutritional advice, which strongly suggests that primary healthcare center nurses are playing a key role in supporting maternal nutrition. In conclusion, this study demonstrated that children aged 6 to 24 months attending primary healthcare centers in Gaza experience a considerable burden of malnutrition. Among all nutritional disorders, anemia and underweight were the most common. The nutritional status of children was significantly associated with age, food shortage, and several socioeconomic factors. In addition, mothers with higher nutritional knowledge generally reported better feeding practices. In recommendation based on these findings, the study recommends the strengthening early nutrition intervention programs, enhancing caregiver nutrition education, improving nutrition counseling with the primary healthcare centers, and addressing household food insecurity and promoting the early detection and management of malnutrition among young children. Thank you very much for your attention.
Thank you very much, Ms. Nina, for your excellent presentation and for addressing such an important public health issues affecting vulnerable children. We appreciate your valuable contribution to today's session. Any questions, please?
Thank you, Dr. Ahmed. Thank you for all.
Thank you, Ms. Lina.
I think we have to finish, Dr. Ahmed. There is no question.
Thank you.
Thank you, Dr. Ahmed. Thank you, Dr. Danke, for your moderating this session for posters. Now we are going to transfer to our third session.
Yeah, thank you very much.
Thank you very much.
Thank you
for all. Thank you for all.
Dr. Hanis, thank you for.
Thank you for you, Doctor. Thank you.
Okay. Good afternoon. Good afternoon, dear participants and attendees. We are going to open the third session in the second day of our conference under the title Immunology and Oncology. Please allow me to welcome and introduce our moderator of this sessions, Professor Mahmoud Sirdah. Professor Mahmoud Sirdah is a professor of blood pathophysiology and molecular genetics at Al-Azhar University, Gaza, Palestine. He is the former Dean of Scientific Research and the former Dean of the Faculty of Science, Al-Azhar University, Gaza. Professor Sirdah is a counselor of hematology and molecular genetics at national and regional organizations. Professor Sirdah is a member of the Experts Advisory Committee of the Palestinian National Institute of Public Health. Professor Sirdah has published more than 50 research articles in international peer-reviewed journals and international conferences. Our second moderator is Dr. Muhammad Taha. Dr. Muhammad Taha is the Dean of Faculty of Pharmacy and the chairman of the conference. Dear moderator, the mic is yours.
Thank you, Professor Ehab. Thank you very much. Dear colleagues, distinguished researchers, ladies and gentlemen, it is my great honor and pleasure to chair the third session of the third International Conference on Pharmaceutical Sciences, together with my friend and colleague, Dr. Muhammad Taha. This session features one keynote lecture followed by 4 research presentations. The keynote lecture has been allocated 40 minutes, including around 10 minutes for questions and discussion. Each research presentation will be 15 minutes in duration, including 5 minutes for audience a question. It is now my great pleasure to welcome our keynote speaker, Professor Inam Hassan Madar from the Center for Integrative Omics Data Science, Unipoya University, India. Professor Madar is a distinguished scientist and academician specializing in cancer proteogenomics with internationally recognized expertise in translational oncology, bioinformatics, and multi-omics data integration. His research has made significant contributions to advancing precision medicine and personalized cancer therapy. The title of Professor Madar's keynote lecture is Multi-Omics in Precision Medicine, a Personalized Cancer Therapy Approach. Ladies and gentlemen, please join me in giving a warm welcome to Professor Inam Hassan Madar. Now, please, Dr. Madar, the floor is yours.
Thank you, Professor Sidra. Am I audible now?
Thank you. Yeah, it's okay. Start. Please share your screen.
Yes. Yep. So the screen is visible now?
Yeah, okay, please start.
Yeah, thank you. So hello everyone. So my name is Dr. Inam, and we are working on like precision oncology and then like personalized medicine. And I'll start with the basic because a few of my students also there, so that like it could be easy for them to understand the things that we are discussing today. We'll be talking about like multi-omics, where we like genomics, transcriptomics, proteomics, metabolomics, and sometimes like drug discovery, biomarker discovery, and the things that we do there with the clinical patient samples directly during the surgery. So we go and get the samples, and then we analyze those samples on wet lab level by extracting the DNA. And then proteomics for the mass spec and everything. While doing that one, like we faced few challenges where we couldn't able to get directly therapeutic targets and then biomarkers. So that time we started to develop on few technological aspect of that with a collaboration from US and the country, and while I was in South Korea, in Korea University. So initially we'll start recording like just like what is the cancer is about. It's like usually the natural process where like apoptosis happens, where in the case of like cancer cell divisions keep completely growing. And then we'll be talking about two particularly important genes, like which is oncogene, which causes cancer if the expression of that specific gene is higher. So that may lead to the cancer in the pathway. And naturally, like we do have like tumor suppressor gene, where gene, I mean those particular genes helps in like suppressing the cancers in a few conditions. And most of the times what happened in the recent past, so the drug which is like already there in the market, so sometime the specific drug was not like responsive to the few percentage of the patients initially. So as the day passes, like due to the mutation or hypermutation we call that, and due to that one, like the response is very low. If you take about like for example 100%, initially it was like 10% there is no responsive. As the decades and then like the generation passes, like the non-responsive patient, but the specific drug which is already like in the market and then FDA approved, which is not due to maybe the DNA changes in the patients. So there are different causes of the cancer, which people can lead, but mostly we talk about like hereditary and then like acquired mutations. Because of that one, like the cancer initiation can be there as well. And in some cases, like where from the like as you get age, so the accumulation of the cancer changes, DNA changes, or like genetic mutation changes are there, which Okay. Hello. I'm audible. Hello. Hello.
Yes, you are. Yeah.
Oh, okay.
We can hear you, but we are listening to you.
Okay. Thank you. Yeah. So as the time passes, the person ages. So the time the accumulation of those genetic changes is higher, and the chance of getting in the older age also. That's one of the conceptual things that we've been considering in some cases. And second part, where metastases happen from the primary to secondary, for example, from head and neck to lung cancer, or gastric cancer to lung cancer, or even some other cancers which may lead to the lung cancer as well. Primary, we were focusing on patients who are suffering from gastric cancer or the breast cancer, and then leading the metastasis to lung cancer. In most of the cases, particularly in India, and South Korea, so we saw the patients who are suffering from head and neck, and we provide the treatment to them, and the patient undergo surgery, and also chemotherapy, radiotherapy, based on the condition, the stage of the cancer, and then the stability of the patient. But after some time, we see the same patient after the patient is recovered, and then get back to home, in 6 months, 8 months, or 1 year. But there are huge patients that we could able to see. So within the duration of 3 years to 5 years, the same patient get admitted with the metastasis happens on that patient, reoccurrence in the lung cancer. And that's what one of the primary thing that we started to work on, why metastasis also is important, that reoccurrence is one of the difficult tasks that we had to handle in the case of oncological patients. So we started working on some genetic aspect of those, which could be made as a biomarker and can be of useful as a prognosis part of it, which could be help in saving as much as patients, based on the novel panel that we can develop on that. So, but as we see, there are estimated more cases out there from the breast cancer and the prostate. But in what happened, the mortality, which we could able to see higher is in lung and bronchus, as we can able to see here. So both in the male and the female as well. But the incidence is higher in higher breast, and higher in the case of female is the breast, and then prostate is the male. But mortality is where we are much concerned about, where we thought we had to put our much focus on that. And one more aspect of in the patient that we get it during in oncology department. So as we always see, if we diagnose this very little late, so the cancer stage and other things are get worsen, and it's kind of very difficult to get treatment and give the success rate higher when compared to the one which get yearly diagnosis of that. So that's why we call that one as yearly detection is really the best thing that in the case of cancer patient, to save their life, and then it also helps in stop recurring as well. So as I told, there is not always the cancer that we have some medicine in the market. So there are only almost only 38 8% of the medicines which is right now in the market, which is not responsive in the case of depression. But in the case of an asthma, 40% is not responsive. So which is statistical that we have did, and then there are few research articles also published on that. But in the case of cancer, because the heterogeneity is very, very high, the kind of Alzheimer and then cancer, so even the prescribed drug, which is FDA approved and everything's done, but still at least 95% of the in cancers, so it's not targetably which can be of confidently curable or something like that. So that's where we have to talk about on personalized medicine, where specific conditions of the genetic landscape or the physical conditions of patients, other things. So we have to start working on personalized medicine on few chronic diseases like cancer. So for that one, initially into for about 2015, so people started working on precision medicine, so which is the individualized approach. So if you have one specific patient, like where we take the genetic makeup, clinical history, and a few other reports as well, which is like going to be a personalized treatment for that, and the success rate of that particular treatment on the specific disease is very high. Yearly, we have the approach of like one size fits for all. Doesn't work in most of the cases, at least as for now. So there we need like genetic information, whole genome information, and clinical information as well, to so that like we can see how the disease has been developed, and then how the course of the disease has been going on as for now. And then like we also get to know that what specific kind of drug is responsive for the treatment of that particular patients. Okay, so that's where like one medicine will not be fitting for all, because sometimes like what happened, it has a good effect in some cases, but there are like some cases there is no side effect, and some are like drastic adverse effect will be there, which is really going to be bad for the patients who's already suffering from cancer. So we started working on like having a personalized therapy for each patients, and where we take the blood and extract the DNA or any other tissue biopsy, or even sometime other biospecimen that we can able to use it. So like we can increase the effect of like drug that we are prescribing, that we like personalized drug or precision medicine drug for that. Okay, so there's like it's sometimes, as we know, it's not about only the drug, even the dosage standard, and then lower, and then higher dosage is depending on the condition, the clinical conditions of the patients, that we have to after we identify the whole what specific drug will be working on that specific cancer, and also we can able to change the dosage as well. So that's how we can able to do the whole molecular profiling of the whole cohort of the patients. Like for example, we were working on like 2-3 different country population. So even we have I can see there are so many other people are there. It could be good to get started working on such kind of like country specific cohort to list out the whole genetic landscape, cancer specific, that could be of good to target. So there are some companies out there who is already in the market, who is doing sequencing and other things. But the thing is as recently, like I see some people are doing it like in a very, very less cost, which can be of helpful for the target therapy for the cancer patient. Okay, so that's where we do the genomics, like clinical genomics, and then pharmacogenomics, other things. And when we talk about a genomics part, we take the genome information and then transcriptomics, RNA, and proteome, where we use mass spectrometry based proteomics, and then a phosphoproteomics, because we take functional proteome of that, and then phenome, how the cell expression happens in that case. And the combination of genome, transcriptome, and proteome, we call this proteogenome. And then we also call functional gene product of that one, whatever we got from the genetic material. So there's a whole genomics, LCMS based proteomics, and transcriptomics that we already got, multi-omics, and then proteogenomics that we do. When we were doing it as a genomic landscape and genomic after the Human Genome Project, so there's a huge research work has already been going on the clinical genetics part. But that is not the primary thing that where we can able to target. And we were thinking we need to have a clear picture of the whole molecular biological aspect of that, like genomic, transcriptomics, and proteomics. And in the case of proteomics, we it's still growing in the field of clinical proteomics, in the particularly in the field of mass spectrometry based proteomics. So we thought what exactly that we need to be working on. So this is the genetic part where we do the mutation, and here the proteomics part where we take the protein and do the digestion, and then we do HPLC analysis and mass spec, and we get identification of the quantification of those proteins. So but when we do this protein is very highly complex, and then each from each segment of the gene and genome, we could able to make 100,000 of transcriptome, and then more, even it translate, that has kind of a different isoform, which makes more than 100,000 and maybe 10,000 of proteome, and then from the different species as well. If each protein is added with the PTMs for that phosphorylation, glycosylation, and other things, that again the diversification of that one is very high. But the limitation of like, you know, in the field of like mass spec, it's very less. So here you can able to see, when if you take the whole protein tissue sample, for example, there is a major portion which is shows in the gray, so which is not detected in the case of like, you know, proteomics. If it's general proteomics that we can able to get the sample from cell line is really fine. When the people like, you know, as like where we work on clinical proteomic data, so the patient samples that we have got, it is like very precious. We may not able to lose many results there. So the detection of the protein from that particular sample is very less. Then we started working on like, you know, I've developed an application for that, where we call this one as the MPAMR. So where if there is a, like if there is unassigned charge states are there in the mass spec, and then like monocytic mass information are not there, or like, you know, correlated peptides are there in the mass spec, that we take it into account, and then like, you know, we tend to map it. It's already reported that like, you know, more than 50% of the MSMS scan doesn't match at all to any other proteins. So that is like kind of huge that we get to lose it. So that's where we develop like, you know, taking each isotopic mass and then like, you know, molecular mass MOC values, and assign the charge state of 1 to 7, and then like we differentiate it to make the UMC cluster for the each things. So UMC cluster is nothing but like, you know, unique mass cluster, where each mass of this particular protein or peptide, and that's elute to the LC at a specific time of that. Okay, so by doing that one, like, you know, we could able to achieve, I'll just gonna be make it little quickly on this. Yeah, so when we see this, like, you know, the conventional method, earlier there was a loss of much peptides, which we could able to draw from there. Then we have developed a technological part, like where MPMR has helped in identification of each peaks very accurately when compared to the conventional method. So it could able to identify the peptides, which is like, you know, really important in the field of like, you know, oncology, and then to the patients to identify the biomarker for the particular patients. So this is where like, you know, we could able to do the power of MPMR identification of the peptides. And then here we talk about like, you know, how this helps when we were using the clinical samples. So we could identify like, you know, almost like 400 like germline mutations and 2 somatic mutations and 2 novel genes which we couldn't able to identify earlier from the same clinical patient samples. And here we can able to see there is a mutation as well, which is the cause like, you know, from variant V to I. So this was identified only with this method, which we cannot able to identify anywhere in the normal method earlier. So this is one of the method that we used it. And again, like, you know, by doing the ELR method, we could identify a little more number of proteins. And then like, you know, still there was like a few, almost like more on like 30 to 40% was left out. Then we started working on how this PTMs affects in identification of the proteins and then disease proteins or the mutation one. So where we like, you know, isolate those spectra and then like, you know, filtered using MSGF, and then whatever the uninterpreted spectra was there, then we use like mod I to identify this, like if that specific peptide is there, what in the case of like PTMs was there, that also included. So in our samples, like in our methodology here, we have used like normal tissue and then like tumor tissue samples. One, we take it for the next generation sequencing analysis for genomics and transcriptomics, and the same sample we use it for like analysis of peptides using mass spec data, and then we convert those genomic data to like, you know, proteomic database, and then after getting the spectra, we just like, you know, map not only the peptides, we do the proteins, we do the SNPs, the indel junction, fusion, other things as well. By doing this, what happened was that like we could able to achieve almost like earlier there was only 14% of the whole protein that we can able to get from the patient samples that was able to achieve. And this was the first of his study where like, you know, around like 63% that we could able to achieve by identification from the clinical samples. So here we can able to see, like, the number of mutation and mutation sites in the specific peptides, which also are targetable sites or specific unique biomarkers to this specific population that we have studied. So where the increase are like almost 76% identification of those particular clinically relevant mutations in genomic level, in transcriptomic level, and also in the proteomic level. So in all the three levels that we have identified using, like, this multi-omics data. So earlier method was like 14%, we could able to, like, 86% was we could able to identify. Now it's like, more than 51% was able to identify in earlier method, 49, and then, like, now we could able to achieve like almost 63% from the, if you take one specific tissue samples, that we could able to get almost all the different kind of, like, cancer specific markers from there. By doing this, one example was there where one specific mutation was there where we considered on like proteogenomics analysis, where in a gene called like TOR1A, where one specific mutation was like, identified, and also it was like phosphorylated. Like mutation and then phosphorylation both are like, present there in that particular sites, which was useful for our diagnosis method. Here we identified, like, even junction, not only the mutation in the case of like genomics and then proteomics. We also saw that, like, how junction formation was there between maybe in the gene level or something. And here we can able to see we have combined all the three different level of the data. So here we use the, like, proteomics data from here, where the genome was used in this. Okay, so the raw genome file and the normal patient file we have used, and where in the conventional method this was never able to identify earlier. But when we use our method, that no, there was a mutation could able to identify that this particular patient has this specific mutations here. Where in the case of, like, the other search that we did on the proteomics, so we identify the, like, there was a PTM as well together on that, so that we could able to identify. It was really helpful to identify the biomarker on that, and that we got published there in the journal called like Cancer Cell, and where we took almost, like, more than 200 patient samples, okay, with normal and tumor from the early like onset of the gastric cancer samples in this particular case, where we took the exome DNA data, and then where we took the RNA data, and we did like, proteomics using mass spectrometry based proteomics. We got the information of the protein abundance, like, identification and intensity of that, and also we got, like, phosphorylation glycosylation information as well. So by doing so, we could able to identify mutation specific phosphorylation correlation, which possibility of, like, signaling interplay in early onset of gastric cancer. And we also got, like, the information recording like mRNA and protein correlation on that, based on, like, that leads to a good patient survival rate as well. So this was one of the first of the study that where we did multi-omics analysis with mRNA and protein, and we identified 4 different subtypes based on that particular multi-omics data. Okay, so this was the team who was working with me in Korea, and we have other teams that we're working in India as well now. I'll be happy if you have any other questions.
Thank you very much for this excellent presentation, Dr. Madar, and for keeping within the allocated time. So now we are waiting for the question from our audience. I still have 2. Yeah, Dr. Lorraine, please, your question, please. Dr. Lorraine, any question?
I didn't raise hand. However, I could ask
yeah,
a
because
question.
I saw your hand raised.
No.
So okay, no worries.
Also, I still have my wonderings concerning the molecular candidates or genetic candidates that you have identified using your techniques.
Yep.
Could you please mention some of these candidates?
So in our case, like we already have there are few methods that we recently are working on it. So where we develop BRCA panel, for example, in the case of breast cancer. So with that one, we add a kind of immunotherapy based panels as well, like HER2 positive, ERPR and other things as well, so that we can able to get to know the therapeutic response in that case. So that's what we do. So if you are interested in kind of gastric cancer specific, then the paper is already out, so I can share you that one.
Okay, thank you. Dr. Muhammad Taha. Dr. Muhammad, are you asking any question?
Yeah. Thank you, Dr. Madar, for the interesting talk. My question that what you presented, the role of multi-omics approach in determination or identification of some mutations for some genes that may play a role in development of cancer. So do you think that by applying this approach, we can detect the proteins or enzymes which are involved in the chemotherapy resistance for some cancers, because as you know that many cancer cells develop chemo resistant during the progression of cancer?
Okay, yeah, that's a good question actually. So I think there are two parts of the query that you have asked. One is the method of which we are using it to identify the biomarker, for example, and other one is therapeutic aspect of that, and then how the resistant plays. So that was your two questions for that. First is on the genomic part and the with methodology that we are using it. So the best way is to develop a specific panel for the specific disease. Initially what happened, people were having a common panels that could be of useful for the people who are suffering from that specific disease anywhere in the world. But later on, the heterogeneity on this patient specific was diversified too much, where one specific panel could be of not suitable. Like for example, there is a company in US, there are 2-3 which is FDA approved drugs was there. If FDA approved test was launched, and they started sending to different countries, and earlier people were using it, but slowly the detection rate for that specific test on that specific patient was lagging. Then that's why people started to focus on that country specific panel that could be of really helpful. That was the first answer for your question. Second was that on the chemo resistant, that is something the hurdle which we keep facing on a different level. One is not only on the resistant level alone, so we also will be facing on how that response of it. Sometimes what happened when we focus too much on the responsive one, it becomes kind of a huge side effect as well in the case of patients results. So we may not be able to focus only on the result part. Sometimes the side effect could be of very high, so we may need to tackle those two things on a single time. So that is one thing which is important. Yeah.
Okay, thank you. I believe that precision medicine and personalized therapy will become the most effective and reliable therapeutic approach for management of many chronic diseases. However, several scientific, clinical, and implementation challenges must still addressed before this vision or this approach become fully realized. So I have two questions for Dr. Madar. What do you see the biggest challenge that preventing personalized cancer therapy from becoming standard practice for all cancer patients? And the second question, how do you balance genomic information with the clinical factors when selecting the most appropriate personalized treatment for cancer patients?
Okay. So yeah, I'll ask you. Okay. Can you repeat my first question, please?
Yeah, my first question is what do you see the biggest challenge that preventing personalized medicine?
Yeah. So in the case of like the first one, it's really one of the best way that we can able to handle and treat cancer patients or any other like kind of chronic disease. But the main challenge is like kind of the cost effective. So it's really going to be very expensive. So that's why like when I started my work a decade back, so as I showed you the slides as well, like when I started my work it was like almost $4,000 to $5,000. So we could able to make that. But recently what happened, like almost 3 years before, it was like $1,000. But now what happened, I mean, we are trying with a few other companies to develop a panel which could be of very short and narrow down, so that like we can able to pull down to like almost 300 to 400 or even less than this. I'll be really happy to make like to reach my goal of like $100 for the whole yeah, because that could be of like anybody can be afford. I mean, everyone wanted to have a best treatment in the world, and but only the cost should not be the barrier. So when I started my work, I wanted one of my aim was like to make it extremely much affordable for that. I think we're gonna achieve in some time soon. Yeah.
Inshallah. So the second question about the balance between genomic information and the clinical factors when decide or when you select the most appropriate personalized treatment for a cancer patient. How do you balance between the genomic information and the clinical factor? Because practitioners, physicians usually depend on clinical factors. Researchers and scientists usually depend on genomic information. How you find the balance between the two wings?
So actually, like instead of telling a wing, because the thing is, the huge problem and hurdle we face, the people who work on clinical oncology or something, the physician or surgeons have no idea what is going on in the genomic part. And the people who works on genomics, like or transcriptomics or personalized medicine on the research part, have no idea what actually, like in reality, what's happening in the clinical settings. So that's kind of like it is always recommended if you're working with on clinical samples or something, to have one of the primary collaborators as a clinical physician, so that
this is a good point. Yes, this is a good point.
Continuously until the work is done, because it's always like fit together, because like each will be having one specific drawback, and they keep discussing and then fulfilling the each things. So that the one best thing was that like I have one of my very good friend. He is a head and neck cancer surgeon. So that's what I was telling. He told me like I'm in agency for about like 18 years, and he has a success rate of more than 90%. Like all the patients are recovering and going home very happily. But
great.
But the worst part was that he see the same patient in the lung cancer department after 3 years, almost 80%. And then in that 50% mortality happens. Then he feels that like oh, it's me who is doing something missing at the first phase, so that's why these people are going to second phase. So then he told me, hey, doctor, can you able to help me up? Because I think like this is something which I keep seeing almost every year, and I feel guilty that there's something is wrong from my side. Then we both sat and started developing a small panel which could be of combination of the genetic information from the head and neck cancer and also lung cancer, and then combine and diagnose it at the first stage. So now whenever the patient goes to him, so he do the diagnosis for the head and neck and also lung, and he give the treatment in the first phase itself, so that the patient mortality should not be there after 2-3 years. So that this kind of study could be of like much more good, and which is like practically application oriented.
Thank you so much. Thank you very much for this excellent presentation, and also for keeping your presentation within the allocated time. We still have 5 minutes for your presentation and discussion. So if anyone of the audience want to ask a question, we can receive one question, more one question. Dr. Muhammad, anyone? Okay, thank you so much, Dr. Madar.
Thank you so much.
We really enjoyed your presentation and this new approach in management of chronic diseases and cancer. Thank you so much. Now I will thank you so much. Now I will switch the mic to my colleague Dr. Muhammad Taha to start the second presentation. Thank you.
Thank you. Have a nice day.
Many thanks, Professor Mahmoud. And again, many thanks, Professor Madar. It's very, very interesting work and topic. Thank you so much.
Thank you so much, Dad. Thank you for having us here. Thank you.
So it's my pleasure to join Professor Mahmoud for moderation this session. And now we are going to the next speaker, Dr. Mira Zomich. Dr. Mira is a postdoctoral researcher at Cancer Research Center of Marseille in France. She's going to share us her work entitled Integrative Single-Cell Analysis of Familial and Sporadic Chronic Lymphocytic Leukemia. So, Dr. Amira, the floor is yours.
Thank you very much. I will just try to connect my presentation.
Okay.
Just tell me if you're able to detect the presentation. Is it visible?
Yes, now we can see.
Okay. So I will try to put it in the full screen. Everything is okay. Okay. So
yeah, everything is okay.
Perfect. Good day, everyone. My name is Mira Kuzmic, and as Dr. Mohamed Taha just said, I'm a postdoctoral researcher at the Cancerology Research Center of Marseille in the team of Dr. Michel Oron-Guillaume. While this work that I will be presenting was performed during my previous postdoc at the Center of Immunology in Marseille in the team of Dr. Pierre Micpied. First of all, just to tell you, I'm honored and privileged to be here today. So thank you for accepting our work entitled Integrative Single-Cell Transcriptomic Analysis of Familial and Sporadic Forms of Chronic Lymphocytic Leukemia. Chronic lymphocytic leukemia is the prevalent leukemia, adult leukemia in Western countries, which involves the clonal expansions of mature B cells that gives rise to monoclonal B cell lymphocytosis, which is a precursor state of the CLL that shares the CLL immunophenotype. CLL subtypes are defined by the somatic hypermutation of the cells, which can be immunoglobulin heavy variable chain mutated, which gives rise to mutated CLL, or unmutated CLL, which is the pivotal prognostic factor. There are additional prognostic markers that include somatic mutations, cytogenetic aberrations such as deletions of 11q, 13q, 17p, and the trisomy 12. And also we have molecular markers like ZAP70, CD38, and LPL. In sporadic CLL, genetic, immunogenetic, transcriptomic changes in malignant B cells and their microenvironment, they drive initiation and progression, which give also rise to malignant B cell heterogeneity, which is on the intra and inter-patient level, which is exemplified here by the circulating cells between the proliferative center of CD5 highly positive cells, which are called proliferative, and the resting states, which are CXCR4 highly positive cells via CXCR4 CXCL12 gradient. Now, CLL is comprised of 90% of sporadic CLL cases, or SCLL that we will call later, and 10% of familial cases. I will present you here by the pedigree of one family that we had in our cohort. Familial CLL means that there are at least 2 CLL first-degree relatives affected by CLL, as opposed to sporadic CLL that has only 1. And also it was found that CLL has the highest clustering among all leukemia among first-degree relatives that carry more than 8-fold elevated risk of hereditary predisposition to CLL. So our principal hypothesis was that the familial germline that will be similar between the patients from the same family will give rise to the similar malignant genome and also to similar malignant transcriptome. So the aim was to characterize familial CLA through single-cell analysis with the hypothesis that the shared genetics in affected siblings will impact similar molecular and functional characteristics of malignant and non-malignant cells. Now, our cohort was composed of 38 patients, composed of 6 CLL families from French National Registry of Familial Lymphoid Hemopathies and 14 sporadic cases where we analyzed their circulating cells, and we have performed integrative single-cell transcriptomic analysis and also investigating BCR and TCR adaptive immune response that was subjected to 10x genomics. Of course, this was also done in the significant collaboration with the bioinformaticians and with the clinicians. After the quality control, we have obtained approximately 126,000 of high-quality cells. Those cells are divided into 3 principal cell continents. It's a B cell continent presented in the malignant compartment in pink, non-malignant compartment in blue, myeloid continent in green, and T and NK continent depicted in violet. We didn't find much of the significant difference between leukemic microenvironments between familial sporadic CLL cases and also with the healthy patients when we compare them. But we found the most of the differences on the malignant B cells, so this will be our focus. When we studied malignant cells, for this time we used the merged data set, not to mask any cellular characteristics by the integration that we had here. Malignancy was assigned based on the patient-specific clustering. This is, for example, the clustering of the patient. Then on the large BCR clonotypes and on the CLL biomarkers such as CD5 and ROR1, which in the end gave assigned malignancy here depicted in pink, and non-malignant cells are depicted in blue. Investigating the malignant B cells, we found something very interesting. This is the UMA-presenting merged malignant B cells, colored by the patient ID. We saw that some patients are transcriptionally very close, which was the first time that was found in my former team. And we found that those patients that are transcriptionally similar are originating from the same family. For example, family 112, the patients 3 and 4, they're transcriptionally very close. The same thing for family 129 and family 196. So we decided to try to confirm this transcriptional proximity using the pseudobulk of malignant B cells from the familial patients using Euclidean distance. And basically we have confirmed that the patients from the same family that have the CLL, that they have significantly higher gene expression similarity as compared to sporadic CLL cases. And this is despite completely distinct BCRs that each of the family members have. The second thing that we ran to was the multiclonal CLL. That is to say that one CLL patient has at least one large CLL clone. In a familial CLL, we found that there is 50% of multiclonal cases as compared to sporadic where there is only 14%. When we investigated deeper those malignant B cell clones presented in this UMAP, I circled one multiclonal CLL familial case that has 4 different clones, and 2 of them are principal depicted in pink and green. As you can see, they carry completely different heavy and light chains. So despite completely distinct BCR clones, those multiclonal CLL are sharing the transcriptional profiles. Again, we evaluated this gene expression similarity using the pseudobulk of malignant BCR clones in the same patient and confirmed that the multiclonal CLL cases are carrying significantly higher gene expression similarity compared to monoclonal CLL cases. Now, one question that I was having all the time is, since we were investigating familial cases, I was wondering if there was some patients that didn't develop CLL, but that they have the pre-CLL state that is not detected yet. So for this reason, in the panel A, we can see the pedigree of the family 22. The patients that I analyzed was the grandmother that had a CLL, her daughter had an AML, a healthy sibling, and the granddaughter that had an AML. This is the UMAP of this family, divided in CLL patient and AML, ALL, and healthy patient. We were investigating the malignant cells in these cases. Here we see the CLL patient that is mostly composed of malignant cells depicted in pink, and some of them are non-malignant depicted in blue. On the other hand, the AML patient had mostly non-malignant cells depicted in blue and some malignant cells depicted in red that were showing the small clonal expansion in the malignant transcription space. Now I was wondering if those cells are carrying the leukemic signature from the CLL patients. So for this, we have compared the signature between malignant cells, non-malignant cells, and the ones that I call the pre-CLL cells. And we have seen that those pre-CLL cells, they indeed carry the malignant signature, and they are significantly different from non-malignant cells, which is basically showing that inherited factors are enabling early leukemogenesis. The last thing that we were interested in was the leukemic heterogeneity, but more precisely, we were interested in the conserved heterogeneity across different patients. So for this, we investigated something that we call the archetypes, using two compatible methods in order to find archetype which is conserved intra-patient heterogeneity. Those are gene expression programs that are similarly expressed in subset of malignant B cells from several patients and samples. We found that we have 7 different conserved functional archetypes across familial and sporadic CLL. Those states that are described here with the heat map marker genes are describing the proliferative state, and the resting ones that were already published. But also we have some activated fractions that are more deeply presented here than published before. Moreover, we have observed that each patient, exemplified in the panel A, UMAP of the familial monoclonal CLL and sporadic monoclonal CLL, carries all 7 archetypes in different ratios, as presented here in all familial and sporadic cases and pre-CLL cases. And we didn't find any difference between familial and sporadic CLL cases or mutated and unmutated CLL cases. This was presented to Philo and obtained for the clinical trial, which is now funded by AstraZeneca, on minimal residual disease in chronic lymphocytic leukemia. So basically, what we have found here is that this is the first single cell analysis of the familial CLL. And we have found that the familial germline has the highest oncogenic impact on leukemogenesis. Then there is the patient's germline, which can also impact the familial and sporadic CLL and the cell of origin. The malignant genome is going to be similar from the familial CLL cases, which, as we suppose, will give the similar malignant B cell transcriptome, which will basically create this transcriptional convergence in family-specific profiles. Then we found that some patients are carrying multiclonal CLL, and some of them are carrying monoclonal CLL. Multiclonal nature that we found that is more prevalent in familial CLL that showed shared transcriptional programs is indicating that there is a common progenitor which is pre-VDJ recombination, which can be detected on hematopoietic stem cell level or common lymphoid progenitor. And in the end, we found that all of those sources of malignant heterogeneity are converging to conserved malignant functional states. So basically, we found that familial and sporadic CLL cases are sharing the core features. They are giving 7 conserved archetypes, which reveal similar diseases with a common life cycle that identifies shared malignant sources and therapeutic targets, with the notion that the familial CLL is a one step closer to leukemogenesis due to the hereditary imprint. With this, I would like to thank to my former lab and to all colleagues that I had to our collaborators, clinicians, scientists, and bioinformaticians, and the patients and the families that we had, and of course to all the fans that we have. And I would like to thank you for your attention, and I would like to open the questions.
Many thanks, Dr. Mira, for this very interesting talk. Many thanks. So now we are going for the questions. Please, if you have a question, you can raise your hand. So I can start. So I'm wondering if you checked the tumor microenvironment between the two populations. So did you find any difference?
Very good question. So yes, we found some differences, but those differences were not as significant as we found for the malignant B cells. So for example, we found the differences in T and NK cells. Some of the compartments of T and NK cells were more present than the others. Also, we found on the B cells that we have different proportions of the naive, intermediate, and memory cells in CLL patients compared to healthy patients. But this remains very discrete. And just one thing to notice here, we were investigated PBMC. In PBMC, we cannot expect to see major differences in leukemic microenvironment as we could have observed in the lymph node. So this was already something that I was expected when I was starting this research, but still we have observed some differences.
And the second point, because you mentioned that 10% is familial and 90% sporadic.
Exactly.
So did you check the signaling pathway in each population, which is the major pathway?
Honestly, we didn't check it. It's a very good point that you make, but I think that we would need to push the analysis further in order to do this signaling.
Thank you. Any question, please? Okay, thank you, Dr. Mira. Nice to meet you. Thank you.
Thank you very much, and good luck for all the presentation.
Thank you so much.
Thank you. Bye.
Now we are going to the Dr. Professor Mahmoud.
Okay. Okay, thank you, Dr. Muhammad. Our next speaker is Dr. Laurine Haboub from the Cancer Research Center in Marseille, France. Dr. Laurine Haboub presentation is about cell cycle regulation and is entitled ZBTB16 mutation promotes a myelofibrosis-like stem cell disease in a cyclin-dependent kinase inhibitor 2A null context. Please join me and give a warm welcome to Dr. Laurine Haboub, please. Thank you.
Thank you, Dr. Mahmoud.
Thank you.
So I'm going to present, to share my slides. Is it shared? Because
Not
I
yet.
can't see my slides. Yeah. I will try again.
Okay, no worries.
Enter screen. Social.
Not yet.
No.
Yes,
strange
now.
because I have
No.
it's strange because I have checked
Back.
up stop share you are okay. I'll try again.
Okay.
Okay. Microsoft Edge. Okay. I apologize for this technical issue. I will switch to another computer. Can you switch to the next presentation in order to switch to another computer? Thank you.
Dr. Muhammad, can you call the next presenter, please? Dr. Ahmed Bashir.
Okay. Thank you, Dr. Mahmoud. So now we are going for the presenter, Dr. Ahmed Bashir. Dr. Ahmed Bashir is also a postdoctoral researcher at the Cancer Research Center of Marseille in France. His talk will be about CRL2FIM1B uses the heme in recruit batch for degradation and regulate ferroptosis in lung cancer. So, Dr. Ahmed, the floor is yours.
Thank you so very much. First of all, I would really like to thank and congratulate all the team for conducting such a great scientific environment. I would like to share the screen. Do you hear me?
Yes, Dr. Ahmed,
yes.
We can hear.
Yes.
Can you please share your slides? Okay.
Now you see my screen?
Not yet.
It's a black screen. Now, yes.
Okay. Okay. Is it okay now?
Yeah, it's okay. Yeah, it's okay. Perfect.
Okay. Just let me move this screen from here. Okay, so today I'm going to present our latest work that we published in the last 2 weeks. So we are a team that work on protein degradation. So we work on heme ubiquitin and proteasome system in lung cancer. So we identify a CRL2 ligase called FEM1B that utilizes free heme to recruit BAC1 for degradation and in turn regulate ferroptosis in lung cancer. So as you know that lung cancer has the highest incidence rate reported by WHO in 2022, accounting for 2.5 million new cases, making it the top incidence rate among all the other cancers. In similar fashion, it also has the highest mortality, accounting for 1.8 million deaths reported in 2022. So there is a need to find better treatment ways to improve the overall patient outcomes. The conventional therapies that are used, like for all other cancers, are similar chemo, radio, targeted, and immunotherapies. But like all other cancer, lung cancer also develop resistance to treatment over time. So we need to find ways or mechanism that can overcome this resistance. One such mechanism that is proposed is ferroptosis, which can be used as an adjunctive therapy among with other therapies to overcome the treatment resistance. So what is ferroptosis? Ferroptosis is an iron-regulated form of cell death characterized by the lipid peroxidation of the plasma membrane. It occurs when there is an increased labile iron pool in the cell, which increases the overall ROS in the cell, which convert the polyunsaturated fatty acids into lipid peroxides. This lipid peroxide under normal condition is converted by GPX4, which is glutathione peroxidase 4, into lipid alcohol, which is non-toxic, by utilizing glutathione, which is controlled by SLC7A11. If it's not converted into lipid alcohol and this lipid peroxide start a chain of reaction in the plasma membrane, and plasma membrane loses its integrity and the cell dies, and this mechanism is called ferroptosis. So this mechanism can be exploited in terms of with other therapies. It is proposed to be used as an adjunctive therapies to overcome the resistance. Recently, it has been shown that ubiquitin proteasome systems regulate ferroptosis. As you know, ubiquitin proteasome system ubiquitinates the protein, and then after ubiquitination, it gives it to proteasome for degradation. This ubiquitin proteasome or ubiquitination process is taking place with three enzymes, E1, E2, and E3. E1 is the ubiquitin activating enzyme, and E2 is the ubiquitin conjugating enzyme, and E3 is the ubiquitin ligase that ubiquitinates the substrate and give it to proteasome for degradation. And the focus of my project is this E3 ubiquitin ligases. To save time, I'm just going into, there are two forms of E3 ligases, monomeric and multi-subunit. So I will just go to the multi-subunit, which are called culin-rin ubiquitin ligases, also called CRL. These are multi-subunit enzyme. It has a scaffold called culin, and on one hand, it holds the substrate receptor on which the receptor substrate protein is bind. On another end, it holds the E2 ligase that has ubiquitin, and this ubiquitinates the substrate and give it to proteasome for degradation. In human, they are classified into 5 major families from CUL1 to CUL5, and these differ only on the basis of the scaffold protein from CUL1 to CUL5, and they also have a different substrate receptors. So the focus of our work is mostly on the CUL2, which is CRL2 complex. So we are interested in finding CRL2s and their substrate which regulate ferroptosis. I find out CUL2 CRL2 ligase called FEM1B that utilizes free heme to degrade BAC1, which in turn regulate ferroptosis. So just going into detail for my result, just to introduce, BAC1 is a transcriptional regulator, which is the master regulator of the redox, and it also regulates ferroptosis, and it's also involved in lung cancer metastasis, and it also regulates pathways that regulate the production of the free heme. So it controls the amount of heme through HO1. So to save time, I just said it, and then we can go further. So the whole story started when we did an IP mass spec of BAC1 with cell treated and non-treated with heme. And we look for the ligases, and we find out that FEM1B is our top hit, and this binding increase upon heme stimulation. To confirm our mass spec data, we did another IP flag FEM1B, and we treated our cell with plus minus heme, and we saw that in non-treated cell, FEM1B bind to BAC1, but when we put heme, the binding between FEM1B and BAC1 increased further, confirming our mass spec data. Then we wanted to further check if it's only binding BAC1 or it's also controlling its outcome, its degradation as well. So what we did generated CRISPR knockout FEM1B cell line, and we treated with heme at different time point. And in our control cells, as you can see, that BAC1 is degraded upon time in both the controls. But when we knock down FEM1B, when we CRISPR knock out FEM1B, we see that BAC1 is stabilized, suggesting that BAC1 does not only bind with FEM1B upon heme stimulation, but it also controls its degradation. Then we wanted to know, how does this heme promote the degradation of BAC1 by FEM1B? Has BAC1 in BAC1 structure, there are 6 cysteine proline motifs, and it has been known that heme bind to the cysteine proline motif of all the heme regulatory proteins. In that case, it's BAC1. So we wanted to find the binding region of where exactly BAC1 binds to FEM1B upon heme stimulation. So we generated a lot of mutants of BAC1 just to save time. I would just go straight to the mutation that worked. So when we did mutated the really C terminus of BAC1, we lose binding with FEM1B. And when we further go, it was like a stretch of 25 amino acid, and then we further did alanine scanning of 5 amino acid, and we find out that this last 5 amino acid from 645 to 649, when we mutate this 5 amino acid, BAC1 lose binding to FEM1B. Interestingly, this last 5 amino acid also contained the last cysteine proline motif of BAC1. So what we did, we mutated this last cysteine proline motif, and we treated our cell with heme. So in wild type BAC1, we did an IP BAC1 plus minus heme as usual. The wild type BAC1 as usual bind, and the binding increased upon heme stimulation. But in this cysteine proline motif, when we mutate this last two cysteine proline motifs, we also mutate just only one cysteine. And then we did the cysteine and proline together. In both, they lose binding with BAC1. It suggested that heme binding to this last cysteine proline motif control binding of BAC1 with FEM1B. Then we wanted to check the heme binding to this last cysteine proline motif also control its degradation, because if it's not binding to BAC1, it should also not be degraded. So we did the same assay. We treated our control cell with heme at different time points, and BAC1 is degraded. And when we put the inhibitor of the calin, which is called MLN, we saw a rescue of BAC1. And when we treat also the cell that has a mutant, CP mutant, and we see that BAC1 is stabilized upon heme stimulation, suggesting that heme binding to this last cysteine proline motif control its binding, and it also control its degradation. Then we wanted to check that if heme is part of this complex, or heme is just binding to somewhere, exposing the binding motif of BAC1 to FEM1B. So what we did, we map this last 70 amino acid stretch containing the last CP. We purified this last CP with 70 amino acid, and we find out that BAC1 binds with FEM1B because of less time. I'm not showing all the experiment that we did, but this is the stretch of 70 amino acid. BAC1 is a 736 amino acid protein. So this 70 amino acid containing the last CP, when we purify it, it binds to FEM1B. So we take this small segment of 70 amino acid of BAC1, and we tag it with his, and we also tag FEM1B with GST. Now we wanted to check if the binding between BAC1 and FEM1B, all the experiment that we did before was in the cell. So was this binding direct, or there was some other triggering thing that were triggering this binding? So we purified FEM1B, and we've purified this small degran of BAC1 containing the last CP, and we also purified the mutant of BAC1, and we saw that BAC1 degran bind with FEM1B only when we put heme. So it shows that the binding between BAC1 and FEM1B is direct. And when we see with the mutant, it does not bind as usual. So the heme binding to this last cysteine proline motif is important, and this binding between BAC1 and FEM1B is direct. It's not for, there are no other regulatory, because it's an in vitro binding assay. We also did BLI, we also did native mass spec, and we use nanobread, but because of the time, I'm not showing all of these. So we confirm that this binding between BAC1 and FEM1B is direct, and it's regulated through this cysteine proline motif of BAC1 that's binding to heme. Then we wanted to check if this region is really the binding region. Can it also, so the question was, if you tag this region to another protein, will it also bind with it if it's the binding region? So what we did, we tagged this degran with GFP, and we did the same IP GFP, and we saw that BAC1 bind with the GFP degran bind upon heme stimulation with FEM1B, and the CP mutant does not bind as expected. It makes us even more enthusiastic that we should see if it does not only control the binding of other protein, can it also trigger the degradation of the other protein as well? So we did the same as we did before. We treated our cell with heme, and we saw that GFP degradant was degraded upon heme stimulation, and the cysteine proline motif that was not binding to FEM1B does not degrade, and it was stabilized. So it gave us an idea that if you tag it with any protein, it can induce degradation of any other protein that you tag with this degradant. After confirming this old binding mechanism, we want to check that what's its role, its functional role. So we use multi-omics, we did transcriptomics, and we did proteomics mass spec, and we combined the data, and we find out the pathway that was highly regulated. It was ferroptosis, as was our prediction. So we used ferroptosis inducer drugs such as Erastin, which block this SLC7E11, which in turn control the glutathione production, which is fuel for GPX4 to inhibit lipid peroxidation. And we treated our cells in control cells, we saw, but when you remove FEM1B, these cells were sensitized to ferroptosis, confirming our hypothesis that BAC1 is regulating ferroptosis, and this regulation is taking place through FEM1B. Then after confirming in vitro, we go in vivo models. So we injected the control cell line, and we also injected the knockdown FEM1B cell line. And after 10 days of inoculation, we started treatment with two ferroptosis-inducing drugs. So these are two individual mouse experiments we did with sulfasalazine, and we also did with IKE. These are also the drugs that have the same mechanism of action. And we saw that the total metastatic burden of the mice, and we see in control there is no difference, but when you knock down FEM1B, the cells, if you see from control to the cell that are treated with sulfasalazine or IKE, there is less metastasis. So confirming our hypothesis that these cells are more vulnerable to ferroptosis, and we see a less metastasis in the knockdown FEM1B cells. And then we check also the total number of metastases in the lungs, and we saw similar result in control. It's non-significant, but with knockdown FEM1B cells, there is a decrease in metastases in lung. These are the visuals of the bioluminescence in control. So these are control cells treated with, so these are the lungs of the mice of all the groups. So these are like one group. And you see in control group, non-treated and treated with IKE, there is no difference. Yes.
Can you just, because we are now in 15 minutes.
It's the last slide. It's the last slide. Yeah,
it's
the last slide. I'm going to the model. And then when you knock down FEM1B, we see that there is increase, but when we treat with IKE, there is less metastasis. So confirming our hypothesis also in vitro, that we confirm in vitro, it's also confirmed in vivo. And we did it in two independent mouse experiments. So the working model of my project is that in non-small lung cancer, when there's a wild-type FEM1B binds with BAC1 upon heme stimulation, and BAC1 is degraded. There's less BAC1, there's less repression of the genes that are involved in ferroptosis, and there is less ferroptosis. But when you knock down FEM1B, there is the degradation, knockdown FEM1B, there's no degradation of BAC1, so BAC1 is more, and there's more repression of the protective gene that are involved in ferroptosis, and there is more ferroptosis. So we propose that this mechanism can be exploited as an adjunctive therapy to treat the tumor-resistant cancer cell line. And we published this work recently last month in Molecular Cell. So thank you so very much for your time, and I'm happy to take your questions.
Thank you so much, Dr. Bashir, for the interesting talk. Thank you so much. So if there is any question, please. So I have a question.
Yes,
so
ma'am,
Dr.
please.
Bashir, so just naive question, because you mentioned in the first of your talk, in the introduction, that there are three enzymes, E1, E2, and E3. And
yes.
you are focusing on, I think, E2 or E3?
E3.
E3. Why you decided to just to focus on E3, not E2 or E1? And the same question for the Carl 1, 2, 3, 4, 5, you said, and just you focused on Carl 2. Why Carl 2, not 1 or 3 or 5?
Sorry, I didn't get you. Which Carl?
Or now for the E1, E2, E3, you focus.
Yes, this question I get it. The second one.
The second one for, you said there are 5 subgroups of cluster of.
Yeah. CRLs. Yeah.
Yeah, CRL, and you focus on the 2-1.
So the first that E1 and E2 enzyme, that ubiquitin ligase is E1 and E2, they are just involved in, so E1 is involved in the activation of the ubiquitin, and E2 is the conjugating enzyme that transfer this activated ubiquitin to E3. E3 is the main enzyme, which on one hand, like I can show you just here, it would be a bit easy. Yeah, like here. So here, this E3 that we are talking about is this calin that on one hand hold the substrate receptor and it contains the substrate protein. Like here, it should be BAC1 and this should be FEM1B. So this is FEM1B and this is BAC1, and this E2 is ubiquitinating the substrate, and then the substrate is given to proteasome for degradation. And regarding to your second question, that why I choose specifically among all these 5 families CRL2, because there is precedence, this CRLs, there are 20 identified CRLs, and there is one CRL called KHLDC13 that has recently been known to regulate SLC7A11. So we were more interestingly going to this direction, that this CAL2 is involved in the regulation of the ferroptosis. So this is why we did an unbiased screen. So I didn't show this mass spec that we did with CAL2. So we did a mass spec with CAL2, and we searched for all the substrate, and BAC1 was in that search. And then we started the story, but because of the time, it was too short. I didn't show the mass spec that we did.
Thank you, Dr. Bashir. And I have more questions. So because we haven't time, just I will type the question in the chat, please.
Yes, please. Thank you.
Okay, thank you.
You're welcome.
Dr. Mahmoud?
Yeah, thank you, Dr. Muhammad. Now we are back to Dr. Loreen Habboub in her presentation about cell cycle regulation, the presentation entitled ZBTB16 mutation promotes myelofibrosis like stem cell disease in cyclin-dependent kinase inhibitor 2A null context. Please join me in giving a warm welcome to Dr. Loreen Habboub. Please, Dr. Loreen. You can start.
Good afternoon, everybody. So now you are able to see my slides?
Yes.
Yeah,
Thank
okay,
you.
perfect. Okay. So first of all, I would like to thank the scientific committee for selecting my abstract. It's my pleasure, and it's my honor to be a student graduating from the Faculty of Pharmacy at Azhar University. So it's my pleasure and honor to participate in this conference. My work today is titled by ZBTB16 mutation promotes myelofibrosis like stem cell disease in CDKN2A null context. This work has been conducted in the team where I'm working as a postdoctoral researcher at Cancer Research Center of Marseille, the team of integrative molecular biology in hematopoiesis and leukemia. So first of all, since we are talking about myelofibrosis, that is aging-related disease, and it's a disease concerning the hematopoietic system, I will introduce briefly the hematopoietic system. It starts with hematopoietic stem cells on the top of the system, which gives rise to the multipotent progenitors, giving rise to further mature progenitors, including megakaryocyte erythroid progenitor, GMP granulocyte macrophage progenitors, and chemolymphoid progenitors, which give rise to the mature lineages in the peripheral blood. Then here I wouldn't go in details because Professor Madar has mentioned that during aging we have DNA damage and recurrent mutations which affect the hematopoietic stem cells in particular in this case, and we have clonal hematopoietic stem cells which cause ineffective hematopoiesis and development of myeloproliferative neoplasm, including the primary myelofibrosis. So what is primary myelofibrosis? PMF is a clonal hematological malignancy that is aging-related disease, so it occurs mostly in the elderly between 50 and 70 years. It is considered the most severe disease among the myeloproliferative neoplasm because of the shortest survival, and 20% of the patients develop AML progression, I mean acute myeloid leukemia. So the main characteristics of the PMF disease include bone marrow fibrosis and splenomegaly, in addition to the accumulation of myeloid progenitors, which give rise to accumulation of the granulocytes and the megakaryocytes in the bone marrow and in the peripheral blood. So this is caused by a driver mutation. These mutations could include a JAK2 mutation, which accounts for approximately 60% of the patients, calreticulin mutation, and MPL mutation. However, 10% of the patients do not show a specific mutational profile and are not yet defined until now, so we call these patients as triple negative patients. In this study, I was using a double mutant mouse model that is considered as a new mouse model for myelofibrosis. This model includes a double mutation of CDKN2A and ZBTB16. This model has been generated by crossing two single mutant mice. The first is CDKN2A mutant mouse model that normally develops variant kinds of tumors with another single mutant model, ZBTB16, that normally develops aging phenotype without a pathology. However, by crossing these single mutants, we have a specific phenotype related to the double mutant mice. It is characterized by splenomegaly, as we can see in the green color. So this means that we have a switch from the tumor phenotype developed by CDKN2A into splenomegaly in the double mutant mice, and for sure accompanied by a decrease in the survival of these mice compared to the wild type and ZBTB16 mutant mice. And for sure we have a decreased survival in the CDKN2A in the blue because we have a tumor. In the spleen of the double mutant mice, I detected a presence of stem cells and progenitors. The stem cells are presented by LSK population, lineage negative, SK1 positive and CKET positive cells. As you can see in the red color, we have an expansion of the stem cells in the spleen as well as the progenitors, indicating that we have extramedullary hematopoiesis, meaning that we have the hematopoietic process which occurs in other organs other than the bone marrow, in this case in the spleen. In addition, concerning the mature cells, we have a myeloid bias in the spleen in the behalf of the double mutant mice. So we have accumulation of myeloid cells that are defined as CD11B positive cells on the expense of the lymphocytes presented by B220 positive cells. And by following the blood from these mice compared to the wild type, we found that these mice showed a global cytopenia related to red blood cells, platelets, and lymphocytes. And this is also specific for the myelofibrosis disease. By the histopathological examination, we detected the myelofibrosis, I mean the bone marrow fibrosis, specifically in the bone marrow sections from the CDKN2A double mutant mice, and also an accumulation of the megakaryocytes in their bone marrow. These manifestations, these characteristics recapitulate the phenotype of the myelofibrosis that occurs in the myelofibrosis patients. Then we wondered about the molecular mechanism that could explain the development of myelofibrosis in the double mutant model, especially that this could explain the gap that we see in the myelofibrosis patients in the triple negative patients. For this reason, we performed a single-cell RNA sequencing analysis for purified cells, the stem cells and the progenitors from the bone marrow of the 4 genotypes. And then by bioinformatic analysis on the single-cell RNA sequencing experiment, we could define 12 clusters in the whole bone marrow from the 4 genotypes. These clusters we analyze, and by deep analysis through bioinformatic tools, we identified the myelofibrosis initiating cell clusters, including the long-term HSC, hematopoietic stem cells, the multipotent progenitors 2, the megakaryocyte progenitors, and the granulocyte macrophage progenitors. And then we focused on these clusters as the focus point for the further analysis. So I performed differential gene expression analysis on these clusters to compare the gene expression between each mutant mouse model compared to the wild type. And these analyses have shown an important dysregulation concerning a gene called CDK2AP1. This is a cell cycle regulator. It was downregulated in the double mutant in the red, as we can see, and also concomitantly, we have a downregulation induced by the ZBTB16. To validate these results, I performed RT-qPCR and I found the same tendency in the progenitor population, meaning that we have a downregulation in the double mutant induced by the ZBTB16. But what is CDK2AP1? It is a regulator of cell cycle that inhibits the G1 to S phase transition by inhibition of CDK2 and cyclin E complex formation. So this initiated a question: what is the relation between CDK2AP1, CDK2, and ZBTB16? For that, we went to use human cell lines that are knockout for ZBTB16. These cell lines showed a downregulation of CDK2AP1 protein expression, which was accompanied by a high expression of CDK2 and cyclin E1. From these results, we could confirm the relation between the 3 factors of this axis. So ZBTB16, which downregulates CDK2AP1, which inhibits CDK2, is able to ultimately increase CDK2 expression that may initiate the myelofibrosis in our model. Then we wanted to validate the axis dysregulation and specifically concerning the CDK2 in the myelofibrosis patients. So we performed analysis, actually transcriptomic analysis on the patients from the Institute Pauli-Calmet. And here we found very interesting results related to the CDK2 expression that was markedly expressed in the myelofibrosis patients. And it was upregulated compared to the normal donors and also compared to other myeloproliferative neoplasms such as essential thrombocythemia and polycythemia vera, which indicates that CDK2 could be a therapeutic target for the treatment of myelofibrosis patients. So to conclude from this study, we could demonstrate that the double mutant mouse is a novel mouse model of myelofibrosis, and that ZBTB16 mutation contributes to the myelofibrosis development through the dysregulation of CDK2AP1 and CDK2, meaning that we have a cell cycle dysregulation, and also that CDK2 is a molecular candidate that deserves to be a target for a therapeutic regimen of myelofibrosis patients. Finally, I would like to thank you all for your attendance and to thank the team members and all the workers on CRCM in the different platforms. Thank you.
Thank you. Thank you very much, Dr. Loreen, for your excellent presentation and for keeping within the allocated time. We truly appreciate your cooperation. So now we are open to have one or two questions from the audience. So please, if you have any question, please unmute your mic and start your question. I didn't see any question yet. No questions, no questions. So thank you very much, Dr. Loreen. And now we are moving to the next presentation. Our next speaker is Mr. Ismail Mansoor from Mangalore University in India. Mr. Mansoor presentation entitled Theragenistic Targets Identification in Luminal Breast Cancer using Multi-Omics Technology. Please join me in giving a warm welcome to Mr. Mansoor. Please, Mr. Mansoor, open your mic and start sharing your presentation. I didn't see Mr. Ismail. Yes, please. You can mute it. Yeah, okay. I will. Okay.
Assalamu alaikum.
Alaikum assalam wa rahmatullah. Mr. Ismail, can you hear me? Yeah, now it's okay. Please, yeah, make the presentation. Click on the presentation icon. Yes. Yeah, you can start now. Thank you. Please, Mr. Ismail, please unmute your mic. Unmute your mic. Your mic is muted. Please unmute your mic. Yes, now.
Okay.
Yeah,
Assalamu alaikum.
now. We can hear. Alaikum assalam. Start.
Good afternoon, respected professors and colleagues. Thank you, Dr. Mahmoud Sardah and Dr. Muhammad Taha. It's my great honor to present my research at the third International Conference on Pharmaceutical Science titled Pharmacy from Rebel toward Recovery. First, I would like to express my sincere gratitude to the conference organizer for their excellent efforts in organizing this scientific event and for providing me the opportunity to present my work. It's an honor to present my work today on identification of biomarker and the theragonistic target in luminal breast cancer through multi-omics analysis. Luminal breast cancer is the most common subtype of breast cancer. The molecular drives behind its progression are still not fully understood. In this talk, I will share how we used on the integrated multi-omics approach to identify a set of key genes that may serve as both prognostic markers and future therapeutic targets. First, this outline, I will start with background, molecular subtypes, aims and objectives, results, conclusion. Yes. The first objective of my project is identify the key molecular feature of the luminal breast cancer using multi-omics TCGA data. The second one, explore altered pathways and regulatory networks involved in tumor progression. Finally, discover potential biomarkers to improve molecular subtyping and precision prognosis. In 2022, approximately 2.3 million new incident cases of breast cancer globally. By 2050, globally, globally, breast cancer cases are projected to increase by 38%, while mortality also is expected to rise by 68%. Now, the luminal breast cancer, either luminal A and luminal B, represent the most prevalent molecular subtype accounting for approximately 50 to 60% of all breast cancer cases. The research gap is current cancer studies are largely based on single omics data. That means limiting the understanding of the complex molecular networks. Therefore, an integrated multi-omics analysis is needed to identify more reliable biomarkers and therapeutic targets. My methodology starts with the data analysis. After that, using the many tools for protein-protein interaction network construction and hub genes identification. After that, functional enrichment analysis, then expression and survival analysis. Finally, therapeutic target evaluation by molecular docking by using many software like we have already used the Maestro Schrödinger software. Okay. Regarding the result, from approximately 45,000 genes, we identified the 500 highly significant differentially expressed genes, 250 upregulated and 250 downregulated. This heat map shows the upregulated genes in tumor versus normal tissues. And this figure shows the downregulated genes in tumor also versus in normal tissues, in which the red color indicate the higher expression, but the blue color indicate the lower expression. Now, the second one is identification of the hub genes. As I mentioned before, we screened the approximately 45,000 of the human genes by using the string tool for protein-protein interaction and identify the key regulatory proteins. We identified 500 more highly significant differentially expressed genes. After that, using by cytoscape for visualization and topology and analysis, and choose the best of the largest cluster, which has 68 nodes and more than 2,000 edges. After that, using CytoHubba, by using CytoHubba and five algorithms like MCC, degree, betweenness, closeness, and MNC to select the best, the top 10 hub genes, like ORCA A, ORCA B, BUB, 1B, CDKA, CDK1, and so on. After selecting the 10 hub genes, now we want to make some validation of these hub genes, okay? By using many tools like GenMania. GenMania, we used GenMania for study the functional connection of the hub genes between other. Also, we use in EnrichKG, depending on main for databases like gene ontology, KEGG, dysgenate, and kinase library for what? For confirming their involvement in mitotic regulation and DNA replication and oncogenic signaling pathways. Also, for further validation of these 10 hub genes, we used the multi-gene expression analysis by using the TN plot, by using the the comparison demonstrating the differential expression patterns of these hub genes across the normal tissue, tumor, and metastatic tissues. Now we want to go to the multi-omics expression of the hub genes in luminal breast cancer. As this figure show the TCGA genomic data in this expressions of the 10 hub genes from A to G. For example, A. This compare between the gene expression between the tumor cells and the normal cells. The conclusion that there is significantly upregulated in tumor samples compared to the normal breast cancer. Also, we can do the same thing at the proteomic level. Also, we can see that most of these hub genes can make the elevated protein abundance for the same in breast cancer related to the normal control. What does that mean? That means these hub genes as potential biomarkers, okay, highlighting their role in cell cycle regulation and tumor progression and luminal breast cancer. Also, further validation of these hub genes make integration of the transcriptomic and immunohistochemical between these hub genes. Like this figure shows the comparison between the protein expression at the normal tissue and the cancer tissue, and these show increased protein expression was observed in breast cancer tissue compared with the normal tissue for most of genes that have genes. Regarding to the survival analysis, we used two tools for studying the survival analysis. From GBA, can use the Kaplan-Meier survival analysis that demonstrate the high CDK1, CCNB1, also ORCA-B expression was significantly associated with the poor overall survival. Also, by using timer making correlation analysis between the expression of the hub genes and the tumor purity of CD8 T cell infiltration. Our results show that honestly there weak correlation between the immune cell infiltration and these hub genes. What does that mean? Does that mean these hub genes are generally or primarily associated with the tumor cell proliferation rather than contributing to immune cell infiltration? Finally, molecular docking. Docking studies by as a translated GUNA step, we screen GUNA candidate compounds against CDK1 and identify this compound AG02432 as a strong CDK1 inhibitor by using the Maestro Schrödinger software. Firstly, we import this protein from PDB, and this ligand can imported from the drug bank and the PubChem, and by using this all the steps of the molecular docking by using the Maestro can take this result. This also show the two-dimensional interaction between the ligand and receptor. Conclusion, yes, now we achieved my goals that I expressed for the first slide. Identify the 10 hub genes that's signature for the luminal breast cancer. Also not only 10 hub genes, but also we can identify the key hub genes, include the ORCA-B, CDK1, and CCNB1. And we can also more studying for gene overexpression. Yes, gene overexpression is associated with advanced tumor stage, lymph node involvement, and reduced overall survival. The hub genes are mainly involved in mitotic regulation, cell cycle progression, and the best conclusion, multi-gene risk model demonstrated better prognostic performance than individual gene. Okay. This finding support the potential of this gene prognostic assessment, patient stratification, precision medicine application in the luminal breast cancer. Finally, I would to express my sincere gratitude to Al-Azhar University, the Faculty of Pharmacy staff, my friend at Mangalore University and Unipoya University, my beloved family in the Gaza Strip, whose resilience in the face of extremely difficult circumstances. Thank you for all. Thank you for listening.
Thank you for your nice presentation and keeping within the allocated time. I am seeing 4 hands. I will start first come, first serve. First, pharmacist Musab, please.
Yeah,
You need to ask a question, please.
Hello?
Yes, Dr. Musab.
Yeah. Do you hear me well?
Yes, can you?
Thank you, Dr. Ismail, for this nice presentation and insightful. So I have a questions. So why do ORCA1, CDK1, and CCNB stand out specifically among the top 10 hub genes?
Yes, thank you, Musab. Basically, these 3 consistently show the strongest and the most significant association with poor survival across the Kaplan-Meier. When comparing with these 3 components with the others, these 3 components is the most important and the most significant, especially for poor survival. Yes.
Okay, thank you. Dr. Loreen, you can start your question.
Thank you, Dr. Ismail, for your presentation. Actually, I was also interested in your results because you are showing one of the factors or genes that regulate the cell cycle, which is CDK1, that you have shown in this presentation. And also this comes in link with my previous presentation where I highlighted the CDK2 as a molecular target that could be used for the treatment. This is very nice. And also I would like to highlight that the recent knowledge focused on the cell cycle dysregulation in breast cancer and also other solid cancers, especially that CDK2 has been recently highlighted as a molecular target. In this context, I wanted to ask you if you found any results related to CDK2 gene among the genes that have been differentially expressed and you highlighted by your results. This is my first question. My second question, I think you have shown that you did enrichment analysis, and I also wanted to know what is the strongest signatures that you could define by the enrichment or the functional enrichment analysis. Thank you.
Okay. Thank you for your questions. About regarding to the first question, firstly, by using the 10 hub genes, okay, by using the like string and cytoscape tools to select these hub genes. Okay, after the selecting for the hub genes and performing a lot of the analysis and for validation of these hub genes and the correlation between or the relationship between these hub genes and the breast cancer, the best three that CDK and CCNB1 and ORCA B. After screening these three genes by the Orcan database, only these genes for that has druggable. Okay. Also the new one research about the other genes, how can related to the are these genes other genes other than these genes can related to the cancer. This new or future, inshallah, research about this. Regarding the second question, I didn't catch this.
Yeah, a second question about the functional enrichment analysis. If I will catch that, you did that, and so I was curious about the first or main signatures that you could be able to detect by this analysis.
Okay. About the enrichment analysis by using the enrich KG by using the 4 library databases, gene ontology, KIG, and also dysgenate. After this, we get the specially functionals of these genes related to the cancer. Also can maybe this functional enrichment, there are a lot of relationship between these genes to the cancer, breast cancer.
Okay, we may have two quick questions, one from Mr. Ahmed Abu Samak and the other from Dr. Muhammad Taha, and I will switch after that the mic to Dr. Muhammad Taha. So, Mr. Ahmed Abu Samak, please, your question.
Hello, do you hear me?
Yes. Yeah.
Okay. Thank you, Dr. Ismail, for this nice presentation. So actually,
I think you have a poor internet connection. So if you please you could send your question through the message of this meeting, because I couldn't hear you. Yeah.
Hello, do you hear me now?
Mr. Ahmed, yeah. Can you ask your question quickly?
Yeah. Thank you, Dr. Ismail, for your nice presentation. Actually, I have a question based on the what you explained about the docking analysis. I was wondering how confident you are about AG0 24322 as a CDK1 inhibitor candidate. Has it been experimentally validated, or the evidence currently limited to computational predictions?
Okay. Regarding this result, this was computational docking result, not experimental. Okay, this result regarding favorable binding of affinity and stable interaction. That means we want to in future further screening and further for experimental experiments to ensure that this component is the best for docking or not. That means this result will be computational, not experimental.
Okay, thank you. Now the last quick question from Dr. Muhammad Taha, and he will continue the last presentation of this session. Thank you so much.
Thank you, Dr. Mahmoud. And thank you, Dr. Ismail, for the nice presentation. So you identified by multi-omics protocol, you identified large number of candidates of the genes. So I'm wondering, did you cluster these genes according the pathways which may be involved in the pathogenesis of the luminal breast cancer?
Yes, the first thing we did the screening of the genes that can get it from the AllCan database. After that, getting the hub genes. After getting the hub genes, we want to screening the pathways and how can relate these hub genes for the breast cancer.
Okay. Thank you, Dr. Ismail.
Thank you.
Okay, now we are going for the last but not least speaker, the researcher Mus'ab Jad al-Haq. Mr. Mus'ab is I think he is like doctoral fellowship in Mangalore University in India. He is going to share with us his work, which is entitled Multi-Omics Analysis for Biomarkers and Therapeutics Targets Discovery in HER2-Positive Breast Cancer. So, Mus'ab, the floor is yours.
Assalamu alaikum.
Dr. Mus'ab, please.
Yeah. Do you hear me well?
Yes.
Yeah. So, welcome to you all. I don't know my screen like is visible now or not.
Yes, now it's visible.
Yes. So, good evening all. Thanks you for all. I am presenting to my topic, multi-omics characterization and identification of biomarkers in HER2-positive breast cancer. So, what is like HER2-positive? As you know, the breast cancer like is still the most frequently diagnosed and mortality at the woman and the worldwide. So HER2-positive is like characterized by two main factors, BB2 and amplifications and HER2-positive receptors overexpression. They are leading directly to aggressive tumor behavior and like a treatment resistance. So our gap in this research is like depend on how to identify the molecular biomarkers that will be essential to improve prognosis predictions and precision treatment strategies. So, just a minute. As you know, single omics is incomplete. Why? Because multi-omics more comprehensive landscape for like genomic and transcriptomic and proteomic level. So my main objectives that I presented in the research and my paper research, like identify the differentially expressed genes in the HER2-positive breast cancer and build the protein-protein interaction network by using string. And also like identify the biologically significant hub genes. What is the like my methodology workflow? My methodology workflow is like mainly depend on mainly in two phases. The first phase is like composed from like collection of like data. Like we have like a human genome nomenclature committee database. We have like 781 genes. Then we'll go like to using several platforms to identify which like higher like upregulated genes and like downregulated genes. So we have like 500 differentially expressed genes, and we have also 250 upregulated genes that higher expression, and also 250 down. Then we'll go by using string to build the protein-protein action. Then we'll go to cytoskip to like calculate or like present what is significantly mathematically hub genes and to rank hub genes. The other things is like validation and clinical relevance, hub genes identifications by going to functional enrichments pathways. We will identify that what is like these hub genes are related to this pathway or not, like clinical pathways, biological pathways, or like kinase pathways. And we'll go also to like 3 independent validations platforms we have like GBI-A3 and CBT-AC and the Human Protein Atlas. GBI-A3 like is for like proteomic quantification, CBT-AC for like proteomic levels, human protein for checking the immunohistochemicals like protein pathology. We are going then to survivor analysis by using KMP plotter, then for immune infiltration and genetic alteration by OncoPrint. And at the first result that we have used, we have like two like distinguished figures. One on the left, one on the right, sorry. In the left, like expression, we have like 25 genes are like highly regulated, and the other hand we have like 25 down regulated genes. Next we are going to sorry, just a minute. Where for hub genes identification by using Cytoscape, we are going to like arrangement for like these 10 hub genes. What is the mathematically used or mathematically like ways, and ranking these hub genes. Network findings like we are consistently identified by MCC and degree betweenness, closeness, and MNC algorithm. So for our like functional enrichment, we are checking the mitotic cell cycles, sister chromatoid, cell division P53. Other thing, we are going to check what is the hub genes across stages and metastasis. We have like here 3 different types of like stages for like at sample type, cancer stages, and nodal metastages. Here we have the highest number of like or hub genes that present tumor per million, UBE2C 80 years versus like 5 tumor per millions. My like major observation for these hub genes like dysregulations like happened and make irrespective of tumor stage or lymph node status. So after that, we are using like a human protein atlas to identification. What is the like protein pathogenic data and immunohistological figures for this protein or for these top 10 high genes. Proteomic validations, we have like a significantly elevated protein expression for like 2 groups of hub genes. First thing like CCNA2, UBE2C, 2C, and CCMB and RRM2. As a human protein atlas findings, we have like strongly immunohistochemistry staining in the CCNA2, CCMB1, CDC8, and BRIC. So our findings and interpretation for the results is like validated at the protein level. Other thing, we are going to checking what is the overall survival for these subgenes by using different kinds of tools like nomogram and KMP plotter. So here we are like we have different findings, results like protective genes, as we see here, like breast cancer 5, PREC5, hazardous ratio is 0.66, DLGAP5 hazardous ratio is 0.58. So the most important standout hub genes that we was founding in the NOCEP15 high risk for death, for like potentially for death happen. Then we are using like nomogram. We are like differentiate between the number of years that the hazardous ratio happens during several years of the cancer prognosis. Then we are going to checking the like immune infiltration and like between the tumor purity and what is the role of like a tumor cell killer in the prevent or like inhibition the tumor. So in the left side, we are seeing the immune microenvironment. If you are seeing here, we have 4 different hub genes, NUCAP1, DLGPE5, CCNA2 and UBE2C. So the most results or the most numbers of cancer tumors happen in the NUCAP1. That means that a few numbers of like immune cell killers have little bindings with this cancer tumors. On the other hand, we have like genomic alteration, which the hub genes have exposed to DNA change level. We have ORCA, highest alteration frequency, like 400%. That means patient number 4 out of 100 patient predominantly amplifications. So this percentage expressed or means we have like more druggable treatment for this hub genes, and can we find this like suitable treatment or for suitable drugs that can inhibit or can treat this mutational level. So the first thing, last slide I presented here, our conclusion like is identify the top 10 high genes that are associated with HER2 positive breast cancer. So also hub genes were consistently validated at the genomic level, transcriptomic level, proteomic level, survival level, and immune microenvironmental level. My future outcomes directions that their experimental validation using in vitro and in vivo models, prospective clinical cohort studies, integration of like epigenomics, metabolomics, single cell transcriptomic. And also the main goal that we can do in the future, development of biomarker that guided precision oncology strategies or approaches. Thank you all. Thank you for like mentors and faculty, starting from my professor, Dr. Mushtaq, and ourselves and other colleagues. Thank you so much for this, my presentation. So the discussion and the question are welcome for everyone here. So, Dr. Mahmoud Saddah, you have a mic. You can like speak. I have finished my. I cannot hear you well. I think.
Dr. Mahmoud.
Yeah.
Yes.
Dr. Mahmoud is muted now. So.
No, I am not muted.
Oh, I'm so sorry.
Can you hear me?
Yeah, I am hearing you.
You were muted,
yeah.
Dr. Mahmoud. You were muted.
Yeah. It's
at
okay.
the beginning.
Yeah, thank you.
Okay,
So please, Dr. Muhammad, please start the session. Start closing the session after getting all the questions from the audience.
Yeah. Dr. Muhammad is also muted.
Dr. Muhammad, you are muted. You are muted now.
Dr. Muhammad. So, what happened today? Okay. Yeah.
Now the first question with Mr. Ahmed. So please, Mr. Ahmed, you can start.
Yeah.
Hello, do you hear me?
Yeah, I'm hearing you well.
Okay. I'd like to thank you, Dr. Musab, for explanation about the biomarker. Actually, you explained how these hub gene were identified and validated. My question is, what's the clinical translatability of these findings? And I mean, could they eventually become part of the diagnostic panel?
So thank you for like this is a very good questions. So as you know, like it is like a candidate prognostic signature, not like a clinical test that can be used. So next future steps, sorry, is like depending on would be like as a prospective validations, not in the like independent cohort. So we can make a comparison between the existing clinical staging to show added predictive value and make like future models. Okay, so I think my answer is enough.
Okay, now for Dr. Loreen.
Yeah.
Actually, Mr. Ahmed, he addressed the same question. Thank you.
Sorry.
Okay.
There is like repeated sounds, Dr. Muhammad, like as a duplicated. That's why, yeah.
Ismail?
Yes. Can you
Please
hear
ask
me,
your question.
Thanks, Dr. Musab. While I listening to your presentation, I noticed that the analysis focuses specifically on HER2-positive breast cancer.
Yeah.
My question is, what was the rationale for selecting HER2-positive specifically rather than performing breast cancer analysis?
Yeah. I got your question. Thank you so much for this question. So, as you know, like HER2-positive is like clinical subtypes and the specific subtypes with like owner resistance mechanisms, despite like using of many monoclonal antibodies subtypes. So, subtype he have like subtypes or genes that can be or can make biomarkers for HER2-positive targeted decisions more than like using pan cancer or pancreatic cancer signature. So our models, our biomarkers that we are developing in this will be useful against like several diseases. Yeah.
Thank you, Dr. Musab. And now we reach the end of this session. I would like to thank Professor Mahmoud Sardah for his effort in the moderation this session. Thank you, Dr. Mahmoud, and also
thank you very much. It's my honor and a pleasure to participate in this very fruitful session and to co-chair this session with my friend and colleague, Dr. Muhammad Taha. Thank you so much.
Thank you, Professor Mahmoud. And also, I would like to thank all the speakers for their valuable presentation. And I would like to thank all my colleagues for their participation in the discussion part. Thank you so much.
Thank you.
And because we are behind our schedule, so there is no break. And directly we will go for the last session. And I would like to invite Professor Nahid Al-Laham and Dr. Muhammad Talib for moderation this session. Professor Nahid Al-Laham is a professor of microbiology from the Faculty of Applied Medical Sciences at Al-Azhar University of Gaza. And Dr. Muhammad Talib is assistant professor of microbiology at the Faculty of Pharmacy, Al-Azhar University of Gaza. So please, Professor Nahid and Dr. Muhammad, the floor is yours.
Thank you so much, Dr. Muhammad. Do you hear me?
Yes, Dr. Nahid.
Okay, thank you so much. Thank you for this kind of kindness, and we would like to start the last session in this conference, which is fourth session in this day, and it's about the microbiology. We have 5 speakers, and we'll start with our first speaker. Our first speaker is Dr. Nabil Al-Aila from Al-Aqsa University, Palestine, and he is now visiting professor at Ghent University in Belgium. He will talk about, or his topic is about correlation of biofilm and extended spectrum beta-lactamase production among gram-negative bacteria in a pediatric hospital, Gaza Strip, Palestine. Please, Dr. Nabil, the mic with you.
Thank you very much, Dr. Nahid. First, it is my great pleasure and honor to be with you today, and I would like to express my sincere thanks and gratitude to the conference organizer for giving me the opportunity to present my work and my research today. I am pleased to present I want to remove this, is okay. to present the screen. You see the presentation, Professor Nahad? Hello.
Yes, Dr. Nabil, I see it. Yes, it's clear.
Yeah, it's okay.
You go ahead, please.
And I am pleased to present our research, which is entitled Correlation of Biofilm and Extended Spectrum Beta-lactamase Among Gram-Negative Bacteria in Pediatric Hospital in Gaza. And this work and this study has been done with collaboration with Dr. El Afifi and Professor Al-Ham. And it is now I will give a brief introduction and we will go directly to the results of our study. I will, first, I will give my introduction about this research, because we are talking about antibiotic resistance, and the antibiotic resistance causes approximately 7,000 deaths annually worldwide. Gram-negative bacteria, it is considered as the major cause of a community-acquired infection and nosocomial infection and opportunistic infection. Two major resistant mechanisms it is present and we will talk about, which is extended spectrum beta-lactamase and biofilm formation. And these two types of resistant mechanism together, they are increasing treatment failure and chronic infection and mortality. And if we go to the extended spectrum beta-lactamase, it is enzymes that confer resistance to beta-lactam antibiotic, including the penicillin, cephalosporin, and monobactam. And why it is occurring, the extended spectrum beta-lactamase, it is occurring as a result of a mutation, and for which genes it is followed to several families like TIM and SHV and CTXM, and also we have OXA. And the mutation, it altered the amino acid configuration around the active site for beta-lactamase. And we here to indicate importantly that the plasmid, it has responsibility
Dr.
for
Nabil,
ESBL.
Yes? Sorry for disruption, but we didn't see your, we can't see your slide.
You have only, Dr. Nabil, the first slide. You didn't show us, the slides, only the first screen. Yes, now you can show it, from screening, from your screen.
Just presentation.
Yeah.
Just show it as you are doing with PPT. Yes, as a presentation.
Now it is shown clearly or not? Yes. When I go, it goes with you, the presentation?
Yes, now it is the background, the slide of the background.
The ESBL, the slide of ESBL. It is
no,
there?
It's the slide, the slide of the background. Second slide, showed on the screen.
It should work.
اعمل دكتور فول سكرين، اعمل فول سكرين، اعمل فول برزنتيشن.
Yes, it's not full screen for us.
No, not full presentation.
Not full screen. You mean, Dr. Ehab?
It's full screen now. It is okay like that?
No.
Yeah, it's okay.
No.
Yeah. And here,
no, Dr. Nabil, it's for us, for all the viewers, it's only the first slide, the slide of the title of your presentation, which is showed now on the screen for us. Now it's the slide number 3, which is regarding ESBL. Yes, but just show it as full screen.
Yes, it's okay. It is shown like this. I shared it. And the common ESBL producing pathogens like Escherichia coli, Klebsiella pneumonia, and Pseudomonas aeruginosa. Here, it is importantly to see in this slide the beta-lactamase, which is resistant to first and second generation of cephalosporin. And when there is a mutation, the mutation occur in the genome, it is considered and form ESBL. And what we will find in ESBL, it is clearly shown that it is resistant to first and second and third and also fourth generation in addition to monobactam. Because of that, the extended spectrum beta-lactamase increased the rate of resistance. Sorry. And
I'm sorry, Dr. Nabil. I'm sorry, Dr. Nabil, to interrupt you, but it's again, your slide is stopped on slide number 3. It's not moved. I know that you are talking about slide number 4 now, and now it's 5. I don't know when you stop. It's coming now. It's now
okay.
Slide number 5.
It's okay like this.
Yes, but we don't know what will happen when you talk.
You can open the presentation from your place. No problem for me.
We couldn't open it. You can open it. I couldn't open it. You sharing your presentation with us from your computer.
Yes, I shared it.
Okay.
اعمل فول سكرين يا دكتور.
اعمل فول سكرين زي ما بتعرض عندنا في البيت كانك تعرض.
انا عارف يا رجل والله فول سكرين.
مش معموله فول سكرين. It's not full screen.
اضغط على تحت البرزنتيشن في ايكون تضغط عليه بتحول لفول سكرين.
ايوه فول سكرين.
لا مش فول سكرين هذا.
ما تظهرش على الجانب دكتور، ظاهر على الجانب السلايدات الثانيه.
هي انا عندي هنا عامل فول. هي
مش هيك الفول سكرين.
معلش انا بدي اتدخل. معلش عفوا دكتور نبيل انت دكتور نبيل انت عامل شيرنج للملف الاصلي مش للبرزنتيشن. يعني هل جيت انت اعمل انشير ومن ثم روح على الديسكتوب واعمل شير للبرزنتيشن.
استنى شوي. هيك
الان اعمل برزنتيشن، اضغط على الايقونه اللي تحت اعمل تبعت البرزنتيشن.
هيك
F5
او F5.
هاي حطيت F5.
مش عارف يعني انا هيك يعني هذا كان
دكتور function F5
اغلق معلش بدي اغلق البرزنتيشن وافتحها ثاني.
ممكن بالضبط.
معلش غريبة يعني كل شيء تمام. هيك تمام. ظاهرة يا دكتور راهب؟
ظاهرة بس مش معمولة بوضع برزنتيشن.
يعني السلايد الأول واضح، بدك تعمل F5 أو تعمل يعني برزنتيشن، تشارك هيك برزنتيشن هيك سلايدز اللي واضح.
تمام هيك واضح تمام.
بنستنى لسه ما تغيرش شيء.
غريبة.
دكتور نبيل بعد اذنك ممكن اتدخل الان؟ انا البرزنتيشن عندي موجوده.
افتحها من
اذا بتحب افتحها بس لما تخلص من السلايد تحكي لي نيكست، فبالتالي انا بتابع معك.
ممتاز دكتور محمد، ممتاز.
شكرا.
غريبة.
تمام دكتور نبيل؟
تمام
تمام
توكل على
دكتور
الله.
محمد، شكرا لك، شكرا. تمام.
بتحب تبدأ من أول سلايد ولا في؟
لا توكل على الله. السلايد الثالثة. تمام. طبعا في هاي السلايد احنا
افتح المايك يا دكتور نبيل، سكر المايك.
تمام. واضح ان البيتا لاكتاميز اللي هي بتكون عندي ريزيستنت للفيرست والسكند جنريشن، والان نتيجه الميوتيشن في الجينوم بصير عندي ريزيستنت للفيرست والسكند والثيرد والفورث جنريشن والبونوباكتم، وهذه طبعا بتكون عندي نسبه الريزيستنس بتزيد في هذه الحاله. نيكست. واضح انه ليش الاي اس بي ال تعتبر مهمه جدا، لانها بتزيد من الهيلث كير كوستس، وبتزيد من المورتاليتي ريت، وبتزيد ايضا من الاميرجنج في الكوميونتي، ولونجر هوسبيتال ستيز. هذه اشياء مهمه جدا في الامباكت للاي اس بي ال. نيكست. نيكست دكتور محمد. الآن هذا كان جانب الاي اس بي ال، الآن لجانب البايوفيلم. البايوفيلم زي ما احنا شايفين في الصورة بشكل واضح عبارة عن بكتيريا ميكروبيال كوميونيتي. هذه الميكروبيال كوميونيتي بتعمل انكلوزنج وذ اكسترا سيلولار بوليميرميك ماتريكس، بحيث انه زي ما هو واضح عندي انها اول مراحل لها بتعمل ادهيجن، بعدين بتعمل مايكرو كولوني فورميشن، بعدين بصير لها ماتيوريشن، وبصير لها ديسبيرشن. طبعا ليش البايوفيلم الان باخذ اهتمام كبير؟ حتى هنا في بلجيكا الان ابحاث كبيره جدا بيشتغلوا عليه هذا الموضوع، نظرا لانه له امباكت كبير من حيث انه بيعمل وبروتكت للبكتيريا من المضاد الحيوي، وبيعمل انهانس لمين عندي؟ للبرسيستنس، وبروموت كذلك للجين ترانسفير. نيكست. الامباكت الان حكينا عن ظاهرتين مهمين جدا حنحكي عليهم كلينيكالي، البايوفيلم والاي اس بي ال. ليش الموضوع هم يعتبر مهم جدا؟ لانه البايوفيلم فورميشن والاي اس بي ال بشكل خطوره صحيه كبيره، بحيث انه بيؤدي الى انه صعوبه علاج المرضى، بحيث انهم وجدوا انه عمليه فشل العلاج للمرضى مش بس عشان الاي اس بي ال، فوجدوا انه البايوفيلم كذلك مشاركته في هذا الامر ووجوده بيعمل عمليه مقاومه بتزيد اكثر من 100 مره، وهذا بيأثر على عمليه علاج المرضى، بحيث انهم وجدوا تقريبا حوالي 65% من المرضى من الامراض و80% من الامراض المزمنه مرتبطه بمين؟ بالبايوفيلم فورميشن. فبالتالي وجود اثنين مع بعض بزيد المصيبه الكبيره، بحيث انه الاي اس بي ال مع البايوفيلم بيؤدي لمين؟ لكرونيك بيرسيستنت وريكارنت كلينيكال انفكشن. نيكست. Objective. The general objective of our study to investigate the relationship between ESBL production and biofilm formation in pediatric patient population at Al-Nasr Hospital. What we will do among our specific objective in this study, we will identify ESBL producing strains among gram-negative bacteria isolate. We will detect the biofilm formation, and we will evaluate the antibiotic susceptibility testing and investigate the statistical correlation between ESBL production and biofilm formation in pediatric patients. Next. Material method. Our study is a cross-sectional study in pediatric hospital. It's a Nasr pediatric hospital. Sample size, we have taken 89 gram-negative isolate. The source of our clinical sample was urine, pus, sputum, blood, ear discharge. We have ethical consideration from Ministry of Health and Helsinki Committee. Next, please. The material method, we identify our isolate by culture, by ABI, and we have done also ESBL identification by double disc synergy test. We have done the biofilm detection using microtiter plate method, and the susceptibility using Kirby-Pur method according to the last CLSI guidelines, and we have analyzed our result using SPSS. Next. Results. What we have obtained, our results among the 89 isolates from
الرانتيسي هوسبيتال Nasser Hospital. We have the ASBL production. If we clearly, we have more than 50% of the isolate were ESBL. It means that they are resistant bacteria. And if we see in our table, we have the Escherichia coli and the Klebsiella. They are among the bacterial isolate which are producing ESBL. Next. Here, it is important slide, shows the bacteria which are producing ESBL, Escherichia coli and Klebsiella. They are more producing ESBL isolate than the non-ESBL isolates. And we have also Acinetobacter baumannii and the Proteus mirabilis. Those bacteria are producing ESBL. Next. This is our experiment in the lab. By this method, the synergy which seen clearly in our plates, by which we can identify ESBL for the clinical isolates from the hospital. Next. Here, we would like to indicate importantly for the biofilm. We indicated the result of ESBL. For the biofilm, we have 65 isolate. They are 73% biofilm producer, and 25 were non-biofilm producer. And among the 65 biofilm producer, we analyze it as strong and moderate and weak producer. And we found 27, which considered as 41% were strong biofilm producer, and the 20, which represent 30%, were moderate, and 27 were weaker producer. Next, please. And next slide, next. Yes, here it is indicated that the biofilm, they are 73, which is high rate of biofilm producer. And also if we see clearly, the strong biofilm producer represent 41.5%, and we have moderate 30.77% for biofilm producer. Next. And if we see our isolate, it is known for many of publication clearly which microorganism, as we see in this slide, more biofilm producer is Pseudomonas aeruginosa, and especially with biofilm formation, especially with the patient of cystic fibrosis. And the Pseudomonas aeruginosa gives high rate of biofilm producer, and it comes then Klebsiella pneumonia, it produces biofilm producer, and then it is Escherichia coli. And among the resistant isolate, we have Acinetobacter baumannii also, and Proteus mirabilis biofilm producer. Next.
Dr. Nabil, I'm sorry for interruption, but try to finish because we are over the time.
Yeah, I will. It is. I am trying now. And here it is shown here in our results the strong and the moderate and weak biofilm producer, and it is shown also the Pseudomonas and the Klebsiella pneumonia. They are strong producer for biofilm. Next, please. Here, which is very important point of our results, that we found very high correlation between biofilm and ESBL, and maybe will be shown more clearly in next slide because the time is very short now. We will finish in minutes. Next, please. Yes. Here, it is very important slide. You see the ESBL and the biofilm. We have here combination between two important things. We have high rate of biofilm production, and we have high rate of ESBL, which indicate that the problem will be exaggerated more for such patients when they have both the biofilm and ESBL producer. And we have a strong correlation between biofilm and ESBL. Next, please. The clinical significance that we have multi-drug resistance, we have persistent infection, we have recurrent infection and increased hospitalization of such patient. And also we found the next slide, the rate of sensitivity. Next slide, please. That the antibiotic susceptibility we have, as shown and as known, imipenem and meropenem and piperacillin and imicacine shows the most effective antibiotic, and the ESBL isolates were resistant to most cephalosporin. You see cefuroxime and cefatoxime and ceftriaxone, it was almost resistant for all antibiotics. And it is also shown clearly in this slide. Next slide. I am almost finished, Dr. Nahad. Here it is shown that all the cephalosporin, and here it is important, and also we give this message in all of our presentation to all of our hospital. You see the first and second and third generation of cephalosporin, how are scoring highest resistant to the antibiotic. Next, please. Conclusion. What we conclude from this study? We conclude, importantly, we have high prevalence of ESBLs in pediatric hospital, and we have done also in other hospitals, and we have high rate of ESBL production, and we have high rate of biofilm production, and we found a clearly significant correlation between ESBL production and biofilm formation. And we found clearly among the antibiotic resistant results that nearly complete resistance to third generation cephalosporin. What remains the antibiotic that we still work with is the carbapenem, is the most effective antibiotic. It gives us the point for public health concern for ESBL and biofilm are difficult to treat. They high risk to transmission in hospital setting, and it is importantly enhanced surveillance and antibiotic stewardship are urgently needed. So our last recommendation in our last slide, next please. Recommendation, briefly I will talk. We have to implement importantly, and that our message to the Minister of Health, antibiotic stewardship program, to restrict the empirical use of third generation of cephalosporin and to promote carbapenem use when used only. And also we have to strengthen the infection control in our hospitals and laboratory practice to detect the SBL and biofilm and regular antibiotic resistance surveillance reporting. And lastly, public health, very importantly in our society to be considered through national AMR surveillance program, public awareness campaigns on antibiotic use, policy and rational antibiotic use. Next. Thank you very much for listening. This is my presentation and welcome for any question.
Thank you so much, Dr. Nabil, for this nice presentation. And we would like to open the questions only for one questions because we spend a lot of time in this lecture. So please, any question to Dr. Nabil? If there is no any questions, I would like to ask one question. Please, Dr. Nabil, just in this study, you mentioned the correlation between ESBL and the biofilm and its importance in antibiotic resistance.
Yes.
Just if it is another kind of resistance, like carbapenem resistance, it could be the same situation will be, or it could be a different situation?
It is worse than my friend. The situation will be more worse. Actually, if we have this situation with ESBL, what the situation will be more danger with carbapenem actually, because here we have the more resistance for the genome, and so because of that we give through our research and publication together, we give more attention that we have to take care for antibiotic prescription because we have high rate of biofilm, we have high rate of carbapenem resistant in last publication, and also with ESBL production.
Again, we would like to thank you very much for your nice presentation. And now I will give the mic to my colleague, my beloved brother, Dr. Muhammad Talib, to present our next speaker. Please, Dr.
And
Muhammad.
also my gratitude to Dr. Talib.
Yes.
Thank you.
Yes, Dr. Nabil and Dr. Nahid. That's nice moment to meet Dr. Nahid and Dr. Nabil in the same moment in our conference.
Thank you, my brother. Thank you so much.
Thank you very much,
That's-
Dr. Talib.
Yes. Most welcome, Dr. Nahid, and partnering and sharing in the moderating of this session. And congratulations for Dr. Nabil for this interesting lectures. This is very good on the fact of Gaza strip in antibiotic resistance, especially in the war and after war and before war.
Yes,
Now, because we have no time, now, inshallah, we will transfer to the next speaker, Dr. Hani Dakka, of UNRWA and the beloved and friend Dr. Hani, UNRWA Palestine, by the research article which titled Microbial Sources Tracking of Fecal Contamination in Fresh and Marine Water in the Gaza Strip using discriminant analysis of antibiotic resistant patterns. Most welcome, Dr. Hani. And
Hello.
yes.
Just a moment, I want to make a share to share the. Where is the chair? Yes. No. As background. I tried to make it share. I think I have the same problem with Dr. Nabil.
Dr. Muhammad Taha, from the beginning.
No, I have to.
Ground, sharing, Dr.
Dr. Muhammad.
Sharing Dr. Hani lecturer slides, because I think that's the same problem of Dr. Nabil. It's not, yes.
Is it infection for the microbiologist, maybe?
I think that's Ain al-Hasud, Dr. Nahid. I think that's Ain al-Hasud.
I don't know what's the problem. I tried to make a chair, but as background, the same problem. Same problem. I tried it before.
Yes, I think that Dr. Taha now tried to share you on by slides.
Okay.
He told me.
I don't know what the problem. I tried it before and it was okay.
Dr. Hani, we diagnosed from Dr. and the Prof. Dr. Nahid, that's
Yes.
infection which will infect microbiologists. Yeah.
Actually.
So,
Yes.
Dr. Hani did.
It is viral infection.
Just let me know when I go to the next slide, please.
Okay. I will let you, inshallah. Okay, my presentation about microbial source tracking of fecal contamination and fresh and marine water using a function in mathematics called discriminant analysis using antibiotic resistant pathogens. This is the address. Next, please. Yes, the background, as Dr. Nabil mentioned before, that there is a very critical issue regarding the antibiotic-resistant bacteria, which killed more than 7,000 per year. Water contamination is a major public health issue, causes many diseases called water diseases like gastroenteritis, like cholera, like hepatitis. The limited freshwater and wastewater discharge increase the risk of microbial infection in water. Next. The problem, traditional fecal indicators, bacteria like total coliform, fecal coliform, can give a prediction of contamination, but cannot identify the contamination source. This is a problem. Next. So the aim of the study is to evaluate antibiotic resistant patterns using differential or discriminant function analysis for the identifying fecal contamination source in fresh and marine water. Next. The study design, actually, it was my PhD thesis. The samples were 431 samples, including human feces, animal feces, birds, marine, and fresh water. Next. The methodology was sample collection and the traditional way for identification bacteria, like membrane filter technique, and then the disk diffusion antibiotic test. Then the result. Yes. We see here in this table, after putting the results in the discriminant analysis function, that over classification rate was 58.8 when make the samples like a human. Then the animals is categorized like cow, sheep, rabbit, horses. When we see these results, we see moderate performance. We see strong overlap among animal species. We see difficult species level of discrimination. The other, please. When make a pooling, not considered the animal feces like horses, like donkeys, like cats. When we pooling it, the classification rate was raised, was improved from 58 to 90 or to 89. There was an improvement when we make a pooling for the feces samples like animal to human. Next, please. This is also for the birds. When we categorize birds like chicken, ducks, and turkey parallel to human, the classification rate was 81. We find a better performance than animal model, and we see some overlap among bird species. Next. When we make a pooling for the bird sources, not like before separated, the correct classification rate was 98% of original group. And this was the highest classification accuracy in the entire study when we make a pooling for the bird parallel to human. Next. This is when we enter the marine and the freshwater analysis and the discrimination, and this parallel to human and cows, sheep, like this separated animals, the overall accuracy was 43.4. It is the lowest performance observed because of source mixing, environmental complexity from marine to fresh to human, like this. Next, please. This is when we make a pooling, animal as a pool, a human, freshwater. If you see the classification rate was the high and was improved, dramatically improved classification. Next, please. This is another thing when we make a pooling or make a comparison with the chicken and duck with the bird source. Also, we can see lowering in the classification rate comparing when we make a pooling for the result. Next, please. This is when make a pooling as a bird, one package, then the human, fresh, marine. We see the overlap of all accuracy of classification was improved, dramatically was improved classification. Next, please. This is a comparison before water and after water according animal. It was 85 when we entered the water. It was lower. The bird is reverse, and the pool it was the highest classification rate. Please, the next. This is another graph. Make a comparison between separated pooling. When we make a pool, the accuracy of classification will be high. Next, please. Another discriminant analysis classification, see the same result. Next, please. Next, please. The discussion. Why did pooling improve accuracy? Explanation. Pooling reduced the variability within groups, enhanced separation between groups, strengthening the discriminant function. Example, animals from 58 when making pool to 89. Birds from 81 to 98. Please, next. Why were bird models more accurate? Possible, distinct gut microbial, different antibiotic exposure, because the birds is treated with antibiotics different from a human, not like a human to animals. Birds' antibiotic resistance are more distinguishable than mammalian antibiotic resistance. Next, please. Why were animal models less accurate? Main reason, overlapping antibiotic resistance. Animals often share similar antibiotics, similar environment, similar farming practices. There is a similarity between antibiotics used in veterinary and the human. Result, poor species level discrimination. Next, please. Next, doctor. Why did water reduce accuracy when put water samples? That accuracy was lower because environmental contain mixed contamination. To go to conclusion, main conclusion, antibiotic resistance based on discriminant analysis effectively distinguish. Why did okay main conclusion to summary, antibiotic resistance, no the previous one, doctor, yes, here main conclusion that antibiotic resistant patterns is effective to distinguish a human contamination from others. No the previous doctor. Yes, best performance obtained with pooled bird sources. Pooled sources greatly improve classification. Environmental water reduce model accuracy. Fine scale animal source discriminant remains difficult. The last one, please. Recommendation using antibiotic and discriminant analysis as a rapid low-cost screening tool, applying primarily for broad sourcing identification. Combine antibiotic resistant with molecular biology to enhance the technique. Incorporate physicochemical water quality parameters. Develop integrated monitoring program for Gaza water resources. This is the last, I think. Thank you very much. And I am happy to listen for your questions.
Yes, thanks, Dr. Hani. Thanks for these lectures, nice lectures and shorted.
Thank you very much.
Yeah. Now with Dr. Nahed.
I'm sorry, I was rabid because the time is running and we have no time to.
Okay. It's nice, doctor. It's nice, Dr. Hani. Now we are transferred to my friend and my colleague, Dr. Nahed, to introduce the next speaker.
Okay.
Thank you, Dr. Muhammad, so much. Thank you, Doctor, for so much. Now we have a lecture that will be presented by Dr. Ahmed Al-Sayed from Applied Science Private University in Jordan. The title of his talk will be Integrating Molecular Diagnostics and Antimicrobial Resistance Surveillance in Hospitalized Pneumonia Patients. Insights from Jordan. I guess we will hear a nice lecture from you, Dr. Ahmed. Please, the microphone with you.
Bismillah, assalamu alaikum jami'an.
Alaikum assalam wa rahmatullahi wa barakatuh.
Good evening, everyone. Before I begin, I would like to express my appreciation to the organizers, despite the many challenges. My sincere hope is that the next conference will be held virtually in a time with Gaza restored and Palestine free, inshallah. My presentation is entitled Integrating Molecular Diagnostics and Antimicrobial Resistance Surveillance in Hospitalized Pneumonia Patients, Insights from Jordan. So why does pneumonia still matter? First of all, pneumonia is the leading infectious cause of death globally, and due to the culture limitation, which is the gold standard, I know, but there's some limitation, particularly the delayed diagnosis or the delay in the time of diagnosis, and increasing antimicrobial resistance, as mentioned from previous doctors. So, meaning we need for a rapid precision medicine. So our research project actually addressed the mentioned challenges, and in the last 2 years we published more than 4 studies related to the pneumonia. Okay, and I will, in this presentation, I will introduce them very briefly. So the overall aim of our research was to advance precision medicines in pneumonia through molecular diagnostics and artificial intelligence. So the first objective was to improve pathogen detection using molecular techniques. The second objective was to detect antimicrobial resistant genes rapidly to guide appropriate empirical therapy and support antimicrobial stewardship. The third objective was to evaluate PCR performance, so compare between PCR and conventional culture to determine the diagnostics accuracy, sensitivity, specificity, and overall clinical impact. The fourth objective was to assess the clinical outcome, to understand factors associated with morbidity and mortality, and finally, to develop AI prediction model, so use machine learning to predict etiology and mortality risk for early interventions and personalized patient management. Starting with the first study, it was a prospective study performed during 2021 and 2022, based in a private hospital in Amman, Jordan, involving 111 hospitalized patients with pneumonia. The aim was to compare multiplex real-time PCR with conventional culture for pathogen detection. PCR actually demonstrated or detected different bacteria, common and uncommon bacteria, and also multiple viruses not detected by culture. One of the interesting findings that the polyvirus and virus related pneumonia was around 1/3 of the cases. So the empiric treatment was not needed for those individuals. And this actually aligned with some global findings. Regarding the clinical outcome and pathogen detection, a non-significant but borderline association exists between the pathogen detected and the clinical outcome. And the worst clinical outcome were reported for Staphylococcus aureus, including methicillin-sensitive and methicillin-resistant Staphylococcus aureus, in addition to Bordetella pertussis, Klebsiella pneumonia, and Pseudomonas aeruginosa. And a better clinical outcome was reported for the different viruses. Regarding antimicrobial resistant patterns, actually, levofloxacin demonstrated the highest susceptibility pattern, whereas ampicillin demonstrated the highest resistance pattern. This heat map highlighted the variable resistance profile across pathogens and antibiotics. Regarding the seasonal variation, actually, meropenem and piperacillin tazobactam peaked during resistance peaked during September and October. Cefepime peaked until the end of the year, so mainly particularly during the colder months. This indicates that antibiotic resistance patterns have heterogeneous and showed temporal variation, emphasizing the importance of continuous antimicrobial surveillance and antibiotic stewardship. Okay. Actually, this gives an overview for the first study. So moving to the second study, which was an observation study involving 114 patients with community acquired pneumonia. The aim was to detect the antibiotic resistant genes. So here we used a qPCR kit that target different antibiotic resistant genes. So using the qPCR, real-time PCR. One of the results, the most frequently detected genes was MIC-A gene, followed by TIM, then OXA51-like and OXA23-like genes. As expected, significant association were found between MIC-A gene and methicillin resistance, and also between OXA51-like gene and ertapenem. So BCR has demonstrated increased sensitivity over traditional culture techniques, so detecting resistance in cases where culture failed to identify pathogen. So moving to the third study, which was also a prospective study performed during 2024 involving 83 hospital-acquired pneumonia, including VAP, ventilator-associated pneumonia patients enrolled at the University of Jordan Hospital. Around 1/4 of the total patients were classified as VAP, and the mortality rate was slightly less than 50%. Again, we use the traditional cultures and also the molecular techniques and compare between these findings in terms of pathogen detection here and also antibiotic resistant genes. So the different study, either the population is different, CAB or HAB, and also some study focus on pathogen detection alone, some with antibiotic resistant genes. Okay, so regarding the antimicrobial or the empiric selections, the most frequently used empiric antibiotic, unfortunately, vancomycin, meropenem, levofloxacin, imipenem, piperacillin, tazobactam. Unfortunately, because these are very broad spectrum antibiotic. Also, the colistin was used empirically in around 5%, and then in around 20% as initial targeted therapy. Regarding the high resistance or high prevalence of multiple resistant microorganisms, Acinetobacter baumannii exhibited, as expected, 100% resistant to imipenem, meropenem, and piperacillin, tazobactam, and colistin was the remaining highly effective agent. Regarding Klebsiella pneumonia, showed 100% resistance to meropenem, piperacillin, tazobactam, which are used as empirically, and also toward the amoxicillin clavulanic acid. Tigecycline was effective. Regarding Pseudomonas aeruginosa, colistin achieved 100% sensitivity, followed by imipenem of around 90%, and the ceftazidime has the lowest sensitivity of around 30%. As well, there was a higher detection rates using BCR compared to traditional cultures. Here's the distribution of the detection of antibiotic resistant genes using BCR. And unfortunately, this actually revealed detrimental consequences in terms of antibiotic resistance. So moving to the last study, which is focus on machine learning, to predict two outcomes, mortality and bacterial infections. Okay, in community acquired patients. So in community acquired pneumonia, sorry. This study was a retrospective study involving 251 CAB patients in Jordan. We performed the study using the following 6 steps. First of all, patient data collection, followed by data pre-processing, and then feature extraction and selection, followed by machine learning training, then results evaluation, and finally predicting outcome probability. So one of the challenges in the machine learning, the multicollinearity, the redundant information. So if you want, we found, multicollinearity among the variables, suggesting overlapping clinical patterns, or what's called redundant informations about the outcome. Variables. So this make us do feature reductions for the variables. Here in this correlation matrix, we found that the zinc and vitamin C demonstrated the highest positive outcome or correlation with the outcome, which is the survival or mortality. And also, but also insulin bolus, the use of insulin bolus and ertapenem showed moderate association or correlation with the outcome. So I'm talking first about the mortality outcome. So using the mutual information algorithms, we found that the most informative predictors for mortality are the kidney function tests, including creatinine and blood urea nitrogen, white blood cells, including eosinophils and neutrophils, and liver function tests, including ALT, in addition to previous hospitalization. Using the lasso regression, we found that the most important predictors are medications, mainly meropenem and vancomycin, also ABG's markers, base excess and partial pressure of CO2, in addition to platelet count. And then using logistic regression algorithm, we found that the most predictors for outcome, for mortality outcome were lower pH, medication, white blood cell count, bicarbonate, and albumin. And we did the same idea for another outcome, which is bacterial infections. So just briefly, one of the important results that the anticoagulants showed significant association with the bacterial infection, the use of anticoagulant, I mean, and also the initial fever findings and basophil exhibited moderate positive correlation with the bacterial infection. Actually, this table shows the performance metrics of the logistic regression used in this study. And this table shows the comparative variable importance for the different algorithm used. We can notice that there are some difference in terms of the correlations of different variables with the outcome. One of the important limitations of this study, the sample size, although it's 251, but in terms of machine learning, we need actually more sample size to gain better insights into the results or the implications. And this is just a summary of the FOR study, just to focus on the integration of molecular techniques, not to replace culture, of course, but adding molecular techniques in the clinical practice is very, very important, with some, of course, limitations, and also using the artificial intelligence machine learning to predict the outcome and to predict the bacterial infection, especially in the cases when we cannot perform the PCR due to, the high cost. And thank you very much. I'm happy to receive any questions.
Thank you very much, Dr. Ahmed, for this interesting lecture. Now we will open to get some questions from the audience. Anyone have any questions, please? Dr. Loreen Hapoub, please, mic with you.
Yeah, thank you. Thank you, Dr. Ahmed, for this presentation. It's really interesting, especially that it has been conducted in Jordan. So I'm so happy for you and for this achievement. Actually, as I am not familiar in the techniques used in microbiology, but I know the same technique but used in another domain. So I was wondering that do you think that a qPCR technique is sufficient to catch and to detect the resistance-related genes? Is it sufficient? Because I think that if I catch it well, what you are doing is that you are doing analysis for the bulk cells from bacteria or whatever. But do you think that analysis the bulk cells is sufficient? You don't expect that you have a bias or that you missed some genes? This is my first question.
Okay. If I understand your question, do you mean like detection of the antibiotic resistant gene that doesn't guarantee the expression of that genes? So doesn't guarantee that the bacteria...
Not exactly to not guarantee, but it's not, if it's sufficient, maybe you miss other genes. This is only my question.
Okay, actually, we depend on the commercial kits, actually, and detecting the most important antibiotics. And that's why we run this test to see if they are highly correlated actually with the real resistance. So for example, MIC-A gene was associated with methicillin resistant, which is as expected. So this is type of some, let's say, validation or confirming that the idea can be useful in clinical practice.
Yeah, so you confirm by this analysis, and because also my question concerns the technical part, is if you did the qPCR analysis on the bulk cells, the total, the bulk of the cells, for sure it's not each bacterial cell.
We used the respiratory sample, mainly sputum sample, and then we performed DNA extractions.
Yeah.
Then we use real-time PCR using a kit that contain different antibiotic resistant genes and also different targets for bacteria itself. So and we compare it with cultures.
Yeah, so it's on the bulk. It's on the bulk. So my second question, you mentioned that you found, or in the bibliography, there is a correlation between anticoagulants and infection. True. My question: do you know what is the molecular mechanism or the pathophysiology behind this correlation?
Related to the machine learning, you mean? So
you mentioned in the slide just before that. Yeah, before. No, just before, I think.
You mean the predictors? From here, I start.
No, it was, I think, before.
It is
anyway,
in the third study, Dr. Ahmed, and not in the machine learning. No, when you correlate the predictive, the risk factors for mortality and morbidity.
I don't think so. The third study focusing on hospital-acquired pneumonia to compare between pathogen detection using culture and PCR and antibiotic resistance using PCR and culture. Can you just repeat your question just to know?
Yeah, you mentioned that there is a correlation between the anticoagulants and infection. This means that using anticoagulants is correlated, favorize the infection. Yeah, that's it.
Yes.
Yeah, there is association, meaning that the patients taking anticoagulants are susceptible for bacterial infection.
Yes, depending on this algorithm, actually.
Yeah, so here I asked if you have a molecular mechanism that has been described in the literature, or you have already known, or what is the pathophysiology behind this association?
Really, I'm not sure, actually.
Yeah, okay.
Yeah.
Okay, Dr. Noreen, thank you. We have other two questions, but please be brief and fast. Dr. Hani Dakka and then Dr. Nabil Al-Aila, please. Brief. Dr. Hani, you raised your hand. Dr. Hani?
نعم دكتور.
Please your question for Dr. Ahmed in brief and fast.
It's not a question. It is apologize from Mr. Afifi. The next presenter have a special condition. He want to delay his presentation for the last one, if you can, please.
So, okay, we will let Dr. Muhammad present the next speaker, but we will finish. Please, Dr. Nabil, the mic with you. Yes, please, your questions.
Tamam. Dr. Ahmed, it is really very nice articles and presentation you made. And here my question of our experience in culture and PCR for the patients who has pneumonia. My questions for the people who are has pneumonic infection, how you can give the general evaluation if it is bacterial, viral infection or fungal infection. And we know we have different causative agents for pneumonic infection. And this is one part of the question, according to the good experience I see of your articles. And the point, what was the correlation between the culture and PCR you have done in your study? I mean, what do you find in case you have culture negative, what are the most common microorganism isolated? Do you have used certain primers or probes, multiplex PCR for certain microorganism? That's the question, please.
Okay, thank you for your questions. To start with the last questions, if you mean the comparison between sensitivity and specificity of the PCR culture due to limited time, I didn't.
Not exactly, Dr. Ahmed. Just the point of view of sensitivity and specificity. I agree. For this point is very important now for comparison. But I mean for the variety of the microorganism you isolate. I mean, in case you have done culture and PCR, how many, I mean, you find the culture negative and you find the PCR positive or vice versa or something like that? Because really it is interesting.
Yeah, as shown here, the positive, the rate of detections for PCR was 74.8%. Okay.
Yes.
Versus 57.7% using the culture.
Yes, and as regards to the type of microorganism, the different. Do you just find the strep? It is specific for strep pneumonia or different microorganism causing the pneumonia? That is the point.
Different microorganisms. There are some microorganisms common between culture and PCR, and some like extra with BCRs and not available in the culture, like for example viruses. But we compare between the percentage of detection for the same sample using culture and BCR. So many cases was positive for BCR but not positive for culture. One of the possible explanation, the use of antibiotic. Even one dose of the antibiotic will affect the culture result, and this in comparison, this advantage of BCR, the higher sensitivity, so it can detect the pathogens even after taking some antibiotics. And I'm not sure if I answered your question.
The types of fungi and viruses, do you detect them for the people who are
the kit itself included one fungus, which is Pneumocystis cerevisiae, but there is no data using the culture. You know, it's the culture for this type of fungus is very challenging.
It's okay.
And we found some positive cases for Pneumocystis cerevisiae. I'm not sure if I answered your question. And what is the just remind me about the first question?
Yeah, actually, the different viruses you detect, is there certain, when you have done the PCR, do you use the primer for different viruses and fungi? When do you have done conventional? Because really this part is very interesting and important, especially in our labs.
Actually, we didn't design the primers and probes. We used it contained 33 different targets, including many viruses and fungus. Actually, it's detailed in the same article, but we didn't design.
I will go through the article also. It's interesting to read. Thank you.
Thank you again, Dr. Nabil and Dr. Ahmed. We will conclude to finish and to move to the next speaker with Dr. Muhammad. But I have one very small question for Dr. Ahmed. You mentioned that some organisms get VAN A and VAN B positive. Is it VRE, vancomycin resistant enterococci, or VRSA, vancomycin resistant Staphylococcus aureus? Just the question.
Enterococcus.
Okay, VRE. Thank you so much. Thank you a lot. Thank you a lot. And we wish, you to welcome you, inshallah, in Gaza Strip, hopefully for the next conference. Please, my colleague, Dr. Muhammad, you can introduce now the next speaker. Mr. Ahmad Afifi asked to delay his lecture, so please you can go with the next speaker. Please, thank you.
Yes, Dr. Nahed. I have many questions for Dr. Ahmed because it's very interesting lectures but Dr. Taha told me that's no times, and Dr. Nahid allow for me, inshallah, to introduce Miss Ruhaifa Al-Haddad, AUG, Al-Azhar University, our friend and our colleague and from Palestine, to introduce this research and her research by titled Antibacterial Activity of Fresh Garlic Allium Sativa L Juice Against Selected Bacteria. Most welcome, Ms. Ruhaifa, and the mic with you. Ms. Ruhaifa. Yes, Ruhaifa. You hear me? Ruhaifa?
She is not there.
Do you hear me, Dr. Muhammad?
Yes, yes.
I am with you.
Yes, most welcome, Ruhaifa, and inshallah, you have short time because we have no time for this session, and finalized the conference, inshallah. Ruhaifa,
Yes.
mic with you.
Yes, yes. Hello, everyone. I hope that you are all well. First of all, it's my honor to be here today. I would like to express my sincere gratitude to the organization committee for giving me this opportunity to present our study. In this presentation, I would like to highlight
Share
our latest findings regarding the...
Share your representation, please.
Yes.
I share
and move slides, move slides, move slides. It may be shared.
Yes. Okay.
Not. No, okay.
No. What's happened?
Try to make F5.
Windows F5.
Yes.
Virus spreading.
This is yeah, this is the disadvantages of microbiology session.
أستاذ رهيفة بالأول اعملي لو سمحتي تمكين التحرير اضغط عليها.
صحيح صحيح فوق.
Yes.
الآن ابدأ شير حتى تمشي إن شاء الله. هذا هو الأنتيبيوتيك للإنفكشن.
شكرا دكتور محمد.
You're welcome, Dr. Nahid.
Okay. Okay.
Till now, not okay. Try to share.
I share it.
Dr. Muhammad,
I think that the shape, Dr. Nahid, is very warm. Or what is the shape this? Yes.
Until now, it's okay?
Yes, yes. اعملي full presentation. ايوه تمام. Score.
Okay.
Yes, yes.
Yeah. Yes. My research, or our research, is titled by in vitro antibacterial activity of fresh garlic juice against standard bacterial strain and clinical isolates. This research was co-authored and conducted together with my colleague, Dr. Munawadi. Introduction. The continuous development of multi-drug-resistant pathogens have become serious threats to public health worldwide. The problem of antibiotic resistance and drug toxicity have made the continuous search for newer antibiotic against multi-drug-resistant pathogen a necessity. Therefore, there is an increasing need for less toxic, cheap, easily sourced, and effective alternative antibacterial agent that can treat disease caused by bacteria and other infectious agents. A newer helper antibacterial compound from a plant are under investigation to overcome the antibacterial resistance. Garlic has long been known to inhibit many gram-positive and gram-negative bacteria. However, despite the extensive use of garlic in traditional medicine and the promising antibacterial property exhibited in a previous study, there is a lack of study investigation the antibacterial activity of a fresh garlic juice against a wide range of bacterial strains in Gaza. Moreover, comparative study assessing the effectiveness of garlic juice against commonly used antibiotics are limited. The main objective of our study was to determine the antibacterial activity of fresh garlic juice against selected standard strain and clinical isolate bacteria, and compare its effect with one of the commonly used antibiotic. The material and method, I will briefly talking about the material and method. The fresh garlic juice was processed, peeled, and crushed and filtered to have a stock solution considered as 100% fresh garlic extract, and stored as in a sterile capped bottle at 4 degrees centigrade in light shielded container. Bacterial strain, we have two types of bacterial strain, standard strain and clinical isolate. The standard strain included Escherichia coli, Staphylococcus aureus, Staphylococcus epidermis, another phenotype of Staphylococcus aureus, and Pseudomonas aeruginosa. The standard bacterial strain utilized in this study were generously provided by the Drug and Toxicology Analysis Center, Al-Azhar University, Gaza, Palestine. The clinical isolate, we have Klebsiella pneumonia, Escherichia coli, Proteus mirabilis, Acinetobacter baumannii, and Staphylococcus aureus. The clinical isolate I obtained from a private laboratory in Gaza, Palestine. This strain were originally isolated from various clinical specimens, such as urine and wound and another swabs. And were selected for this study due to their prevalence in local clinical settings and their known resistance to conventional antibiotic. The identity of these isolates was reconfirmed in the laboratory using a standard biochemical test before testing. Antibiogram profile determined by the disk diffusion method revealed resistance of all tested organisms except Escherichia coli to at least 3 antibiotics following a standard microbiological protocol. This table shows the antibiotic susceptibility profile of the clinical isolate included in our study. Evaluation of antibacterial activity, the antimicrobial assay of garlic extract was performed by agar wheel diffusion method. Amikacin was used as a positive control. The diameter of the observed inhibition zone were measured and recorded in millimeter. The antibacterial assay was performed in triplicate. MIC determination by serial dilution technique, we determined the MIC serial dilution from 50 milligram per ml were done and determined the MIC. MBC also determined by loopful of the inoculum of each non-turbid tubes of MIC assay, streaked on a nutrient agar plate and incubated at 37 degrees centigrade for 24 hours. Lowest concentration at which there was no visible growth was regarded the MBC. Statistical analysis where data were analyzed by one-way ANOVA, and differences were considered statistically significant at level B less than 0.05. Results The results in this table shows us fresh garlic juice exhibited significantly varying antibacterial activity against tested bacteria at concentration 25%, 50%, and 100%. At 25%, fresh garlic juice shows largest inhibition zone for the standard bacteria Escherichia coli with 36.6 millimeter inhibition zone, then Staph epidermis with 35 millimeter inhibition zone, while the smallest inhibition zone observed against Proteus mirabilis and Acinetobacter baumannii with inhibition zone 10.33. At concentration 50%, the largest inhibition zone was also for Staph epidermis, standard isolate with inhibition zone 40 millimeter, and Escherichia coli also standard isolate with inhibition zone 41. And the smallest inhibition zone was for Pseudomonas, Proteus mirabilis and Acinetobacter baumannii with 20 and 19 millimeter. Garlic juice with 100%, the most potent inhibitory activity was recorded for Escherichia coli, the standard strain, and Staph epidermis with 45 millimeter and 42 millimeter. Pseudomonas aeruginosa also gives a high inhibition zone with 42 millimeter. The Staph aureus clinical isolate also give inhibition zone or wide inhibition zone with 42 millimeter. While the smallest inhibition zone was recorded for Proteus mirabilis with 24.3 millimeter. These photos shows the inhibition zone of the 3 garlic juice concentration against various bacterial species. And 100%, and 2 500%, 25%, and 3, and 0 was sterile distilled water, which is used as a negative control. In this table, shows the MIC and MBC result for our bacterial strain. MIC values range from 3.1 milligram to 9.3 milligram. MBC ranges from 6.25 milligram to 12.5 milligram. The low MIC, MBC and MBC reflect the strong activity of fresh garlic juice against Staph aureus and Proteus mirabilis and Acinetobacter baumannii. This table shows the comparison of antibiotic activity between a fresh garlic juice with the 3 concentration and amikacin against the bacteria. As shown in this table, a fresh garlic juice demonstrated a strong concentrated dependent antibacterial activity, notably at high concentration 100 and 100. Garlic juice not only match the standard antibiotic amikacin, but actually outperformed it against several strains with highly significant statistical differences be less than 0.05, especially in Staph epidermis and Staph aureus standard strain. Conclusion, The broad-spectrum efficiency of Eliamistevim juice against both gram-positive and gram-negative pathogen, including multi-drug-resistant strain, underscore its potential as a source of novel antimicrobial agent. However, several limitations must be acknowledged. First, the research was conducted slowly as in vitro study, which may not reflect the in vivo effect of garlic juice against human infection. Second, we evaluated a limited number of bacterial strains. In addition, the chemical profile of garlic varies significantly according to geographic origin, cultivation condition, and extraction technique, which may impact the concentration of bioactive compound such as allicin. Our recommendation, therefore, we suggest that the future in vivo studies evaluate the effect of natural garlic juice against the broader range of a bacterial species, while our in vitro findings are promising. Further in vivo and toxicological study are essential to assess potential interaction with biological matrix and establish clinical safety and efficiency. Further, research should prioritize the isolation and characterization of the pure active constituent to provide definitive scientific evidence for their clinical application as alternative or syringetic antimicrobial therapies. Acknowledge, we would like to express our sincere gratitude to Al-Azhar University for providing the laboratory facilities and technical support necessary for this study. Special thanks are extended to Drug Toxicology Analysis Center in Al-Azhar University for their valuable assistance in providing standard bacterial isolates. We express our deep appreciation to the scientific committees of the Third International Conference of the Pharmaceutical Sciences for accepting our research and providing us with the opportunity to present this work.
Yes, thanks.
Thank you very much.
Yes.
And I hope that you are not boring.
Yeah?
I hope that you are not boring now because it is the last lecture in the conference.
Not the last. Mr. Ahmed Afifi, but who has told us that delaying his lectures to the last. Inshallah, we hope that's a good condition now for Ahmed Afifi to introduce his lectures. Thanks, Miss Ruhaifa. And Alhamdulillah, that you are good. You are still in Gaza, yeah?
Yes.
Yes.
We are still living.
Yeah. God blessing to keep you and others and all beloved persons. Anyone have a question? Dr. Hani Dakka, that's question or any attention? Dr. Hani, and briefly, please, if a question. Dr. Hani? Okay, I have one question, inshallah, a small question, Ms. Ruhaifa. That's because we have no times and prepare to ending the conference. That's garlic, yeah? Yes, okay. Garlic is have active ingredients. I think that it's known for most people. That's allicin. Yes, allicin is very deteriorated substance because in some research, I think that's dealing with it. So how to keep the antibacterial activity of allicin, because it's hydrophilic, more hydrophilic, and some lipophilic. Yes. How the conditions, because it's I think that's yes.
We try to keep the not allicin only all the the constituent of garlic with storing at 4 degrees centigrade and
yes. I think that's
in shaded container and do our research or experiment and we don't what's happened to allicin, we don't what's happened to any constituent. This is in my recommendation, our recommendation that this constituent must be measured to make the issue more scientific.
Yes. يعني some recommendation can be added for your recommendation. That's garlics when it make experiment for antibacterial should be prepared allicin and kept in fridge or in refrigerator under temperature less than 8 centigrade.
We write it at 4 degrees centigrade. We store it.
Yes. The second question and the small, inshallah, it's have biofilm experiment for antibacterial because now it's yes.
No. We don't work the biofilm.
Yes, because it's epidermidis that the biofilm is very. I think that Dr. Nahed and Dr. Nabil, that is very important. It's epidermidis for biofilm.
Of course.
Yes, Dr. Miss. Any question, friends? Okay.
Just maybe very small question, Dr. Muhammad.
Yes, Dr. Nahed, we cannot.
Dr. Ruhaifa, do you find a difference clearly in the effect of the garlic? That's a very small question. The effect of the garlic on gram-positive and gram-negative bacteria, do you find such a difference? That's the question.
Between the bacteria itself?
Yeah, the effect of the garlic, well, it is very clear. The effect of the garlic against the gram-positive and gram-negative bacteria. When you was manipulating like Staphylococci and Escherichia coli and the Klebsiella, do you find such, like Staph aureus?
Yes, but not notably. يعني مش كتير فرق كبير بين الستاف اوريوس او الجرام نيجاتيف والجرام بوزيتيف. بس there is a difference here at 100%. The inhibition zone of staph epidermis 42, but the inhibition zone of Escherichia coli was 32.
It is not significant.
Yes.
Okay. Yes. Thank you.
There is differences, but it may be not significant.
Not significant. Yes.
Okay. Thanks, Doctor.
Thanks,
Doctor. We now transfer to Dr. Nahed.
Thank you.
And inshallah, ya ختامها مسك.
Inshallah.
Dr. Nahed and Mr. Afifi.
Thank you so much, my colleague, Dr. Muhammad Talib. Now we will move to the last speaker, our colleague, Mr. Ahmed Al-Afifi from Ministry of Health, Palestine, the Central Laboratory. He is working at Al-Nasr Hospital. And he, inshallah, will talk to us about a topic entitled Antimicrobial Resistance Under Fire, High Burden of MDR, XDR, and PDR bacterial infections in Gaza hospitals during armed conflict. Please, Dr. Ahmed, the mic with you.
ظاهرة الشاشة عندكم؟
الشاشة ظاهرة والمحاضرة ظاهرة وضايل تعملها بس مشاركة. تمام. Go ahead.
الله يعطيكم العافية جميعا وسعدت بسماع صوتكم جميعا. بالنسبة
Thank you.
الانتيميكروبيال ريزيستنس اندر فاير high burden of MDR, XDR, and BDR bacterial infection in Gaza hospital during armed conflict, supervised by Dr. Nabil Laila and Dr. Abdul Hakam, with cooperation with Dr. Khalid Abu Laish. Antimicrobial resistance is one of the most serious global public health threats, limiting treatment options and increasing mortality rates. Period to October 7, 2023, the humanitarian situation in the Gaza Strip was already deep concerning. Armed conflict accelerated the emergence of spread of resistance bacteria through healthcare distribution and inadequate infection control. In Gaza, the ongoing war has severely damaged healthcare services, creating favorable conditions for infectious disease transmission. Previous studies in Gaza reported alarming levels of resistance among gram-negative bacteria, including carbamazepine-resistant pathogens, like studies for Dr. Nabil and Dr. Nahid. Overcrowding, limiting laboratory capacity, and extensive empirical antibiotic use may further worsen the antimicrobial resistance crisis. This study investigates bacterial epidemiology and the burden of MDR, XDR, and BDR organism in Gaza hospitals during armed conflict. As we know, MDR refers to acquired resistance to at least one agent in 3 or more microbial categories. XDR means non-susceptibility to at least one agent in all but 2 or fewer antimicrobial categories, and BDR refers to resistance to all agents in all antimicrobial categories. Our objective for this study is to characterize bacterial epidemiology, antimicrobial resistance pattern, and the burden of MDR, XDR, and BDR organism in hospitals across the Gaza Strip during armed conflict. Material and method, we study a multi-center retrospective study was conducted across 5 major governmental hospitals between the period from May and October 2025. These 5 hospitals include Rantisi Al Nasser Hospital, Al Shifa Medical Complex, Al Hilo Hospital, Al Ahli Hospital, and Al Aqsa Hospital. Rantisi and Al Aqsa were the 2 hospitals that provided the microbiological services for the patients from all Gaza Strip regions during the period of the study. Antimicrobial susceptibility testing was performed using CLSI. Isolates were classified as MDR, XDR, or BDR according to international criteria. Resistance patterns were analyzed by specimen type and hospital ward, and association with mortality were evaluated using odds ratio with 95% confidence intervals. The results, a total of 3,138 gram-negative isolates were identified from clinical specimens recorded in microbiological laboratory databases. Isolates were predominantly recovered from pus, urine, and sputum, as we saw here. Pus 48 and urine 36% and sputum 9.6%. Here we see that Al Aqsa Hospital take about 50%, and Al Aqsa as a hospital in the middle area of Gaza Strip, it is express the patients from the middle and the south area, because Nasser Hospital and European Gaza Hospital, there is no services for microbiology. But Al Shifa, Rantisi, Al Hilo, Al Ahli express the north area of Gaza. Here we see that the distribution of bacterial isolates, E. coli and Klebsiella were the most higher percentage, as E. coli were 40% and Klebsiella 25%, but others like Pseudomonas, Proteus, Acinetobacter, Enterobacter, Providential, Citrobacter, all are in a low percentage in this study. Here we see the percentage resistance among E. coli. As we saw here, all cephalosporins, even second or third generation cephalosporins, show a high percentage of resistance. For E. coli resist for cephalosporins in high percentage, but Klebsiella show more resistance, more and more resistance in this study. It is beginning from 65 and reach to 95%. And as we see, ciprofloxacin, cefoxytin, even fourth generation cephalosporins, a mix of families of antibiotics. Here, percentage resistance among Pseudomonas, anti-Pseudomonal become like gentamicin and ceftazidime, ciprofloxacin were the highest percentage. Here, percentage resistance among Proteus, also Proteus become more resistant than the previous years. As we see, ciprofloxacin 57, gentamicin 62, and raised until it reached to 94 for cefazolin. Lastly, percentage resistance among Acinetobacter, it is begin from meropenem, which is 54, and raised to reach ceftazidime 86%. Here, the prevalence of, as we see in MDR, XDR, and PDR, for this 5 major organisms, Acinetobacter was the most organism that make MDR for antibiotics, followed by Klebsiella and the Proteus, Pseudomonas, E. coli, as we see in this table. Here, the number of isolates become 3,000.40, not as we mentioned before, because we delete the others, which is low numbers for this. Here we saw that MDR, XDR, and PDR with different words. As we see, ICU in MDR and XDR and PDR show the highest percentage, followed by inpatient departments and neonatal ICU, then outpatient, and finally emergency department. In this figure, we saw the difference of MDR, XDR, PDR in different specimens. Here, pus in MDR was the highest number, but sputum in XDR show the highest percent. But PDR, there is no percentage. It is low percentage, especially here, mortality rate, as we saw here. MDR E. coli show 73% from died patient, which infected by MDR E. coli, followed by Klebsiella 68. But Pseudomonas aeruginosa, any patient infected with Pseudomonas and died, it has become 80% from died patients that infected by Pseudomonas. Proteus 59 and Acinetobacter 67. Here we saw that there is no PDR for Pseudomonas and Proteus, but E. coli and Klebsiella show high in percentage. It is 11 and 10%. Association of MDR, XDR, PDR status and patient mortality in comparison of not MDR and MDR. Here we saw that MDR show statistical significance and odd ratio 1.69. But XDR and PDR, there is no difference between XDR and not XDR, or PDR not PDR. And and this number is due to shortage in information and insufficient information about the patients, because I can just take information about ICU patients and some in ICU in RNTC patients, but others there is limitation about taking it due to some faculties to reach for this information. In conclusion, this study reveals a critical high burden of MDR, gram-negative infection in Gaza hospitals, characterized by substantial carbamazepine resistance and significant association between MDR status and mortality. The predominance of advanced resistance phenotypes, particularly in ICU setting and among Acinetobacter and Klebsiella species, underscores the severe therapeutic limitation. Urgent implementation of strengthened antimicrobial stewardship, enhanced infection prevention and control measures, and sustained international support is essential to mitigate the escalating antimicrobial resistant crisis in conflict-affected healthcare systems. And finally, I would like to thank you, everybody, and thank all conference team for this sharing, and thank you.
Thank you very much, Mr. Ahmed, for this nice presentation. And now we want to conclude with one or maximum two questions, please. Anyone has a question for Ahmed, for Mr. Ahmed? Okay, if no one has a question, just I want to finalize with one question. Mr. Ahmed, this is a retrospective study, I'm right?
That's right.
Yeah, and that means maybe you haven't the isolates that you have those results for them. I mean, do you have those isolates if you would like to do further investigations under the molecular level to diagnose the resistance genes that make ASBL or carbamazepine, or in terms of gram-positive MIC-A and so on? Or you haven't the isolates right now? You haven't?
I have just 300 isolates for these patients that's received to RNTC hospital, but others I take the information from their patient records.
And you just only worked with gram-negative, am I right?
That's
so no,
right.
you don't touch the gram-positive. Yes, these results again, I mean, make or introduce us to the very big problem in Gaza regarding the AMR antimicrobial resistance, which we are suffering a lot. Thank you so much.
There is a high difference between this result and the previous years.
Yes, I understand the rule of the war and what happened in the last 3 years. Actually, we would like to thank you very much, and also my sincere and extended thanks to Dr. Muhammad Talib.
Yes, Dr. Nahid, I have one comment and one ask question for Mr. Ahmed.
Please.
Yes,
please.
Because this is, I think that it's important point. I'm now supervised on a master degree in this point under the fire. Under the fire about resistance of antibiotics in Gaza Strip. I note that from pharmaceutical view, please, that not all antibiotics is standardized as before war. So ciprofloxacin under the war, not ciprofloxacin as before war, because we have the capability and accessibility and flexibility to make all tests from analytical and pharmaceutical to check the bioavailability and bioequivalence and percentage of active ingredient. So I think that is important points that taken in consideration. Comycin which entered Gaza Strip under the sun and which not illegal, and I think that's it's clear for the all that the medicine which entered Gaza in the war, it's not under the circumstances and rules and instruction of pharmacy administration as before war.
Also, Doctor, we faced the shortage in antibiotic discs. It is very important because, but we use a large number of samples to cover this shortage, and a large number of data from more than one hospital.
Yes, Mr. Ahmed. I think that's I speak about 2 points: the discs and the antibiotics which taken by the patients. Two problems. The disc is not entered as the same rules which before war, because the companies now all which know that's the corruptions in all fields in Gaza Strip. I think it's clear for the all, and Allah, inshallah, help in this phenomena, and it's disappeared it. But antibiotics which taken by the patient and the disc which enter and applied in vitro for resistance of bacterial is not standardized, is not the pharmacopoeial DS. This is my note. I think that it may be right, it may be wrong. Congratulations for these nice lectures, and very hopeful for Dr. Nahed.
Okay, Doctor. But in these days, we use ATCC isolates to test the antibiotic discs and to ensure that it is no problems during storage or during transferring from other countries to Gaza.
Yes. I know. This hints.
Also in this period of study, there were no services for microbiology just in شهداء الأقصى Hospital and الرنتيسي.
I know, Mr. Ahmed. I know.
In 2025, just in الرنتيسي and شهداء الأقصى Hospital services for microbiology.
Yeah, I know. This is a challenge for my for my supervision for my students. No problem.
I'm head of microbiology department, Doctor, in Al-Rantisi.
I know. Inshallah, we will discuss on the special ways by you. And finally, Dr. Nahed, microphone with you. Should be finalize the conference with the big boss, Dr. Nahed.
No, I am just a little colleague and a friend for
all.
No. Thank you so much, Dr. Muhammad, Dr. Ihab, Dr. Muhammad Taha, Dr. Hani Dakka, everyone here, Mr. Ahmed Afifi, for all your efforts and also the previous brothers and friends who presented. Just one sentence confirming what Dr. Muhammad Talib said about the antibiotic resistance and the cartridge of discs that we used, Ahmed. The last collection that I bring, or we brought, me and Nabil, Dr. Nabil, to Belgium, to France here to work with, we found a big discrepancies between the AMR results in Gaza and the AMR results here, and also the genus species identification in Gaza and here. Not because we are not good. No, no, no. We are excellent. Our MTs are very excellent. But the war, and even before the war, there was siege for 18 years.
Yes.
So we are very excellent as a human resources, but we haven't equipments and instruments. We are working with 16S ribosomal RNA, with MALDI-TOF. We haven't these things in Gaza. That's all.
Yes. That's 100%.
So I will let the microphone for the organizers now, the scientific committee, the head of the conference, and all nice people there. Thank you so much.
And you forgot Dr. Nahid, Dr. Nabil Aila from congratulations. And Doctor, yes, no, this is good. Yeah. Dr. Nabil, many congratulations.
Thank you.
He's my big brother.
Thank you.
Thank you, Dr. Muhammad Talib. Thank you, Dr. Nahid. So as we come to the end of the scientific sessions of the Third International Conference on Pharmaceutical Sciences, we would like to sincerely thank all our speakers, researchers, participants, and attendees. Your valuable participation has made this conference successful and inspiring. It is now my pleasure and honor to invite Professor Ihab El-Masri, Chairman of the Organizing and Scientific Committees, to present the conference recommendations and to summarize the keynote, the key outcomes of this conference. Professor Ihab, the floor is yours.
Thank you, Dr. Loreen. Thank you all, distinguished guests, esteemed colleagues. We have reached the conclusion of the Third International Conference on Pharmaceutical Sciences. Over the past 2 days, we have witnessed pioneering research, inspiring keynotes, and invaluable academic exchange. I want to express my deepest gratitude to our keynote speaker, presenters, session moderators, and participants, and everyone who made this event possible. Advancing pharmaceutical discovery and clinical innovation in a digital space requires a huge collaboration, and today's success is evidence on the collective effort. My sincere thanks to the organizing and scientific committee members, to our IT unit and media department. Finally, many thanks to all attendants for their active participation and insightful discussion. Now to the recommendation of the third conference. This conference is not just about discussion, it is about action. Based on our sessions and presentation, the organizing committee has summarized the most critical and important recommendation. The first one is to promote the international collaboration to restore resilient and sustainable pharmaceutical service in Gaza. Strengthen involvement of pharmacists in antimicrobial stewardship is essential to mitigate the global impact of antimicrobial resistance. Third, strengthen pharmaceutical supply chain with real-time inventory monitoring and emergency stock management. Fourth, enhancement nutritional surveillance and emergency nutrition response system in humanitarian settings such as Gaza by integrating regular nutritional assessment and culturally appropriate dietary support into primary healthcare services to mitigate the health consequences of food insecurity and a prolonged crisis. Implement routine nutrition screening and targeted interventions for high-risk populations, including women of reproductive age, pregnant women, and patients with chronic diseases such as chronic kidney disease. Strengthen the role of clinical pharmacists in decision-making in hospital, especially during devastating situations such as the genocidal war in Gaza, which is vital to ensure faster, safer access to life-saving therapeutics and will attenuate medication-related problems, MRB. Moreover, increase the awareness of the importance of pharmacogenomics and pharmacovigilance as important and helpful tools for decreasing MRB and increasing treatment success rate. Also, prioritize patient-centric treatment. Future research should heavily focus on personalized treatment and advanced drug delivery system to improve patient compliance and treatment outcomes. Also, increase the awareness and accelerate the integration of the AI tools in healthcare system as diagnostic tool, as well as in treatment. Moreover, we must prioritize artificial intelligence and machine learning in early stage drug discovery in order to reduce the development timelines and increase the success rate. Also, enhance research in pharmacovigilance and drug safety through the integration of experimental, computational, and clinical data to improve the early detection and the prevention of adverse drug reaction. Lastly, encourage continued research into innovative therapeutic strategies, including natural product, targeted therapies, and drug repurposing to improve the outcome across wide range of disease. Thank you all for your attendance again, and the mic to Dr. Lori.
Thank you. I highly thank you, Professor Ihab Al-Masri. So now we are listening to Dr. Muhammad Taha, the Dean of the Faculty of Pharmacy, to close the conference.
Hello, everyone. As we come to the close of the third International Conference on Pharmaceutical Sciences, we reflect on 2 inspiring days filled with outstanding scientific presentations, engaging discussions, and meaningful exchange of knowledge among experts from around the world. On behalf of the organizing committee, I would like to express our sincere gratitude to all our esteemed session moderators for their dedication and excellent moderation throughout the conference. I also extend my heartfelt thanks to our respected keynote speakers, presenters, researchers, and participants for their valuable contribution and presentation, all of which have greatly enriched our conference. We also extend our deepest appreciation to all members of the conference committees, the media and public relations department, and the IT department, and everyone who worked behind the scenes to make this conference a success. And also, I would like to offer special thanks to the engineering Rami Nafi for his outstanding effort in designing the conference website, and to engineering Ahmed Dahouk for his exceptional work in the technical preparation and management of the conference activities through the past 2 days. Also, I would like to convey the warm greetings and appreciation of the president of our university, Professor Omar Milad, who had hoped to be with us today, but regret that he is unable to attend due to a prior commitment. We are pleased to inform all esteemed keynote speakers, participants, researchers, and session moderators that their certificates of participation and appreciation will be sent to their registered email addresses shortly after the conference. Finally, we look forward to welcoming you again at the 4th International Conference on Pharmaceutical Sciences, which we hope will be held in person at our university, Al-Azhar University of Gaza, to meet you face to face and continue our scientific collaboration. And finally, please, can you just turn on or activate your camera to take a photo? Thank you, and thank you again. دكتور ناهد، باشمهندس أحمد.
معك. Yes, I'm here.
ممكن الكاميرا لو سمحت؟
أنا فاتح الكاميرا، بعرفش ليش مش طالعة يعني. ما فيش صورة، مش عارف.
لا ما في صورة.
واي.
دكتور باشمهندس أحمد الكاميرا مقلوبة.
اللابتوب قلبت.
طب لا اقلب اللابتوب.
I don't know. I open it. It's open all the time. خلص يا سيدي، الله يحفظكم. هاي الباشمهندس أحمد، وهي إيهاب، وهي الدكتورة لورين.
بجهودكم جميعا.
هاي الدكتورة مي. حياك الله دكتورة مي، شفناك على خير.
Okay, thank you all and see you soon in Gaza. إن شاء الله.
إن شاء الله.
يا رب يا رب يا رب.
نلقاكم على خير.
شكرا.
جميعا.
فمان الله دكتور نبيل، فمان الله دكتور إيهاب، فمان الله دكتورة لورين، دكتور محمد، دكتورة مي.
لكم جميعا.
حبيبنا دكتور ناهد.
دائما
الله يحفظكم ربنا، الله يقدركم جميعا. الله يحفظكم. السلام عليكم. فمان الله.
على مؤتمر القادم بإذن الله إن شاء الله.
يا رب يا دكتور محمد، يا رب يا رب يا رب.
في غزة، في رحاب جامعة الأزهر إن شاء الله.
اللهم آمين. الله يسمع
يا
منك. الله
يا رب يا رب يا رب
يا رب.
تحيات. السلام عليكم.
أنا علقت في أولها مش عارف ليش.